Nah, selesai.
Kenapa? Sehat. Sambet kali.
Ayo, ikut aku.
Mau mana?
Udah, ikut aja.
Di sini.
Iya, di sini.
Mau ngapain?
Udah, daripada katanya sosmed, mending ikut kuliah tamu imunologi.
Yuk.
Baik, hari ini kita akan bahas topik tentang autoimun. Kasus kanker itu juga merupakan salah satu dari autoimun, ya. Nah, dan apa penyebabnya dari autoimun? Dari apa yang sudah saya jelaskan tadi, ada yang mau ditanyakan kira-kira?
Ini kan membahas tentang autoimun. Nah, kalau misalnya tentang depresi, ada tidak hubungannya dengan autoimun? Makasih, Prof.
Cocok sekali ya, kebetulan di kurikulum kami ada kurikulum tentang psikoneuroimunologi. Jadi keterkaitan antara sistem imun dengan psikis.
Izin Prof bertanya. Nama saya Astika Juniansari. Apa benar Prof, makanan itu penyebab autoimun? Terima kasih.
Makanan itu apakah bisa merupakan salah satu penyebab autoimun ya? Bisa. Ya, kenapa? Karena saat ini makanan-makanan modern sekarang ya, itu banyak yang artifisial.
Seru juga ya belajar imunologi. Bener banget, masuk imunologi aja. Lagian karena ini multidisiplin ilmu, bisa dari beragam keilmuan kok, nggak harus melulu kesehatan.
Bisa jadi bidang kesehatan bukan passion saya, tapi dunia imunologi begitu menggoda. Tidak hanya tentang keilmuan yang luar biasa atau karena multidisiplinnya, tapi dengan imunologi, aku, kamu, dan kita bisa berbuat yang terbaik untuk orang-orang tercinta.
Indonesia memiliki mimpi besar menjadi Indonesia maju di tahun 2045. Sekolah Pascasarjana Universitas Airlangga memiliki 8 program studi multidisiplin, hadir menjadi institusi berkelas dunia dengan kemampuan untuk mengembangkan pemimpin-pemimpin transformatif yang berdampak bagi dunia. Selamat datang di Sekolah Pascasarjana Universitas Airlangga, dan inilah program studi yang kami kelola.
Saya Profesor Dr. Teresia Indah Budi sebagai Koordinator Program Studi Magister Imunologi Sekolah Pascasarjana Universitas Airlangga. Prodi Imunologi merupakan satu-satunya program studi Magister Imunologi di Indonesia. Prodi kami ini adalah multidisiplin, jadi bisa dari berbagai keilmuan. Sekolah-sekolah yang berkaitan dengan imunologi bisa mengikuti program studi imunologi di Universitas Airlangga. Kami punya kompetensi, yaitu bahwa mahasiswa-mahasiswa yang mengikuti program studi imunologi ini bisa langsung terjun di dalam penelitian-penelitian yang mengikuti program dari penelitian dosen-dosen yang ada, sehingga mahasiswa semakin mempunyai wawasan yang semakin banyak di bidang imunologi. Dan juga kami juga menyelenggarakan salah satu seminar yang kita sebut sebagai World Immunology Day, merupakan satu-satunya juga seminar yang kita selenggarakan, baik itu tingkat nasional maupun internasional.
Kita disupport dengan adanya penelitian berpayung, pendanaan yang support luar biasa dari Pascasarjana Universitas Airlangga. Kita juga sebagai mahasiswa mendapat support untuk publikasi dan juga melalui pelatihan-pelatihan berkelas fokus dari Q1 sampai Q4.
Karena imunologi itu banyak bertanya. Itu PR-nya mahasiswa online harus lebih aktif dibandingkan kalau mahasiswa offline, dosen ada di depan kita dan kita.
Ladies and gentlemen, a delightful morning here at Universitas Airlangga in Surabaya, Indonesia. We are truly honored to have you join us in the 4th Airlangga World Immunology Day 2025 International Conference held by the Postgraduate School of Universitas Airlangga with the theme of Immunological Frontiers in Infectious Diseases from Molecules to Translational Applications, where today's conference will bring together exciting speakers from all around the world. Please allow me to introduce myself. My name is Nabil Ahmad Wafiuddin, where today I have the immense privilege of serving as your MC for today's conference. Without further ado, let us extend a warm and enthusiastic greeting to all of our esteemed and honored guests in attendance with us today. The Honorable Director of the Postgraduate School of Universitas Airlangga, Professor Badri Munir Sukocho, herewith is represented by the Vice Director for Human Resources and Information, Professor Sri Panca, and also the Vice Director for Research, Internationalism, Digitalism, and Information, Professor Suparto Wijoyo. The Immunology Magister Program Coordinator of the Postgraduate School of Universitas Airlangga, Professor Theresia Indah. Our expert conference speakers here with us today, Professor Karina Citra, Professor Cita Rosita, Dr. Eugene Ong, Dr. Arif Nur Muhammad, Dr. Pramila Rijal, the lecturers of Universitas Airlangga, our moderators for today's conference, Dr. Vivin and Dr. Ayu, the organizing committee of the Airlangga World Immunology Day 2025, and all of the participants of the 4th International Conference of the World Immunology Day 2025, whether you are joining us here offline at Universitas Airlangga or if you're joining us here online, we wish you an exciting conference ahead of us. Please allow me to read the rundown of today's conference. First, we will start by singing the national anthem of Indonesia, followed by the university anthem. We will then hear an opening speech by the program study coordinator and the director of the Postgraduate School of Universitas Airlangga, herewith will be represented by the vice director. We will then have a group photo session and then dive deep into our expert lectures that will be brought by our speakers today. We will then have a break time and poster presentation that will be conducted at the same time, and we will have lunch. After that, we will have our fifth speaker before going into our oral presentation on the top oral presentation that will run parallel in the main room, room 1, room 2, and room 3, followed by a jury assessment. After that, we will have an award ceremony followed by the closing. Now, we kindly request everyone to please rise for the national anthem of Indonesia, followed by our university anthem. To all the participants and distinguished guests, please rise. Indonesia Raya. seated. Thank you. Now, it is our pleasure to introduce our esteemed speakers who will deliver the opening speeches of the 4th International Conference of the World Immunology Day 2025. We will begin with Professor Teresia Indah, the Immunology Magister Program Study Coordinator of the Postgraduate School of Universitas Airlangga. To Professor Teresia, the stage is now yours.
Thank you, MC. Okay. Dear colleagues, professors, lecturers, researchers, esteemed speakers, ladies and gentlemen. Good morning. The Honorable Director of Postgraduate School, Universitas Airlangga, Professor Badri Munir Sukoco, hereby represented by the Vice Director for Research Internationalism, Digitalism, and Information, Professor Suparto Wijoyo, and Vice Director for Research and Information, Professor Panca Madiawati. Good morning and welcome to the 4th Airlangga World Immunology Day. Welcome, Prof. Zita. International Conference. We are honored to have with us Prof. Dr. Karina Dewiju, PhD from Universitas Airlangga, the expert in vaccine and one of the maker of the COVID-19 vaccines. In addition, we are privileged to welcome Dr. Pramila Rijal, PhD from Oxford University, an expert in antibody development. Equally deserving for admiration and gratitude, Prof. Dr. Cita Rosita Sikit, welcome. As Director of Sutomo Hospital, expert in clinical and policy hospital. Furthermore, it is a great honor to have Dr. Arif Nur Ansori from Kumamoto University. Among us, an expertise in bioinformatics, computational virology, immunology, and molecular biology. And also we have Dr. Eugene Ongbun Beng from University Science Malaysia. Expertise in infection disease, especially leptospirosis, molecular and structure biology, diagnostic, mutagenesis, recombinant DNA, and medical microbiology. And to all our participants, I offer my sincerest appreciation for your patience, curiosity, and dedication to advancing the field of immunological mechanism infection disease. And I would also like to thank the participants who attended the hands-on workshop on June 30 at Institute of Tropical Disease, Universitas Airlangga, with them unlocking gene expression using real-time PCR, best practice, and trust-based setting. It is my great pleasure to welcome you to this International Conference on Immunological Frontiers in Infection Disease. From molecular to translation application, as the head of Immunology Study Program, I am thrilled to see our collective expertise gathered here today to explore the latest advances in immunology and infectious disease. The theme is conference is particularly significant. It is highlights the critical role of immunology in understanding the complex interaction between the immune system and pathogens, from the molecular mechanism underlying immune response to translation application that can benefit human health. Our conference will delve into the immunological frontier in infectious disease, covering topics such as molecular immunology, immune regulation, and vaccine development. We will also explore the latest translational application, including immunotherapy and immunodiagnostic. I'm confident that this conference will provide a platform for knowledge sharing, collaboration, and innovation, and will inspire new ideas and approaches to tackling infectious disease. Last but not least, my deepest gratitude goes to the advisor board, organizing committee, scientific committee, institution, company, and volunteer who have directly and indirectly supported the success of this conference. Your contributions are invaluable, and I have so doubt that insight shared here will shape the future and healthcare for generations to come. Let us embark on this exciting journey of knowledge sharing and exchange and work together to advance our understanding of immunological frontier in infection disease. Thank you and enjoy this exciting conference.
Thank you, Professor Teresia, for your inspiring words. And before that, we would like to extend our warmest welcome to our esteemed speaker, Professor Cita Rosita. Welcome to this conference, and we hope you enjoy today's exciting conference. Next, we invite the Director of the Postgraduate School of Universitas Airlangga, Professor Badri Munir Sukocho. Your work will be represented by the Vice Director for Human Resources and Information, Professor Sri Panca, to please give her opening speech. To Professor Panca, the time is now yours.
Thank you, Mas Nabil. Assalamualaikum warahmatullahi wabarakatuh. Good morning, dear college, professor, lecturer, esteemed speaker, and ladies and gentlemen. It is my great pleasure to welcome you all to this fourth World Immunology Day 2025 International Conference on Immunological Frontiers in Infectious Diseases from Molecular to Translational Application. As the Vice Director in Resources and Information Postgraduate School, I'm honored to see our institution hosting such as a prestigious event that brings together experts and scholars from around the world, such as Professor Dr. Karina Dewi Ju from University of Oxford, Dr. Pramila Rijal from Oxford University, Professor Dr. Cita Rosita Sigit Prakuswa from Sutomo Hospital, Dr. Arif Nur Amsori from Kumamoto University, and Dr. Eugene Ong Bon Beng from University Science Malaysia. World Immunology Day 2025, organized by the Master of Immunology program, also held a workshop activity on June 13, 2025 at the Institute of Tropical Disease, Universitas Airlangga. The workshop activity had the theme Unlocking Gene Expression Using Real-Time PCR, Best Practice and Troubleshooting. As the Vice Director of the Postgraduate School, I would like to thank 61 participants from various institutions, such as Hiroshima University, Universidade Nacional Timor-Leste, University of Global Health Equity, Universitas Airlangga, Brawijaya University, Gajah Mada University, and so on. This conference showcases the cutting-edge research and advancement in immunology and infectious diseases. And I'm confident that the discussion and presentation will be enlightening and thought-provoking. All participants present will greatly benefit from the insight shared by our esteemed speakers. And I'm excited to see the potential collaboration and partnership that may emerge from this event. The theme of this conference is particularly relevant in the world where infectious disease continue to pose significant challenge to global health. Understanding the immunological mechanism underlying these diseases is crucial for developing effective prevention and treatment strategies. Once again, I would like to express my gratitude to our speakers, participants, organizers for their hard work and dedication in making this event a success. I'm confident that this conference will not only advance our understanding of immunology and infectious diseases, but also foster a spirit of collaboration and innovation that will benefit our community and the world at large. Last but not least, I welcome you all to this international conference and wish you an enriching experience, and I wish you all very good health. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you, Prof. Panca, for your inspiring opening speech. Ladies and gentlemen, the next agenda is a bestowance of a token of appreciation to our speakers today. We would like to invite to the front the Honorable Director of the Postgraduate School, hereby represented by the Vice Director for Human Resources and Information, Prof. Panca, and the Vice Director for Internationalism, Research, Digitalism, and Information, Professor Suparto Wijoyo, as well as the Magister Program Study Coordinator for Immunology, Professor Theresia, to the front to bestow upon the token of appreciation to our speaker today. We would like to also invite our speaker, Professor Chita Rosida, to the front as well. To our esteemed guests, please come to the front. To our honored guests, please remain standing as we will now have a group photo session together with the participants. So to all the participants, please rise. And for those participants sitting on this side, please kindly rise and move to this side. We will now have a group photo session. We will now have a group photo session with our esteemed guests. So to all of the participants sitting on this side, please kindly move to the right side of the conference room. For the participants in the back, you may please kindly move forward so that you can be included in the group photo. All right, smile and look at the camera. 1, 2, and 3. Thank you to our honored guests and participants who may now return to your seats. Thank you, ladies and gentlemen. The next agenda is now a series of expert lectures that will be given by our expert speakers. But before that, I would like to announce that throughout today's conference, a series of door prizes will be given to some of our lucky participants. So please, to all our participants, stay active, stay tuned, and stay focused, because here's the catch. At the end of today's conference, there will be a quiz. So prepare yourselves for that one. Now, we will now begin our lectures, where the first and second lecture will be moderated by our moderator, Dr. Niluh Ayu. Before I hand this session off to our moderator, please allow me to briefly introduce the moderator to you all. Dr. Ayu is a leading expert in infectious diseases, virology, and molecular epidemiology. She is a lecturer here at the Postgraduate School of Universitas Airlangga and a collaborative researcher between Kobe University and the Institute of Tropical Disease at Universitas Airlangga. She holds a PhD in medicine and a master's in tropical medicine with a strong foundation in science and nutrition. She has been recognized for her contributions, and her expertise bridges research, education, and global collaboration in tropical medicine. Ladies and gentlemen, please join me in welcoming Dr. Ayu as she moderates today's insightful discussion. To Dr. Ayu, the time is now yours.
Thank you very much, Mr. Nabil. Good morning, ladies and gentlemen. It is such a great pleasure for us to have you join the World Immunology Day International Conference 2025. My name is Ayu Megasari, and I will be moderating the first session of this conference. Okay, as for the opening speaker, I'm pretty sure that many of us has already known her. So is there anyone in this room who have received the Oxford AstraZeneca vaccine during the COVID-19 vaccination program? Can you raise your hand? Ada yang mendapat vaksin AstraZeneca di ruangan ini? Oh, okay. I think several of us got the AstraZeneca vaccine, and I'm pretty sure that you who has who got the vaccine already known our first speaker. So, well, our first speaker is one of the co-researcher of the aforementioned vaccine. And before she deliver her lecture, I would like to give a little bit of introduction. The CV, please. Okay. So our first speaker is Professor Karina Citra Dewijo, and she received her PhD from the RMIT Australia in 2019. And then she was honored with professorship from Universitas Airlangga. And Prof. Karina Jo has published many academic articles of her expertise, which has been cited by more than 11,000 times. Can we give her a big applause? Professor Karina Jo has extensive theoretical and practical knowledge of all aspects of vaccine development and process development for large-scale CGMP manufacturing, and she has contributed to several vaccine development and commercialization, including the rotavirus vaccine, rabies vaccine, and the well-known COVID-19 vaccine. Ladies and gentlemen, please welcome Prof. Karina Dewi Jo. Good morning, Professor Karina.
Good morning. Thank you for inviting me. Maybe I will start with sharing my screen.
Can you hear?
I can hear you. Can you hear
Can you
me?
hear us, Professor Karina?
I can hear you. Can you see my screen? Can you hear me?
Okay. We have a little bit of technical difficulties.
Can you hear me?
Professor Karina, can you hear us?
I can hear you. Can you hear me?
Mm. Professor Karina, can you hear us? We cannot hear your voice here. Maybe we will short the problem a little bit. Your voice is not very clear, Professor Karina. We cannot hear you here. So is it possible if you can? Would you try to talk now, Professor Karina?
Hello, can you hear me? Can
We
you hear
still
me?
cannot hear.
I'm talking. Can you hear me? Check,
We
check.
still cannot hear you. We're very sorry. Maybe you can say hello to us when you are ready, Professor Karina. We still cannot hear your voice.
Hello, hello, hello.
Hello.
My audio is working. Hello, hello, hello.
Okay, please wait a little bit.
Hello, 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 hello.
Would you mind to wait a bit, Professor Karina? We will try to short the problem.
Okay.
Would you mind to talk again, Professor Karina?
Hello? Hello, can you hear me?
Okay. Professor Karina, we are very sorry. We are currently still trying to solve the problem. Okay. Would you mind if we change your session with the second speakers first, because our Zoom sound system cannot catch your voice? Is that all right, Professor Karina? So maybe your session will be pushed back around 45 minutes.
Okay.
Okay, now we can hear your voice, Professor Karina.
No, it's working?
Oh yeah, now it
Yeah,
is.
okay.
Mhm.
Okay, let me share the screen again.
Okay, so you will have 30 minutes to deliver your lectures. And when the time is running out, I will hit your chat box. Is that all right, Professor
Yeah,
Karina?
it's all right. Okay.
Okay. Okay, then the time is yours, please.
Yes, good morning, professor, researchers, lecturers, and students attending the World Immunology Day today. Thank you for inviting me as a speaker. We will be diving into topics that both fascinating and urgent. So it's about emerging infectious diseases, or EIDs, which is a critical global health concern due to their potential to cause widespread harm. So next. So what is exactly are EIDs? So these are pathogens that have either newly appeared in the populations or are re-emerging after a period of decline, often crossing from animals to humans. So it's diseases like SARS-CoV-2, which triggered the COVID-19 pandemic, uh, monkeypox, which its recent outbreak, avian influenza such as H5N1, with its worrying potential for human transmission, and Ebola, which is notorious for its high fatality rates, which are prime examples. So what makes EID so dangerous is their ability to spread rapidly and overwhelming the healthcare systems and leaving us scrambling to respond. The consequences are profound. They lead to high morbidity and mortality, meaning they cause significant illness and death. But it's not just health. So think of the economics and the fallout from the COVID-19. So with soaring healthcare costs, trade restrictions, and workforce disruption, it ripples across the economies. And to understand why EIDs are on the rise and what we can do about them, so we will explore like a key factors driving this threat and their implication for our connected world. Next. So now we have established what is emerging infectious disease and their devastating impacts. So let's explore why they're becoming more frequent. So the major drive is the zoonotic spillover. So about 60% of the EIDs originate from animals. They're jumping from species like bats or birds to humans. And we've seen with diseases like Ebola or H5N1, the World Health Organization estimates there are 1.7 million undiscovered viruses lurking in mammals and birds. So that's a staggering reservoir for potential threats just waiting for the right conditions to emerge. So the climate change is another culprit. So it's reshaping the ecosystems and expanding the habitats of the disease-carrying factors like mosquitoes, which spread Zika and dengue to new regions. And then there's a global connectivity. So think about how quickly you can hop on to the plane and then cross continents. That's the same speed allows pathogen to spread from the local outbreak to a global pandemic in days, meaning that an outbreak anywhere can become a crisis everywhere. So these factors, animal-human transmissions, and the environmental shift, and our interconnected world are fueling the rise of the EIDs. So to illustrate this, and there's a showing the virus leaping from the animals to the humans. It's setting the chain of the transmissions. About how do we stop these diseases? The vaccines are actually the critical weapon, yet they face the unique challenges in development and deployment. So we'll talk about it in a little bit more detail later on. So emerging infectious disease evolve rapidly, and they are undergoing swift mutations that enable immune escape. Effectively, they're dodging our body's neutral defenses. So this antigenic variability, so is where pathogens frequently alter their surface proteins, create significant hurdles for the vaccine development. So the challenge is to design the vaccines that remain effective against constantly changing strains. And moreover, the limited cross protections between pathogen variants restrict the ability to achieve the broad, long-lasting immunity, leaving the population susceptible to new or mutated strains. So the effective vaccines must engage both the innate and the adaptive immune systems to establish the robust immune memory. So will enable the body to quickly recognize and combat future infections. So thankfully, the cutting-edge platforms are tackling these challenges by harnessing innovative technologies such as mRNA, the viral vector, and also the nanoparticle-based vaccines to develop solutions that keep pace with rapidly evolving pathogens and deliver broader and more durable protections. So traditionally, vaccines such as live attenuated and inactivated types used for diseases like smallpox. So they have a proven track record, but often required years of meticulous development to balance the safety and the effectiveness. But modern platforms, including mRNA, the viral vectors, and nanoparticle-based vaccines, have transformed this landscape by accelerating development timelines and generating the robust targeted immune response. So while the traditional vaccines could take a decade to reach the public, so modern platforms like mRNA have reduced this to months and vividly demonstrated by the rapid deployment of the COVID-19 vaccines. So this unprecedented speed was a game changer, showcasing the power of the innovative technologies to combat the global health crisis. So I will see this cutting edge platform further to understand how they are revolutionizing the vaccine development and paving the way for the faster and more effective response to EIDs. Firstly, the mRNA vaccine. So it builds on the evolution of the vaccine platform. It represents a groundbreaking forward in immunization technology. So this vaccine functions by delivering mRNA that encodes the pathogen's antigens, essentially providing cells with a precise recipes to produce the specific proteins, which mimic the pathogen, thereby triggering a robust immune response without causing the disease. So this mechanism allows the immune system to recognize and prepare to combat the actual pathogen. So if encountered later, for example is the Pfizer BioNTech and the Moderna vaccines for COVID-19, which were developed in under a year. So it's very fast. It's an unprecedented achievement that showcase the speed of mRNA technology. So where vaccine design can be completed in mere days. So this rapid development is paired with scalability for global production and the ability to elicit strong T cell and B cell response, which is critical for both immediate and the long-term immunity. So the mRNA platform is flexible, which allows scientists to quickly adapt to the genetic code to target new pathogen variants, making it a powerful tool against rapidly evolving threats like EIDs. And this highlight why mRNA vaccines have become a cornerstone of the modern vaccinology. Next is the viral vector vaccines. So these vaccines operate by using a modified harmless viruses like a Trojan horse to deliver antigen encoding DNA directly into the host cells. These prompts produce pathogen-specific proteins that stimulate a robust immune response. So the prominent example is the Oxford AstraZeneca vaccine, which employs an adenovirus-based vector to protect against COVID-19. So this platform excels in generating the strong T cells and the B cell response, often achieving durable immunity with the potential for single-dose efficacy. This simplify vaccination campaigns. So, and the most important hurdle is the pre-existing immunity to the viral vector, which can reduce the effectiveness in some populations. But by neutralizing the vector before it delivers its genetic payload. So these limitations requires a careful selection of vectors, such as using the rare adenovirus or the non-human adenovirus, which we use in Oxford AstraZeneca vaccine. We use the chimpanzee adenovirus instead of human. So continuing this journey through the cutting edge vaccine technology, let's turn to the next new coming technology, which is nanoparticles. Next is nanoparticle vaccine. These vaccines function by displaying the pathogen antigens on the surface of the engineered nanoparticle, which are designed to mimic the structure of the actual pathogens, thereby boosting immune recognition and the response. So the prime example is the Novavax COVID-19 vaccine. They use a protein-based nanoparticle to present viral spike proteins to the immune system. So this platform offers the significant benefits, including the exceptional stability that eliminates the need for stringent cold chain storage, making it particularly suitable for low resource settings where the refrigeration is limited. Additionally, nanoparticle vaccines elicit strong antibody responses, often surpassing other platforms in generating high level of neutralizing antibodies, which is critical for pathogen defense. Their designs allows for precise antigen presentations, optimizing the immune system's ability to recognize and remember pathogens. However, their production can be complex, requiring sophisticated manufacturing to ensure consistent antigen display. So this stability and potency position nanoparticle vaccines as a vital tool for the global health, especially in regions with logistical challenges. The next technology that is used in the vaccine development is structural biology, which rely on the precise antigen presentation to enhance the immune response. So we can use the cryo-electron microscopy, cryo-EM, which is a pivotal role in advancing vaccine development by providing the detailed structure into the pathogens. So the cryo-EM acts like a high resolution camera giving us a detailed blueprint of the pathogen surface by mapping the molecular structure of the near atomic resolutions. For example, the cryo-EM was instrumental in stabilizing the SARS-CoV-2 spike protein, enabling the design of highly effective mRNA vaccines, such as those developed by Moderna. So by revealing the precise architecture of the immunogenic site, the cryo-EM allows scientists to engineer antigens that target the most vulnerable parts of the pathogens. This elicits a stronger and more specific immune responses. This precision is critical for overcoming challenges like antigenic variability and immune escape, which ensure vaccines focus on the conserved, highly immunogenic region of the pathogen. However, the cryo-EM requires advanced equipment and expertise, making it resource intensive, though its impact on the vaccine efficacy is undeniable. So this technology has ability to provide structural clarity has revolutionized antigen design, as seen in the rapid development of COVID-19 vaccines. So looking ahead, the system immunology takes this precision further by integrating structural data with comprehensive immune profiling to optimize vaccine strategies against complex pathogens. So following our exploration of the cryo-EM, which provide detailed structure of blueprints for the pathogens to enhance antigen design, the system immunology takes this precision to the next level by leveraging the vast datasets to identify the optimal vaccines target. So this approach uses high-throughput sequencing to analyze immune response and pinpoint immunogenic epitopes. So specific regions of the pathogen that trigger strong immune response, just like we're looking at the diagram. So we will process a lot of data from the workflow of the system immunology. So from the data, we'll generate the prediction, and then from the prediction, we will acquire the design. So by integrating tools like AI and machine learning, the system immunology acts like a super smart assistant, predicting which epitopes are the most likely to elicit the effective, long-lasting immunity. So for instance, the AI-driven models have been used to identify conserved regions in the influenza viruses, enabling the design of the vaccines that target stable, less mutation-prone parts of the pathogen. So this method processes enormous data sets, genomic, proteomic, and immunological, to uncover the patterns that would be impossible to detect manually. So ensuring the vaccines are both precise and adaptable to evolving pathogens. So while the computational intensive and reliant on robust data, the infrastructure, this approach significantly enhances our ability to develop vaccines against complex variable diseases, and the impact is clear. The system immunology empowers the creation of more effective and targeted vaccines. So the next frontier in vaccine development aims to achieve the vital goal, so protecting against multiple strains or even entirely new pathogens. So this accomplished through innovative approaches like targeting the conserved epitope, the stable regions shared across pathogen variants, and using mosaic antigens that combine elements from various strains to broaden immunity. So the prime example is the universal influenza vaccine, which focuses on the hemagglutinin stock and the conserved region of the virus, offering a potential protections against the diverse flu strains, including the deadly H5N1 that could spark the future pandemics. The universal flu vaccines could stop the H5N1 pandemics by providing the shield that adapts to the virus evolutions, reducing the need for the annual strain-specific shots The conserved epitopes are the key, which enable the immune system to recognize and neutralize a wide range of strains, enhancing the broad spectrum immunity that transcends individual pathogen variants. So this strategy holds the promise for addressing the challenges of the antigenic variability and the rapid pathogen evolutions. So the next ambitious goal in vaccinology is to develop the single vaccines that capable of protecting against all coronaviruses. So the pressing need, given the global impact of the outbreaks like SARS, MERS, and the COVID-19, this innovative approach focuses on targeting the conserved S2 subunit of the spike protein, the stable region shared across diverse coronaviruses, which play a critical role in viral fusion with the host cells. Coronaviruses share structural similarities in their S2 subunit, offering a universal target that the immune system can recognize across species, from bat origin strains to human pathogens. So preclinical trials have shown promising cross-protection with animal models, demonstrating immunity against multiple coronavirus variants, including those with pandemic potential like SARS-CoV-2 and related bat coronaviruses. So this breakthrough could prevent future pandemics by providing a proactive defense, potentially saving millions of lives and reducing economic disruption caused by emerging threats. So this precision of strategy hinge on leveraging this conserved region to train the immune system effectively. So moving forward, the immune memory is critical as it ensures long-lasting protection and rapid response to any future coronavirus outbreak, building a resilient global health framework. So in the cornerstone of the long-term protection lies the role of memory T cells and B cells, which form the foundation of immunological memory. So these specialized cells are trained during the initial exposure to a pathogen or vaccines, enabling a faster and more robust response to re-exposure by quickly recognizing and neutralizing the threat before it can come and cause significant harm. So memory cells enable faster response by remaining in the state of readiness, with memory B cells producing antibodies and memory T cells coordinating targeted attacks, so often within days rather than weeks. So a standout example is mRNA vaccine. Pfizer-BioNTech, Moderna excel at inducing durable memory by effectively stimulating both T cell and B cell response. So this durability is enhanced by the mRNA platform ability to mimic natural infection signal, promoting long-lived plasma cells and the central memory T cells. However, the effectiveness of this memory depends on the vaccine's initial priming and the pathogen evolutions. So moving forward, translational challenges remain. So as scaling these memory-inducing vaccines globally while overcoming the production and distribution will be critical to realizing their full potential. So the significant hurdle is realizing these advances is the issue of manufacturing bottlenecks, which hinder the vaccine scalability. So one prominent example is the cold chain requirement for mRNA vaccines, such as those from Pfizer-BioNTech, which demand ultra-cold storage at temperature as low as -70 degrees. Marine stability posing logistical challenges, especially in the regions with limited infrastructure. So this dependency on the sophisticated cold chains increases costs and complicates distribution, particularly in the low resource setting. So the promising solution lies in the modular production system, which allows for the flexible, decentralized manufacturing to meet the global demands, and the development of the lyophilized or the freeze-dried vaccines that eliminate the need for the ultra-cold storage by remaining stable at standard refrigeration temperatures. These innovations are not only simplify productions, but also enhance accessibility by reducing reliance on the complex supply chains and enabling rapid scaling during the outbreaks. For instance, we can lyophilize the mRNA, which have shown the stability for months at 2 to 8 degree. So this is a game changer for the global equity. And moving forward, we have another issue, which is the equitable distribution and the affordability, which remain a critical barrier to ensuring the widespread vaccination coverage. So in a pressing global concern is the issue of the disparities in the vaccine distributions, which continues to threaten public health worldwide. So these inequities leave millions at risk, particularly in the low-income regions where access remains severely limited. So in mid-2021, only about 20% of the low-income countries had any meaningful COVID-19 vaccine. So it exposed vulnerable populations to ongoing outbreaks and variants. So this stark gap highlights how wealthier nations secured early supplies, leaving poorer regions behind. So a key example is the COVAX, which is an ambitious initiative launched to deliver 2 billion doses by the end of 2021 to ensure equitable access, especially for the low and middle income countries. However, COVAX faced significant logistical hurdles, including supply shortage, export restriction, and delays in funding and production, which fall short of its target with only about 1.1 billion doses delivered by early 2022. So these challenges underscore the complexity of global coordination and the need for robust infrastructure to support such efforts. So another critical barrier is the issue of the misinformations, which significantly reduces the vaccine uptake and undermines public health effort. So misinformation spreads faster than vaccine, with social media myths during the COVID-19 pandemic, like claims or severe side effect of the microchip implantations, gaining traction and eroding the trust, particularly in 2020 to 2021, when the uptake lacked in many regions. So the study shows the direct correlation with area experiencing with high misinformations, seeing vaccination rate drop by up to 20% compared to the well-informed communities. So to counter this, the effective solutions include the community engagement, where the local leaders and the healthcare worker build trust by addressing concern in culturally relevant ways, and providing transparency from the health authorities, providing clear evidence-based data on the vaccine safety and efficacy, such as the rigorous testing of the mRNA and the viral vector vaccines. So this strategy has proven successful, with pilot program in 2023 showing a 15% increase in the uptake in the targeted regions. So the World Health Organization's Global Influence Surveillance and Response System, or the GSRS, is the cornerstone of the global health security, established over 70 years ago to monitor influenza and other pathogens with pandemic potential, forming a network of over 150 national influence centers across 125 countries, alongside specialized labs and the reference center. So this system diligently tracks genetic mutations, antigenic shift, and the transmission pattern of the viruses like influenza, the SARS-CoV-2, and the emerging zoonotic threats like others, providing the real-time data critical for the vaccine development, diagnostic updates, and the outbreak preparedness. But collecting data is only the first step. Artificial intelligence takes this further by analyzing the vast data sets, including genomic sequences, animal host data, environmental factors, and human mobility patterns to predict and prevent outbreaks. The AI models, often powered by machine learning and deep neural networks, process complex multidimensional data to detect subtle patterns such as the genetic marker or the virulence or unusual spikes in the animal to human transmission, which might elude traditional surveillance. For instance, is H5N1, which is a highly pathogenic avian influenza. And the AI integrates historical outbreak data, the bird migration patterns, and farm density to pinpoint high-risk regions, enabling the GISRS to flag potential outbreaks weeks ahead of the traditional method, which saving times and the life through targeted containment vaccination campaigns and the public health alerts. So to see this action, consider the ISRS network, the global web of surveillance points, so the National Influence Center labs and the WHO or WHO collaborating centers, linked by the dynamic data flows, where each of the nodes collect samples, sequences, genomes, and report findings.
Professor Karina, you have only 3 minutes left.
Okay.
Sorry.
It's okay. So CEPI aims to produce vaccine in 100 days, a groundbreaking goal to deliver safe, effective vaccines against emerging infectious disease within just 100 days of identifying the pathogen genetic sequence. So this outpacing epidemics and saving millions of lives by compressing timelines that once took years. So this mission, a global call to action, relies on cutting-edge tools like pre-designed platform and stockpile antigens to achieve rapid response. So viral vector is one of the platforms that make this possible. Like those used successfully for the Ebola vaccines, deliver genetic instruction to our cell to trigger a strong immune response. And this proven with the COVID-19 vaccine rapidly encodes the viral protein for a swift adaptable defense, and this platform are pre-optimized and tested, allowing scientists to plug a new pathogen sequence, say from a novel virus, and scale production fast, a strategy honed through collaborative research. So I'm proud to share that my project at Oxford advanced this technology, refining how we adapt viral vectors to tackle emerging threats. So strengthening our ability to respond to unknown diseases like disease X or the future pandemics. So CEPI 100-day mission in the timeline. It kicks off with the pathogen sequencing in day 1 to 10. So by day 30, we use our platform-based vaccine design, leveraging the pre-existing viral vector system to craft a candidate, and we can do the preclinical testing on day 60, and by day 100 we push through clinical trials to approval, and fit made possible by pre-stock antigens and standardized protocol and global partnership. So future direction of the vaccine include AI and synthetic, with AI driving predictive antigen design and outbreak forecasting. So picture this, the AI selects the antigen pre-trials, harnessing advanced machine learning to shift through massive data sets, genomic sequences, protein structure, and immune response profile, and even climate and migration pattern to identify optimal antigens that spark strong, precise immunity before even step into the lab. And then AI forecast the outbreak, predicting where and when pathogen like coronavirus might surge by analyzing mutation trends and environmental shift and the global trend. Now we enter synthetic biology, pushing boundaries with engineered antigens through gene editing and protein design. We craft tailor-made antigens fine-tuned to trigger robust, adaptable immune response against fast-evolving virus. So we have the bold vision of synthetic biology could cover entire pathogen families, paving the way for pan-coronavirus and universal vaccines that shield us not just from today's strains, but from the future variants too, thereby revolutionizing our fight against unpredictable threats. Lastly, for conclusion, so we have learned about EID, the new resurging pathogens like SARS-CoV-2, monkeypox, avian influenza, H5N1, and Ebola. So it emerged from genetic spillover with 60% originating from animals. So WHO estimate 1.7 million undiscovered virus lurks and driving high morbidity. So this pathogen challenge us with the rapid evolution, antigenic variability, and limited cross protection. But modern vaccine platform rise to meet their demands, such as viral vector and mRNA vaccines, And tools like cryo-EM and map pathogen structures such as the SARS-CoV-2 spike protein, while system immunology use high throughput sequencing and AI to predict effective epitope for influenza and beyond. So we push through towards the broad spectrum protection, targeting conserved regions for pan-coronavirus and the universal influenza vaccines to tackle multiple strains. So thank you for listening to today's speak. If there are any time for questions, I will be glad to answer.
Okay, thank you very much, Prof. Karina. Give a big applause for Professor Karina Jo. And we have a little bit of time. So is there any question to Prof. Karina? For now, we have one question slot. You may raise your hand if you have any question. Okay, please. Please introduce yourself first and then the question.
Okay, thank you very much, Bu Ayu. Let me introduce myself. My name is Iqbal Aminullah Putra Ghazali. You can call me Iqbal. I'm from immunology student. Thank you very much, Prof. Karina, that you have delivered such an exciting and so much insight for us about the vaccine development. I want to ask you about that. What have you explained before about broad spectrum vaccine, especially in coronavirus? How about the efficacy? Is it better than the specific strain vaccine for corona, or what, or how? And in this, I want to ask about the efficacy. Is it the same with the specific spectrum vaccine, or yeah, something like that? And then the second question about me is about vaccine hesitancy. As you know, in Indonesia, there's so much people who still avoiding vaccine because maybe their background or something like that. As a vaccine developer, maybe you can give the tips and trick how to raise the people uh, awareness about the vaccine and how to reduce the vaccine hesitancy. Maybe that's all my question. Thank you.
Okay, so yeah, broad spectrum vaccines again the specific the strain. So yeah, there are some broad spectrum vaccines that are developed by Caltech. I think Pramila Rijal, Dr. Pramila Rijal is one of the researchers that contributes to this study by Caltech and Oxford. So they designed a mosaic nanoparticle presenting the spike protein receptor binding domain from the SARS-CoV-2 and 7 others of like beta coronaviruses. So if you're asking for the efficacy in the preclinical study of the broad spectrum, it suggests the broad spectrum vaccine may provide more consistent protection across the diverse variants, including those not directly targeted by inducing the antibodies against conserved viral lesions. But these vaccine have not entered any clinical studies. So yeah, they have designed it and then I think they have just published the clinical study. So we have yet to have any efficacy data in humans, unfortunately. So yeah, I mean, yeah, in the normal non-pandemic time, it will take time for these newly developed vaccines to enter the clinical studies. So the second question is about the vaccine hesitancy and how our researcher aim to handle this. Okay, so I mean, I mean, I will can only make a suggestion for everyone to take the vaccine because all the vaccines that is been commercialized have received the approval from the health regulator, of course, and then we already assess the risk and benefit of it, but we have to be transparent to all the consumer and the public about all the data that we have done in the lab and also in the preclinical and the clinical settings. So we will have to inform the efficacy how the study goes and what is the risk, yeah, for the developed vaccine or the drugs itself. So for me, it's just being transparent to the public and to develop the trust of the public.
Yeah, thank you very much, Prof. Karina. And we have one more question in the Zoom chat box. Is it okay if I read the question?
Please.
Okay, so we have a question from Miss Amana, and she would like to ask if that you mentioned the key to vaccine is the spike epitope, and will this spike epitope remain effective in immunocompromised individuals? And how about the other target epitopes such as the membrane or the nucleocapsid of the virus? And is there any difference in efficacy for people with immunocompromised?
So what is the other parts? The immune.
So the first one is
Sorry.
you mentioned that the key to the vaccine is the spike epitope. And will this spike epitope remain effective in immunocompromised individuals? And how about the other target epitopes, such as the membrane or the nucleocapsid? I think that is the first question.
Yeah, I mean, the all the spike. I mean, some of the vaccines use all the spike proteins, because they have not conduct the severe rigorous experiment before, so they use the whole spike protein, and then they use the templates before because it was related to another virus called MERS, and that vaccine is having the positive result in the phase 2 trial. I mean, in other settings, such as the nanoparticles that targeted the broad spectrum vaccine has used the RBD only. So RBD is located in the spike proteins and is the attachment of the virus into the cells. So binding domain, one of the binding domain. It is used in the Caltex mosaic vaccines. It also works too. Yeah, I mean, the spike protein can trigger some of response, include the memory T cells, and quite critical for the long-term protections. But actually, the efficacy is lower in immunocompromised groups because their ability to induce this memory T cell is compromised. Yeah, but it was still suggested for this immunocompromised group to take these type of vaccines, either made by RBD or the spike protein, because yeah, it still protects them from the severe case of symptoms from the COVID-19. What about the membrane protein and nucleocapsid? I think so far there's some preclinical vaccines that include all of these regions, but I have yet to see their publicized data. However, none of them reaches the clinical settings, so I will assume that those antigens is not the one who will trigger uh, the neutralizing antibody or the important T cells that will protect for COVID-19 infections. But I will have to see the data in the future if there's some publication that can prove that other antigens like the membrane or the nucleocapsid can trigger the protective immune response.
Okay, thank you very much, Prof. Karina. I think that answers the question from both our participants. So before we close the opening session of Professor Karina Jo, we would like to give a token of appreciation. So maybe Professor Teresia, as the program coordinator, can give a little bit of appreciation to Professor Karina.
Okay. Thank you very much, Professor Karina, for your supporting in the World Immunology Day. Yeah, maybe we hope in the next we can collaborate again. Thank you.
Okay. Thank you.
Okay, so this is a appreciation from the committee, and we will send a copy of this certificate to you later, Professor Karina. Thank you very much for your insight and experience in vaccine development. And we hope that the vaccine technologies can keep up with the evolving, emerging and re-emerging pathogens. And thank you very much, Prof. Karina. And once again, please give Prof. Karina a big round of applause. So that would conclude the first, the opening session. And before we are moving to the second speakers, there is a little bit of door prize to enlighten the session, the conference. So Mr. Nabil will lead the door prize.
All right. Thank you so much, Dr. Ayu. And once again, thank you so much to Prof. Karina for that insightful lecture. So as I said before, we will be handing out lucky door prizes to some of our lucky participants throughout the conference. And now I will announce the first group of lucky participants who have received this door prize. Now, how do we choose these participants? Well, in Indonesia, we have this saying called si paling pagi, or the early bird. Now, these participants are exactly that. These are the participants who have come so early, who have come very early before even us at the committee is even prepared for the conference. So without further ado, these are the winners of the first door prizes. Let's give it a second for there we go. So congratulations to Robbie Wijayanto, Faris, Rega, Rania Shafira, Calvin Skontatine, and Hanifa Rizki. So for these participants, you may contact the committee at the end of the conference to receive your door prize. Now, let me give you a little bit of a hint. One of the door prizes is a special voucher from a dental clinic that you can use to get a big discount, if I'm not mistaken, right, Prof? Yes, so that is one of the door prizes that you may be lucky wise to receive throughout the conference. That is it for my intermezzo. Back to you, Dr. Ayu.
Very much, Mr. Nabil. So congratulations for the door prize winners. And I think everyone should stay until the end of this conference because there are still many door prizes. Because if you leave the conference, then you will not be eligible to get the door prize. So if you want to get something back home, please stay tuned until the end of the conference. Okay, so we are going to move to our second speakers. So we have Professor Dr. Cita Rosita Sigit Prakuswa here, and her Prof. Cita speak TV. I think many of us already know her. And currently, Prof. Cita is the director of Dr. Sutomo Hospital. And Dr. Sutomo Hospital, I think, is the main referral hospital in East Java and also a research institution. So if we are wondering how we bridge the research and the management of the patient, the clinical management of the patient, Prof. Cita is I think Prof. Cita will give a lot of her experience about translating the research result from the bench to the bedside. And I think Prof. Cita has already received many awards. And she has already many experiences. So without any further ado, we will give the time to Prof. Cita, and she will be delivering her lecture about translating immunological knowledge into clinical practice and public health policy. Without any further ado, Prof. Cita, the time is yours. Thank you.
Assalamualaikum warahmatullahi wabarakatuh. Selamat pagi, salam sejahtera, Om Swastiastu, Namo Buddhaya, salam kebajikan. Good morning, our distinguished guests. I will present in Bahasa, but my slides in English. Please slide. Sebelumnya terima kasih untuk kesempatan yang diberikan pada pasca sarjana. Baru beberapa minggu yang lalu saya ke sini ya, tapi tentang manajemen rumah sakit, sekarang ke sini lagi tentang imunologi. Terima kasih Prof. Jojo, Prof. Sebagai tetangga, jadi harus saling membantu. Prof. Panca, Prof. Tere, teman S3 dan juga Prof. Nunu, kakak saya, moderator dan semuanya. Jadi judul yang ditugaskan adalah Translating Immunological Knowledge into Clinical Practice and Public Health Hospital. Dalam hal ini karena saya praktisi di hospital policy. Jadi kata kuncinya ada 3. Translating Immunological Knowledge atau kita kenal Immunological Translational, Clinical Practice, and Public Policy. Jadi ini presentasi outline-nya. Yang pertama translational immunologi itu sebetulnya apa, kemudian best practice, karena kata kuncinya ada 3, selain translational immunologi ada clinical practice. Yang pertama adalah dynamic interaction of gut microbiota in immunopathogenesis psoriasis vulgaris. Ini saya banget ya, karena ini kulit banget dan dermatologi alergi imunologi memang subspesialis saya. Jadi terkait clinical practice, dan yang kedua hospital policy ini sebagai direktur ya. Jadi bagaimana kita membumikan imunologi, yaitu development of excellent pediatric immunotherapy services through innovation and entrepreneurship. Dan setelah itu apa sih sebenarnya translational immunologi itu gap-nya, limitasi, dan bagaimana rekomendasinya. Tadi sempat ngobrol sama Prof. Nunu, sebetulnya imunologi itu menarik, jangan dipikir bulatnya aja, tapi bagaimana kita membumikannya ya. Jadi kenapa sih kok imunologi itu penting pada era sekarang? Karena ya memang dasarnya adalah imunologi, dasar dari ilmu ya. Dan peningkatan kebutuhan itu jelas, tetapi yang lebih penting adalah bagaimana impact-nya setelah kita mengetahui tentang molecular level understanding dan seterusnya. Sehingga pertanyaan utama adalah bagaimana immunological konsep yang dimaknai sebagai bulat untuk sebagian dari kita mungkin ya, harus belajar dulu, harus membayangkan, berimajinasi, itu bisa secara efektif ditranslasikan pada measurable improvement pada healthcare system. Jadi translational immunologi seperti disampaikan oleh moderator, from bench to bedside, dan ada 3 visinya, yaitu triple E, evidence-based, equitable, and effective intervention. Untuk translational pathway, jadi kita sering berkutat di molekul dan mekanisme ini jadi itu dan seterusnya. Kemudian kadang-kadang nyandet titik ya, kadang-kadang terus ke diagnostik ke titik, kadang-kadang bisa terus ke terapi, tapi sampai ke clinical practice itu jarang. Jadi tugas kita semua di sini adalah nyambungkan ini. Ya tentu enggak mungkin satu orang mengerjakan semua, ini mesti kolaborasi. Dan public health strategy termasuk hospital, ini ada diagram yang kita tahu bahwa enggak bisa kita berhenti pada belakang-belakang, yang bawah-bawah ya. Jadi harus terus sampai bagaimana dia berdampak pada global. Yang pertama, best practice pertama tentang translational immunology pada clinical practice. Dynamic interaction, gut microbiota, immunopathogenesis, psoriasis vulgaris. Itu kata kuncinya. Jadi kawan-kawan sekalian, psoriasis vulgaris itu sebuah penyakit yang kronis residif dan autoimun. Saya mungkin ada nanti gambarnya 1, dan diperankan oleh proinflammatory cytokine terutama TNF-alpha, IL-12, 23, dan 17. Dan kalau di sini kita lihat bahwa ini ya bentuknya yang seperti lilin, tebel, bisa sebadan gitu ya. Mengapa psoriasis ini terjadi? Jelas karena ada immunity dysregulation, ada ketidaktercapaiannya homeostasis. Dan kenapa immunity dysregulation terjadi? Ada penyebab atau ada peran dari gut microbe dysbiosis. Kita tahu gut organ axis, kulit ini termasuk organ ya, dan di sini gut skin axis termasuk psoriasis. Oral probiotik diharapkan dapat memotong di beberapa titik sehingga menurunkan gejala psoriasis. Dan saat ini kita punya kolaborator kami dari Bina Nusantara, probiotik asli Indonesia, Lactobacillus plantarum IS10506, ini penciptanya Prof. Ingrid Surono, yang sudah terbukti safe dan terbukti efikasinya baik untuk atopic dermatitis. Psoriasis kita masih nggak tahu. Jadi kita lakukan riset. Pertanyaannya, atopic dermatitis itu alergi, psoriasis itu autoimun. Kok bisa diekstrapolasikan ya? Kita kembali. Orang sehat itu ada 2 syaratnya. 1, sistem protektifnya berjalan dengan baik, TH1, dan homeostasisnya berjalan dengan baik. Kesetimbangan ini. TH1, TH2, TH17, kira kalau digambar semua nggak cukup ya, TH macam-macam. Tetapi yang 4 ini adalah intinya. Dan kita bisa lihat di sini bahwa meskipun allergic disease dan autoimmune disease sangat berbeda dari patomekanisme dan dari tampilan klinisnya, namun sama-sama defisiensi relatif Treg. Jadi kalau regulatornya nggak ada, jomplang, ini jomplang ya, Tregnya kecil gitu ke atas, sehingga terjadilah bisa allergic disease, bisa autoimun. Sehingga kalau Tregnya baik, ya itu orang itu sehat atau asimptomatis. Dan kenapa riset kami pada atopic dermatitis diekstrapolasikan? Ya karena peran dari T-Rex. Dan probiotik itu perannya memang menginduksi T-Rex. Ini adalah evidence pertama kami melakukan RCT di atopic dermatitis dengan probiotik oral pada anak-anak dan dewasa, hasilnya baik. Nggak ada tentang mikrobioma ya, tetapi tentang dysregulasi imunitas dengan klinik. Hasilnya baik keduanya. Kemudian berlanjut kepada evidence yang kedua. Dilakukan meta-analisis untuk atopic dermatitis baik. Bagaimana dengan psoriasis? Nggak banyak risetnya. Tetapi beberapa sudah dilakukan lab in vitro, kemudian pada animal. Pada manusia ada, tetapi dengan sampel yang kecil dan case review atau case series ya. Sehingga kita masih positioning, punya tempat untuk melakukan riset. Evidence yang ketiga adalah melakukan analisis gut microbes dibandingkan pasien psoriasis dengan healthy control. Apakah memang benar mikrobnya itu berbeda? Kita lihat di sini. Kalau kita bicara tentang alpha diversity yaitu intra group, ini tidak berbeda bermakna. Bagus ya, masing-masing grup tidak berbeda. Intergroup beta diversity berbeda bermakna. Bagus juga, berarti ada temuan baru di sini. Di Indonesia psoriasis berbeda dengan healthy, orang-orang yang sehat. Dan ini merupakan temuan penting atau novel dan sedang dalam proses publikasi. Kemudian kalau kita lihat di film level ini tidak berbeda bermakna. Tetapi bagaimana dengan selanjutnya family level berbeda bermakna. Yang atas ini sama-sama tinggi tetapi tidak berbeda bermakna. Nah yang kedua dan ketiga ini berbeda bermakna antara psoriasis dengan healthy control. Pada family level, dua yang berbeda. Bagaimana dengan genus, tiga yang berbeda. Bagaimana dengan spesies, 4 yang berbeda. Dan ada sesuatu yang menarik, tidak pernah ditemukan pada di luar sebelum ini bahwa Succinifibrio dextrinosolvans itu merupakan sesuatu mikrob yang berperan pada psoriasis. Dia menginduksi short chain fatty acid yang diinduksi oleh probiotik. Nah, Succinifibrio ini kurang di psoriasis dan setelah dilakukan probiotik nanti hasilnya bisa meningkat. Kita akan lihat di slide berikutnya. Kemudian tentang pre dan post probiotik dan placebo. Di sini kita lihat hasilnya. Alpha dan beta diversity tidak berbeda bermakna. Tidak apa-apa, namanya penelitian, Prof. Nyoman sering bilang penelitian itu bukan praktikum ya, jadi ya tidak apa-apa, tidak berbeda itu tidak apa-apa, tidak sesuai. Dan di sini kita lihat bahwa pada film dan family pun tak berbeda bermakna. Pada genus dan spesies pun tak berbeda bermakna. Apakah memang tidak ada peran probiotik? Belum tentu. Maka kita melanjutkan analisis tentang perubahannya. Nggak berbeda bermakna secara statistikal, tetapi ternyata secara klinis berubah dan ternyata perbedaan itu ada, perubahan itu ada walaupun tidak signifikan antara pre dan postbiotik. Ada sesuatu yang menarik di sini. Kalau film memang nggak berbeda bermakna, saya tidak tampilkan, tapi di sini pada family, pada Prevotelacea ini untuk psoriasis yang biru lebih rendah dibandingkan orange yang sehat. Dan kita lihat di sini, setelah dilakukan probiotik yang hijau itu meninggi ya, meningkat ya, sehingga mendekati yang sehat. Demikian pula ini, Bactericidase ini yang biru adalah psoriasis lebih tinggi daripada yang merah ya, yang kontrol. Dan ternyata sebelum dilakukan probiotik sama setelah turun juga. Jadi ternyata meskipun tidak signifikan difference untuk pelimpahan mikrobnya, namun family dan genus dan juga spesies itu berbeda bermakna dalam trennya. Sehingga secara ringkas key finding-nya adalah family, genus, dan spesies dari semuanya ini menuju approaching the healthy control profile. Jadi awalnya itu sangat berbeda tadi pada bukti yang ketiga, setelah diberi probiotik mendekati yang sehat, tapi belum signifikan. Kenapa? Banyak ya sebabnya, mungkin kurang panjang dan seterusnya, kurang inklusi eksklusinya kurang ketat dan seterusnya. Tetapi ini temuan penting karena belum pernah dilaporkan sebelumnya. Kemudian bukti yang berikutnya adalah double blind RCT, immune dysregulation dan clinical dan quality of life. Ini PASI itu psoriasis severity index, kemudian DLQ adalah dermatologi untuk quality of life dan dysregulation imunologi pada sitokin pro-inflamasi IL-17 TNF alpha dan IL-10 dan FOXP3 merupakan representatif dari T-regulator. Demografiknya tak ada perbedaan bermakna pada baseline data, kemudian ini yang akan kami lakukan dan alhamdulillah yang drop out hanya 1 ya, jadi masih valid, kurang dari 20% drop out. Ini clinical outcome. Kelompok yang diberi probiotik before after, kelompok yang diberikan placebo before after. Yang berikutnya, kelompok yang diberikan probiotik before after, kelompok placebo before after. Kita bisa lihat yang placebo ada penambahan lesi. Dan ini sesuatu yang menarik bahwa pada pro-inflammatory sitokin, interleukin 17 dan TNF-alfa kita bisa lihat yang biru, langsung turun. Memang psoriasis itu ditandai dengan naiknya TNF-alfa dan IL-17 sehingga biologic treatment itu memblok TNF-alfa dan interleukin 17. Namun yang placebo ini cenderung naik, ya ini cenderung tetap. Bagaimana dengan T-reg sebagai tokoh homeostasis dari imunologi tadi direpresentasikan IL-10 dan FOXP3. Kita bisa lihat yang biru meningkat. Dan di sini juga yang biru meningkat. Sementara yang orange cenderung turun. Sehingga probiotik memang berdampak, berperan pada disregulasi imunitas. Bagaimana dengan klinis yang biru-biru ini yang psoriasis, significant difference, tapi di LGA tidak, quality of life ya, quality of life dipengaruhi oleh berbagai hal. Dan kami melakukan pathway analisis dan di sini yang menarik bahwa probiotik ini menurunkan IL-17, justru menurunkan DLQI, tetapi kemudian setelah itu menurunkan PASI atau klinik. Di sini untuk analisis pathway-nya demikian, meskipun di LQI tidak berbeda bermakna before after, karena memang kami tidak melakukan terus-menerus. Important finding-nya adalah suplementasi probiotik pada pasien psoriasis di Indonesia, di Sutomo, sudah menunjukkan perubahan mikrobiota dysbiosis, awalnya memang berbeda, setelah dikasih penggeserannya juga berbeda, kemudian perbaikan imun dysregulation, dan clinical symptom. Dan tentu ini bisa ditingkatkan dengan pemberian yang lebih panjang, kemudian multi-strain, dan psychological support atau intervensi. Kemudian ini novel finding-nya belum pernah dilaporkan sebelumnya tentang microbiota profil, dysbiosis, dan juga succini vibrio dextrinosolvan. Dan ini adalah Indonesian Local Probiotic. Rekomendasinya tentu kita harus melakukan riset-riset lanjutan. Dan di sini ini sudah beberapa publikasi dari 1, 2, 3, 4, 5, 6 sudah dilakukan. Jadi yang pertama sudah selesai best practice tentang bagaimana translational imunologi membuktikan bahwa dari telaah, ekstrapolasi riset sebelumnya, ekstrapolasi untuk imunologi, ekstrapolasi untuk mikrob, kelimpahan dari mikrob, kemudian klinik sampai quality of life itu sebuah rangkaian translational imunologi. Yang kedua, ini mungkin agak kurang menarik untuk teman-teman di sini karena hospital policy, tapi karena ini imunologi, ini adalah mengambil contoh imunologi, maka saya ingin share karena di pasca ini semua ilmu kayaknya bisa nyambung ya Prof. Jojo dan Prof. Panca. Jadi yang kedua judulnya best practice tentang translational immunology in hospital policy dengan judul Development of Excellence in Pediatric Services through Innovation and Entrepreneurship. Jadi kita bisa lihat di sini bahwa entrepreneurship sendiri, karena kami bekerja di rumah sakit, based healthcare services, itu harus ada value driven oriented, harus ada inovatif dan adaptif, mau nggak mau, harus evidence dan database, nggak boleh ngawur, performance dan financial sustainable oriented harus, karena rumah sakit bisa amblek juga ya kalau finansialnya rugi. Kolaboratif dengan oriented ini yang paling penting. Kalau kita bicara ilmu imunologi titik hanya di dasar itu kurang menarik dan kurang berguna. Berguna pasti untuk keilmuan, tetapi untuk pasien, untuk masyarakat itu harus dilanjutkan. Dan itu hanya bisa kalau kita saling bergandeng tangan. Dan tentu building patient image dan loyalty. Gimana kita bisa retensi. Kalau ini rumah sakit ya, rumah sakit banget gimana pasiennya bisa retensi. Ini beberapa tahun yang lalu semua KSM diminta untuk menelurkan layanan unggulan. Semua menawarkan dan beberapa kami akomodir dan beberapa hasilnya baik, beberapa stagnan, beberapa amblas sampai akhirnya ditutup. Ini contoh baik dari penyakit dari anak ya, ilmu kesehatan anak. Kalau kita bicara tentang significant increase incidence ada 8 penyakit. Kalau ditambahi karena penyakit pasien kami 80% BPJS. Large increase in prevalence that are covered by BPJS. Yang 8 tinggal 7 karena yang 1 tidak tercover. Kemudian positif harmoni. Rumah sakit itu harus survive. Bukan berarti matre dan mata duitan, tapi karena Sutomo ada BLUD, maka yang positif harmoni harus dikencengi untuk gendong indit yang negatif harmoni. Ini ngerasani BPJS, ada anak BPJS nggak ya di sini? Jadi BPJS itu membuat kami bleeding, sehingga kami harus berpikir gimana caranya supaya nggak bleeding, antara lain layanan unggulan. Kalau ngomong positif harmoni yang 7 ini tinggal 2. Kemudian setelah itu, ya tentu kami juga tidak pengen hanya ngopeni pasien BPJS meskipun itu positif harmoni. Jadi mana yang commonly found in upper class families, Ada satu, dan itu juga ternyata upper class families itu sering keluar negeri. Jadi often cause patient to seek medical treatment abroad itu ya satu alergi ini. Sehingga kami kenapa memilih alergi imunologi yaitu imunoterapi. Jadi rasional atau justifikasinya adalah climate change bisa membuat polusi, mempengaruhi respiratory tract alergi sehingga trennya naik. Aeroalergen antara lain house dust mite juga demikian. Dan tentu ini dipengaruhi temperatur, kemudian cuaca dan perilaku manusia tentu saja. Kenapa sih kok inovasi di imunoterapi itu diperlukan? Tadi ya climate change dan dust allergy is increasing. Imunoterapi sampai saat ini masih terbatas penggunaannya. Dan pasien Indonesia kebanyakan yang punya, yang mampu, ke luar negeri yang nggak mampu ya sudah gitu ya. Ini imunoterapi mekanisme, jadi hampir sama sebenarnya dari kalau natural exposure yang di atas ini, kalau low dose alergen, kemudian intinya ujung-ujungnya mast cell dan basofil dan eosinofil ini membuat gara-gara tadi ya antara lain asma alergi. Imunoterapi itu high dose alergen, tetapi ya enggak langsung mencolok tinggi ya lah, orangnya bisa kolaps, tapi bertahap. Jadi 1 minggu naik dosis, cucunya Prof. Anja ya sudah menjadi pasien kami, alhamdulillah matur nuwun Prof. Dan ini naik, kemudian setelah itu 3 bulan, tiap 1 bulan, terus sampai 3 tahun, akhirnya tiap 3 bulan. Dan dia mengeblok di sini ya, Treg, ini kata kuncinya di Treg, kemudian menghasilkan IgG4 dan IgA yang akan memblok gejala-gejala alergi sini. Jadi sebenarnya ya mirip yang natural, tapi bertahap didesensitasi, dilatih gitu ya, sampai dia mengenal dan aman. Kemudian apa bedanya dengan vaksin? Oh ya, disclaimer bahwa imunoterapi di sini adalah allergic imunoterapi. Jadi kita tidak bicara tentang imunoterapi kanker ya yang sudah banyak dilakukan atau sel terapi atau yang lainnya. Dan di sini kalau vaksin itu prevention ya sebelum diinduksi oleh yang lemah. Tadi disampaikan juga oleh Prof. Siapa tadi yang pertama? Karina ya, Prof. Karina sangat hebat beliau. Kalau imunoterapi dia sudah sakit, sudah banyak mast cell dan basofil yang ada di badannya, kemudian subkutan imunoterapi atau sublingual imunoterapi, kami tidak punya yang sublingual, kami punya subkutan. Kemudian ini dilakukan dan akhirnya Treg, sekali lagi kata kuncinya Treg untuk homeostasis, ini akan ngeblok sehingga berbeda tetapi sama-sama bermain di imunologi. Perkara devisa, karena rumah sakit selalu hitung-hitungannya uang ya. Jadi kita outcome klinis dan outcome finansial, karena sebagai BLU harus survive, harus mandiri. Dan di sini kita lihat, kalau dibandingkan kalau ke Singapura, jadi teman-teman atau pasien-pasien yang biasa keluar negeri itu ya cerita, tiketnya sekitar sekian range-nya. Kemudian akomodasi, makan, dan seterusnya. Sehingga total-total per visit itu 9,7 juta sampai 22 juta sekian. Dan kalau di Sutomo, 300.000 sampai 450.000, murah banget ya, sehingga masalah berikutnya apakah mereka percaya pada kami gitu ya, itu yang menjadi tantangan. Kemudian kenapa kok alergi imunoterapi, maka kita harus tahu sebenarnya apa sih imunoterapi itu, tadi ya latihan medical treatment for allergies through desensitization. Jadi ini kata kuncinya, didesensitisasi, dilatih dikit-dikit gitu ya, dan targetnya sebetulnya selain rinitis dan asma saat ini juga mulai di dermatitis atopik dan di ini ya apa namanya THT ya, alergi THT. Dan benefitnya adalah dari hipersensitif menjadi toleran. Kemudian pertanyaan gini, boleh nggak ya kita pakai sendiri aja alergennya, kemudian biar toleran? Ya bisa iya, bisa ngeblak ya, karena alergi itu, alergi imunologi terutama yang tipe 1 itu kalau dalam bentuk severe itu life saving. Jadi life threatening, sehingga hati-hati dengan coba-coba sudah tahu alergi tapi dicoba sendiri, jangan ya, harus dengan ahlinya. Dan bagaimana tadi sudah saya sampaikan, awalnya tiap minggu, kemudian sampai 3 bulan, kemudian 3 mingguan sampai 3 tahun. Jadi pasiennya nggak hilang ya pasiennya terus, apalagi kalau seneng terus ya. Jadi ini tadi sudah ya. Jadi inovasinya. Inovasinya apa? Karena untuk menghambat 11,5 billion US dollar ini loh. Jadi harus dilaksanakan subspesialis yang bisa menembus batas waktu dan geografik dan tentu diperluas cakupannya. Di sini kenapa dibutuhkan, tadi ya sudah ada, intinya imunoterapi itu sulit untuk didapatkan. Sehingga kita harus melakukan implementasi dan akhirnya merambah ke medical tourism, bagaimana kita menarik pasien-pasien kita yang ke luar negeri, tetapi di samping itu kita juga nggak boleh mengabaikan yang BPJS ya, karena 80% pasien kami BPJS, jadi BPJS bisa ngerasakan imunoterapi tapi yo antri ya. Kemudian kalau yang untuk VVIP itu bisa langsung mendapatkan layanan. Prospeknya imunoterapi, ini problemnya tadi ya, climate change, kemudian kita tuh Indonesia tuh pulau-pulaunya tersebar nggak karu-karuan dan areanya ada exposure house dust mite. Solusinya gimana? Ya harus kita sudah melakukan sebenarnya, tetapi waktu itu belum di cover BPJS dan belum besar-besaran pada saat sebelum ada inovasi ini. Ada opportunity karena affordable prices ya dibandingkan kalau ke luar negeri. Evidence kami melakukan jurnal beberapa kali publish jurnal, kemudian haki untuk pembuatannya, kemudian buku dan seterusnya. Dan yang penting kolaborasi dengan rumah sakit se-Indonesia, pasien-pasien yang mau ke kita nanti pada saat yang tiap 3 mingguan udah kembali ke sana dan bisa video conference nanti berapa bulan sekali kembali ke Sutomo. Dan setelah itu actionnya dilakukan dan saat ini merambah ke medical tourism. Inovasi imunoterapi ini inovasinya ya merubah before afternya. Private service tadinya tidak diakomodasi, sekarang diakomodasi. Limited service hours, fleksibel bisa di mana-mana. Remote service tadinya tidak diakomodasi, sekarang diakomodasi. Sehingga kita bisa lihat ini kalau ini tempatnya pasca ini ya, manajemen untuk marketing ya, harus ada positioning, brand, and differentiation. Dan kita lihat di sini bahwa strateginya double impact marketing. Jadi kita nggak boleh berpihak pada yang kaya-kaya saja. Upper to middle socioeconomic group kita lakukan, tetapi yang lower to middle juga yang disupport oleh BPJS. Sehingga kita adil tadi ya. Ini kok mati. Ya. Kemudian yang berikutnya, strategi berikutnya adalah promoting eksklusif VVIP service, ngajak omong-omongan dokter-dokter yang punya pasien yang biasa keluar negeri, diajak omong-omongan dan kemudian setelah mau ke Sutomo dan getuk tular ya lumayan banyak nanti ada hasilnya di belakang. Dan untuk yang BPJS tentu saja nggak boleh dilupakan sehingga bergantian yang praktek di BPJS dan imunoterapi. Ini goal dan targetnya, dengan demikian maka tiga-tiganya ini bisa tercapai. Development strateginya gimana? Yang pertama market segmentation tadi ya, untuk yang upper socioeconomy, middle dan lower. Kemudian differentiated service offering, tentu yang ditawarkan servicenya berbeda. Namun relevant communication kedua-duanya harus diopeni, yang non eksklusif atau BPJS maupun yang eksklusif atau private. Kemudian partnership program tidak boleh dilupakan. Dan ini tentu saja ya, out of town atau out of Java pasien-pasien kami banyak dari luar kota dan luar pulau, dan tentu local home based services yang juga harus dilakukan. Ini development proses yang terjadi pada transnasional imunologi pada imunoterapi. Awalnya kami melakukan penelitian dan kemudian terpublikasi, banyak yang Scopus Q1, kemudian melakukan ijir-ijiran ya, jadi analisis outcome finansial dari imunoterapi itu, ini juga publikasi dengan teman-teman S1 dan S2, kemudian buku-buku, kemudian kita melakukan launching dan retention dan advokasi. Jadi marketing funnel berjalan. Step pada imunoterapi. Yang pertama di VVIP imunoterapi klinik, kan Prof. Anang, ketua imunoterapinya, ini yang punya BPJS. Awalnya nggak karu-karuan gini, antri lama, ngamuk, bingung gitu ya. Kemudian setelah dilakukan inovasi jadi rapi. Kenapa? Karena kami melakukan pendaftaran online. Pendaftaran online jadi ya jam kerjanya tambah pendek, orangnya datang cepat dan tetap banyak ya, malah lebih banyak 200 tapi enggak grubel dan enggak marah-marah lagi. Kemudian ini marketingnya. Jadi waktu Hospital Expo di Persi saya woro-woro jualan gitu ya. Kemudian Sutomo Expo ngundang semua rumah sakit se-Indonesia Zoom dan datang. Kemudian di Jatim Expo kami juga Instagram dan seterusnya. Pokoknya apapun yang bisa dijual gitu ya, karena kami memang benar-benar ingin ini berhasil. Dan ternyata kita bisa lihat impact pertamanya. Di awalnya itu hanya ada 8 kota, kemudian 162 kota. Dan pulau, hampir semua pulau memang paling rengket di Indonesia, di Jawa ya, hampir semua pulau itu ada pasien-pasiennya. Ini menunjukkan bahwa kalau kita serius menggarap sesuatu program, outcome klinis baik, outcome finansial pun mengikuti ya. Ini ya, jadi setelah 2 tahun segini, tadinya cuma 8 kota. Dan kalau kita bisa lihat di sini ini kemajuannya ya dari 2023 ke April 2025 sangat meningkat ya 18 kali. Kemudian ini kalau per bulannya ya trennya memang naik ya alhamdulillah. Ini distribusi ya imunoterapi dan ini mulai kota-kota dan seterusnya. Tidak ada satu pulau yang besar ya yang tidak ada pasien kami. Dan ini adalah visit bulanan pasien di Rumah Sakit Sutomo dan ini pasien di tempatnya sana di kotanya. Jadi kita setelah berapa kali ke sana dilakukan oleh dokternya telemedicine berapa bulan sekali balik ke sini dan seterusnya. Jadi juga membuat financial outcome untuk dokter yang mengirim ya. Jadi ini dua-duanya dapat. Kalau ini visit bulanan kita bisa lihat meningkat dan kumulatifnya juga meningkat. Ini di Sutomo yang merah, yang hijau ini di tempat asalnya pasien dia totalannya meningkat ya. Jadi kalau mau sukses itu memang tidak boleh sukses sendiri dan tidak boleh egois ya, itu kuncinya. Kemudian kalau tadi jumlah visitnya ini revenue-nya juga meningkat ya. Revenue-nya ini untuk totalnya ya kumulatifnya. Kita lihat di sini, kalau bulan ke-26 yang bulan April kemarin, ini sudah 230, 88 sama 319 ya. Kemudian untuk totalnya sudah mencapai sekitar hampir 5M. Kita bisa lihat di sini kenaikannya ya. Jadi pasien 272 tuh pasiennya 272, tetapi nanti kunjungannya tuh 9.520. Dan itu berbagi antara Sutomo dengan yang di luar. Sehingga sekali lagi ini dibutuhkan memang Prof Anang itu mantan guru saya ya, mantan Wadir Diklit SDM, mantan Wadir Yanmed, orangnya sangat konseptor dan senang sama yang ruwet-ruwet dan ini membuktikan bahwa keruwetannya bisa berdampak yang happy untuk pasien dan untuk Sutomo. Ini sebuah contoh pasiennya cerita bahwa saya ini senang di Sutomo karena perawatnya ramah-ramah. Ini menyejukkan karena sejak jadi direktur hampir 2 tahun yang lalu kerjaan saya tiap pagi itu lihat Google review ya. Kalau ada maki-makian, alhamdulillah dari awalnya 3,8 sekarang 4,4, alhamdulillah. Tetapi setiap ada Google review saya share ke teman-teman, oh ini nggak benar. Loh kalau nggak benar ya sudah alhamdulillah. Tapi kalau benar ayo dibetulkan. Memang Google review itu penghakiman sepihak ya. Tapi enggak apa-apa. Jadi lumayan ini di antara makian-makian itu ada yang happy ya anak-anaknya. Kemudian di sini kita lihat bahwa impact-nya di divisi anak sendiri ada divisi alergi dan divisi respiro juga meningkat. Kita lihat di per bulannya maupun secara kumulatif. Pasiennya meningkat, pasti uangnya juga ikut meningkat. Jadi ini visitnya coba kita lihat, 40 per bulan jadi 1.300 per bulan. 1.500, 12.500. Kalau ditambah sama yang di pulau-pulau sana ya tambah lebih banyak lagi. Ini yang di Sutomo saja ya. Jadi kita bisa melihat di sini bahwa tidak berarti outcome klinis yang baik itu tidak disertai dengan outcome finansial yang baik di rumah sakit plat merah. Kalau di swasta ya jelas ya. Tapi kalau di plat merah ini tantangan tersendiri. Kita lihat di sini impact yang kedua bahwa di pediatrik alergi dan ini semua pediatrik enggak cuma alergi ya. Kita bisa lihat kalau dibandingkan sebelum COVID menaiknya 270. Tapi kalau dibandingkan COVID naiknya 650%. Ini enggak main-main ya. Kemudian pediatri secara keseluruhan ya naik 180 sebelum COVID, setelah COVID 300%. Tetapi kalau dibandingkan ya kira-kira ini yang bikin naik dari divisi pediatri ada 10 divisi ya. Ternyata ya alergi yang selama ini dianggap kering itu ternyata bisa menjadikan revenue untuk rumah sakit. Kalau tadi adalah visitnya, ini revenue-nya. Grow up-nya sekian, 369% sebelum COVID. Setelah COVID itu 929%. Ini luar biasa teman-teman. Dan juga kalau ini di outpatient clinic memang tidak setinggi yang di sini, tetapi alergi ini merupakan bagian dari seluruh pasien poli.
Prof. Cita,
Ya,
3 minutes
tinggal
left.
3 slide. Ya, pas kok. Jadi ini annual revenue kalau kita lihat bahwa pediatric division meningkat, kemudian all pediatric juga meningkat dengan jumlah kota-kota yang meningkat. Ini suksesnya adalah Pak Jonan ya, kami sudah tiga kali dibimbing oleh beliau membuka wawasan kita termasuk untuk layanan unggulan dan supaya menjadi agen transformasi budaya dan ada champion-championnya. International collaboration, oh ya pasien kami juga ada yang dari Malaysia dan Singapura, alhamdulillah ya. Ini national collaboration dan ini adalah internasional dan nasionalnya. Kemudian ini telemedicine yang kami lakukan di Sutomo dan alhamdulillah dapat juara 1 Persi Award 2023 dan ini dapat surat penghargaan, kemudian ada beberapa penghargaan lainnya yang sudah didapatkan. Summary-nya, value market dan segment orientation itu penting. Service innovation dan marketing strategi juga tidak boleh dilupakan dan optimization dari resources dan efisiensi. Selain itu, financial sustainability dan economic impact, networking strategy, collaboration, loyalty, social influence. Ini publikasi yang sudah dilakukan. Dan ini 2 step terakhir, mohon maaf tadi. Jadi ada gap, challenge, dan implementasi. Intinya, bariernya adalah setelah lab mandek, itu enggak boleh ya. Healthcare harus dilanjutkan sampai healthcare. Dan gap antara untuk mengakses immunological diagnosis dan terapi. Jadi kita memang harus lebih banyak bekerja sama. Dan tadi rangkaian sukses tadi semua adalah kerjasama, bukan kerjanya satu orang atau satu unit atau satu rumah sakit, tapi rumah sakit se-Indonesia, bahkan ada yang di Malaysia dan Singapura. Sehingga implementasi strategi penting untuk integrating immunology research into clinical service and health policy. Jadi solusinya integrasi dari health aspect, role akademik dan hospital leaders ini penting. Jadi peran atau komitmen dari direksi dan akademik yang ada di hospital dan tentu kawan-kawan yang bergerak di imunologi untuk membangun integrated immunology ecosystem, interconnected innovation ecosystem. One innovation open up another innovation. Saya rasa ini terakhirnya take home message yang 3 pillars yang harus diperhatikan pada translational immunology. Kita tidak boleh berhenti di science saja, tidak boleh hanya uplek di clinical practice saja, tetapi harus menggandengkan science. Hanya science yang baik akan mendukung clinical practice yang baik, clinical practice yang baik akan mendukung policy yang baik. Sehingga kami di Sutoma menganut evidence-based policy, policy berdasarkan evidence data. Dan tentu diharapkan kita juga berperan pada global health. Terima kasih, demikian dari saya, semoga bermanfaat. Assalamualaikum warahmatullahi wabarakatuh.
Thank you very much, Prof. Cita. Terima kasih banyak, Prof. Ekstensif sekali. Jadi memang intinya tidak hanya ngopeni pasien dengan evidence-based research tersebut, ya, Prof. Jadi harus ngopeni rumah sakitnya juga. Jadi memang dibutuhkan kolaborasi yang luar biasa, ya, Prof. Bapak Ibu, apabila ada pertanyaan, if you have any question to Prof. Cita, we have one slot for one question. Dr. Puji,
Iya, menarik sekali ya yang tadi disampaikan ya. Sudah masuk kan? Ya. Jadi yang pertanyaan saya ada 2. Yang pertama yang tadi tentang klinik imunoterapi tadi ya. Kalau kelewatan gimana? Apakah yang sudah katakan lebih dari 18 tahun? Jadi waktu itu memang sudah sempat dicek oleh Prof. Anang, tapi masih pakai probiotik terus sampai sekarang, dan kemudian kelewatan sudah sekarang 24 tahun masih pakai probiotik. Cuman kadang-kadang masih ada muncul kayak jarinya kayak gelodoki gitu ya. Tadinya mungkin ada batuk pilek awalnya waktu masih kecil. Nah, apakah ini kemudian bisa dimasukkan ke yang klinik tadi? Apakah khusus untuk anak-anak maksud saya? Kalau itu bisa untuk mengcover yang dewasa, menyenangkan sekali. Itu yang pertama. Kemudian yang kedua, tentang saya dengar bikin Heart Research Unit ya, HRU yang tempoh hari di broadcast. Nah, untuk katakanlah precision medicine yang lagi ngetren sekarang, ada banyak obat-obat yang targeted terutama untuk CA ya. Nah, karena mutasinya itu banyak, ada beberapa yang sudah memang di cover BPJS pemeriksaannya dan obatnya juga ada yang sudah di cover BPJS, tapi ada juga yang belum. Tapi obat mutasi yang belum, ada beberapa mutasi yang belum ada obatnya, ada beberapa mutasi yang obatnya sedang dalam fase 2. Nah, maksud saya, apakah Sutomo ini tidak menginginkan untuk terlibat dalam riset-riset seperti itu supaya pasien-pasien yang kebetulan mutasi yang nggak ada bisa dapat akses untuk obat-obatan itu. Terima kasih.
Terima kasih, Dr. Puji. Boleh langsung saya jawab ya. Jadi yang pertama, imunologi itu kan ada sekolahnya di badan kita. Kita waktu kecil sebelum di kandungan itu dia sudah mulai belajar. Kemudian bayi lahir itu mulai belajar lagi terus sampai tua. Nah, ini ngomong probiotik dulu ya, karena ini kan tadi probiotik yang depan, walaupun psoriasis kami juga melakukan di alergi dan imunoterapi. Probiotik itu paling bagus pada awal kehidupan, pada saat ibunya hamil. Ibunya hamil, bapak ibunya alergi kuat, kemudian anaknya menjadi tidak alergi. Itu sesuatu sekali. Dan itu ada meta-analisisnya dan kami juga punya riset. Sehingga kalau anak-anak diberi probiotik, bagus, masih riset saya bagus. Dewasa juga bagus, tapi tidak sebagus anak-anak. Kenapa? Karena multifaktorial. Kayak kita lah, kalau diajari waktu masih kecil, ayo jangan gini, masih nurut kan ya. Kalau sekarang ya sudah mau jangan gini, ya sudah gitu aja gitu ya. Kalau sudah tua, sudah terbentuk. Jadi tentu yang paling bagus evidence-nya adalah anak-anak. Imunoterapi pada orang dewasa itu kami sedang berproses karena riset yang dilakukan oleh tim kami dermatitis atopik ya, mungkin nanti kita bisa diskusi di luar forum, itu imunoterapi baik tapi masih animal. Dan saat ini kami akan berlanjut pada dewasa. Jadi kalau untuk imunoterapi ya, untuk imunoterapi. Jadi mau probiotik, mau imunoterapi itu yang evidence baik itu adalah anak-anak. Dewasa, kenapa bisa ada yang baik? Tergantung. Tergantung apa aja itu, tergantung komorbid, tergantung lamanya, tergantung severnya, tergantung kompleksiti atau yang apa penyakit yang menyertai dan lain-lain. Jadi apakah bisa? Tetapi kami tidak melakukan karena evidence base-nya tidak cukup, belum cukup gitu ya. Kemudian yang kedua, alhamdulillah Dokter Puji Sutomo sudah punya tempat di Scopus, Sutomo General Academic Hospital, dan kami terakreditasi Joint Commission International Academic Medical Center, itu ada standar Medical Professional Education dan Human Subject Research Program, sudah akan ketiga kali, dan kami sudah mendeklar sebagai Global Research Site dan banyak penelitian tentang global. Barusan tadi malam saya juga ditelepon lagi TBC. Tentang targeted, saya mesti cek ya, tapi yang saya pasti beberapa penelitian tentang global AI saya ingat karena itu onko paru, kemudian ada tentang pemeriksaan mamoprint karena kebetulan ada saudara saya yang ikut penelitian itu, jadi akhirnya bisa mamoprint yang dikirim ke Amerika itu gratis. Jadi kami memang melakukan. Kalau Dokter Puji punya koneksi, monggo silakan saya bisa jadi makelar langsung. Jadi kami melakukan dan teman-teman sangatlah niat ya, karena ternyata riset yang di Open itu juga menjadi revenue untuk rumah sakit. Diklat kami itu sekarang tahun lalu tuh 2M ya, sekitar 2M, sekarang udah hampir 20M. Jadi ningkatnya pesat diklat dan riset tentu bergabung apa dua tapi dalam satu direktorat. Jadi kami sudah melakukan dok untuk beberapa targeted. Targeted itu jadi masalah. Jadi di kami ini sering terjadi perdebatan antara manajemen dengan klinisi. Saya seorang klinisi, ya guru besar, mestinya maunya memberikan yang terbaik buat pasien. Tapi harus ingat rumah sakit ini tidak boleh ambruk. Sehingga sering teman-teman tuh memberikan targeted treatment, tidak di cover BPJS atau di cover kecil, minusnya di rumah sakit. Itu saya protes, tapi saya juga tahu sebagai tabib ya, sebagai dokter, saya juga pengen memberikan yang terbaik. Sehingga ada KMKB, Komite Kendali Mutu, Kendali Biaya, disepakati. Yang seperti ini, ya wis rugi ya rugi, tidak apa-apa. Boleh. Tetapi yang masih bisa tidak dengan targeted, ya jangan, karena rumah sakitnya ambruk. Jadi untuk targeted memang kami menjadi masalah dengan teman-teman onko, dan alhamdulillahnya juga saat ini kami bisa konsolidasi untuk pengadaan beberapa obat dengan Darmais ya untuk onko, sehingga harganya juga bisa lebih murah, sehingga minusnya tuh nggak gede-gede gitu. Karena kalau rumah sakit plat merah tuh selalu BPJS menjadi masalah ya. Dan alhamdulillah kami sih nggak pernah fraud. Tapi pertanyaannya bagus sekali Dokter Puji. Mungkin kalau ada tanggapan atau cukup ya nanti mungkin tak lanjut diskusi. Iya betul. Silakan. Tepuk tangan untuk Dokter Puji. Beliau adalah sahabat saya. Monggo silakan.
Terima kasih banyak Prof. Cita. Mohon maaf karena waktunya terbatas. Mungkin nanti diskusinya bisa di belakang ya Prof. Mungkin nanti Prof. Tere akan sowan langsung ke sebelah. Iya boleh. Iya. Iya. Oke. Baik, sebelum kita menutup sesi ini, akan ada sedikit penyerahan cindera mata dari panitia kepada Prof. Cita. Monggo Prof. Cita berserta Prof. Panca dan Prof. Tere.
Terima kasih.
Ya, terima kasih banyak Prof. Cita. Boleh satu kali lagi tepuk tangan yang meriah untuk Prof. Cita. So that would conclude the first session of this international conference. And I think it's door prize time again. So I will be giving back the time to Mr. Nabil.
Yes, thank you so much, Doctor. So, as you know, I'm back here and you know what time it is. It is time for me to announce the next batch, the next group of people who will receive the next group of door prizes. So, who is the next group? To the committee in charge, you may now display the names. Congratulations to Iqbal and Amana and also Dr. Puji. You can contact the committee towards the end of the conference to receive the door prize. All right, ladies and gentlemen, the 4th International Conference of the World Immunology Day 2025 has been sponsored by Senseware, Kairos, BPI, and Wello. We would like to invite the representatives of Sainswerk and Kairos to please present their company videos and products to the committee in charge. The time and place is now yours.
Selamat datang di PT Kairos Jaya Sejahtera, sebuah perusahaan PMDN yang didirikan di Kabupaten Sleman, Daerah Istimewa Yogyakarta pada tahun 2014. Kami hadir untuk memberikan pelayanan distribusi khususnya dalam bidang laboratorium. Visi kami adalah untuk menjadi distributor berskala nasional yang menjadi pilihan utama bagi pelanggan dan mitra usaha. Dengan berlandaskan pada nilai profesionalisme dan humanisme, mendorong kami untuk melakukan kegiatan distribusi yang terstandar, sehingga mampu memberikan layanan dan produk yang berkualitas. PT Kairos Jaya Sejahtera hadir untuk menjadi mitra dalam setiap kegiatan laboratorium Anda. Kami memiliki rentang produk yang luas untuk melayani kebutuhan laboratorium, seperti produk untuk penelitian genom dengan menggunakan PCR, penelitian protein dengan metode Western blot, immunohistochemistry, ELISA, dan bioassay lainnya. Penelitian mikrobiologi, sel kultur, kimia analisa, dan penelitian terkait life science lainnya. Pelayanan informasi yang detail terhadap produk dimulai dari tahap pre-order hingga after sale service menjadi kunci keberhasilan kegiatan laboratorium pelanggan kami. Standar operasional prosedur dalam kegiatan distribusi diterapkan sedemikian rupa sesuai dengan kebutuhan setiap produk. Proses penerimaan, penyimpanan, dan pengiriman barang diawasi dengan ketat sesuai dengan standar mutu yang ditetapkan. Didukung oleh sumber daya manusia yang terlatih dan sarana-prasarana yang terkendali, menciptakan sinergi dalam menjaga mutu produk selama distribusi. Sistem informasi perusahaan yang diintegrasikan pada semua divisi meningkatkan kecepatan dan akurasi data dalam kegiatan distribusi, sehingga proses distribusi dapat berjalan dengan prima. Kami, PT Kairos Jaya Sejahtera, memberikan layanan purna jual untuk memastikan terciptanya kepuasan pelanggan. Kami melayani diskusi pada tahap pemilihan spesifikasi demi mendapatkan produk yang sesuai dengan kebutuhan pelanggan. Pendampingan kegiatan laboratorium dalam rangka menciptakan keberhasilan penggunaan produk adalah salah satu fokus kami dalam melayani pelanggan. Selain itu, dengan senang hati, kami akan berkolaborasi dalam kegiatan pengembangan laboratorium. Berkat anugerah Tuhan yang Maha Kuasa, serta segala usaha dan komitmen dari seluruh tim PT Kairos Jaya Sejahtera, sampai saat ini PT Kairos Jaya Sejahtera dapat melayani distribusi ke sebagian besar wilayah di Indonesia. Dengan semangat, doa, dan upaya dalam menjamin mutu distribusi, PT Kairos Jaya Sejahtera akan melayani dengan lebih baik dengan harapan akan terciptanya kesuksesan dan kemajuan dunia usaha, ilmu pengetahuan, kesehatan, dan bangsa Indonesia.
All right, ladies and gentlemen, we will now resume our plenary of lectures for today's conference. So the third and fourth lectures will be moderated by our moderator, Dr. Vivin. To the committee in charge, please kindly display Dr. Vivin's CV. Please allow me to introduce our moderator. Dr. Vivin holds a PhD in medical science from Universitas Airlangga and has completed her postdoctoral training at Indonesia's National Research and Innovation Agency, or BRIN. Her extensive research has focuses on the immune system, microbiome, homeostasis, and the impact of nutrition on health, particularly in burn patients and infectious diseases. With numerous publications in reputable journals and active participation in international conferences and webinars, Dr. Vivin brings a wealth of knowledge and a dynamic perspective to today's discussion. So please, ladies and gentlemen, join me in welcoming Dr. Vivin as she moderates the next lecture.
Okay, thank you, Master of Ceremony, Mr. Nabil. Good morning, ladies and gentlemen. Thank you very much for attending for this international conference, World Immunology Day. Now we will continue our discussion. I'm Vivin Ervina. We'll get you for this discussion. And now for the first speaker is Dr. Eugene Ong Bong Ben. Is the Dr. Eugene join the Zoom?
Yes, good morning, doctor. I'm here.
Hi, Dr. Eugene. How are you? Please correct me if I'm wrong. Spell your name, Dr. Eugene.
Yeah, it's correct.
Yes, yes, correct. Alhamdulillah. Okay, Dr. Eugene will speak about unprevailing leptospirulence factor with yes. But before I continue, allow me to read curriculum vitae Dr. Eugene Ong Bong Bing. Please screen curriculum vitae Dr. Eugene. Okay, Dr. Eugene, nationality Malaysian, age year of birth 47, and now his age and current position of Deputy Director, Postgraduate in International, Coordinator for US American Interdisciplinary Collaboration and Advanced Science. And now the qualification is PhD in 2011, and in the University of Science in Malaysia. And research interest of Dr. Eugene is mechanism of bacterial virulence factor, recombinant protein production, and protein-based rapid diagnostic. And then, next. Okay. Preview data from Dr. Eugene Ong is a senior lecturer of University Science Malaysia at the Institute for Research in Molecular Medicine. He completed his master's study supported by the Malaysian National Science Fellowship in Molecular Biology, USM, in 2005. In 2007, Dr. Eugene was awarded in the ASEAN Program Associated Scholarship for his doctoral research at the Chemical Biology Department in Riken, Japan's primary research institute. Upon completion in his PhD in 2011, with the Best Thesis Award, he was appointment as a postdoctoral fellow and was appointed a lecturer in 2014. His research focused on the fundamental understanding of pathogenic bacterial protein that can be converted to diagnostic application. He group specialists in the use of yeast as a model for identifying and understanding bacterial virulence factor. He had been awarded several nation and university grants, mashallah. He was a visiting research associated professor at the University of Tokyo in 2016 and a visiting science at Riken, Japan in 2023. He had a graduate 6 MSc student and 1 PhD student as main supervisor. His lab's current focus on the characterization of novel bacterial virulence protein identified from a yeast Crohn inhibition screen. And next. And so many teaching experience from Dr. Eugene, you can read is so many. So Dr. Eugene focus on the microbiology, right, Dr. Eugene? And then next. Yeah, research activity also focus on the microbiology. You can read it is so many research activity. I read your curriculum, but it's so many publication have you written. And then next. Okay. So many complicated grants that Dr. Eugene have done. And next. Yeah, next. Okay, and achievement awards recognition from Dr. Eugene is visiting research associated professor, laboratory of signal transduction, and then graduate school of Frontier Science University of Tokyo. And next, okay, I just say that so many research activity from Dr. Eugene. Okay, next. And this is a research activity and publication also. Next. Okay, I think it's so many, Dr. Eugene. Yeah. Okay, because we have a limited time. Maybe Dr. Eugene, can do your presentation now, and your time presentation is 30 minutes. After that, we continue with a Q&A session. Okay, Dr. Eugene, time is yours.
Yeah, thank you, Dr. Waude. Right, I hope that's right as well. Yes, thank you very much for the kind introduction. A very good morning to everyone. Selamat pagi and salam sejahtera to Dr. Teresia, and also the organizing committee of the 4th Immunology Day International Conference 2025. This is a last minute invitation, but I'm very glad that you have kindly invited us to share our work with you today. And this is a fundamental work starting from the identification of virulence factors, as mentioned just now in the introduction by Dr. Waude. So briefly, I'm going to share with you what we've started roughly about 10 years ago until today. And we start with the kind invitation by Universitas Airlangga. So I do not know much about Universitas Airlangga, although we have heard about it in USM. There are many talks by many speakers from Universitas Airlangga, but I didn't know that it was in Surabaya, and I found out recently that there is a direct 3-hour flight. So we hope there will be more collaborations in the future if you're interested. And speaking about that, in connection with our talk today, we live in a tropical area. So as you can see here, Malaysia and Indonesia has a significant amount of annual incidence of leptospirosis. And perhaps the higher regions will be towards the east and also in some parts of Africa as well. So the reason for that is because of our tropical climate. So we enjoy a nice tropical climate in the equatorial belt. We're very close to the equator. And in Penang, as you can see here, we have nice national parks, just like in Indonesia, where families go for picnic and also for a swim. But there is also a warning sign, if you can see here. So the warning sign is both in Bahasa and also in English. Please be careful while you are enjoying the swim. The water, it can also be contaminated by bacteria or virus or parasites. And this is because annually, I think it's also the same in Indonesia, there are some cases of infections from various bacterial pathogens, such as leptospirosis. So next to the warning sign, next to the natural swimming pool, there is also a leptospirosis swimming sign, warning sign that says that, as you can see here, about the penyakit kencing tikus, right? That's in Bahasa, and tells about the various the risks of swimming in those waters, and if a person is not careful, if they have open wounds and so on, so they may be infected by leptospirosis or penyakit kencing tikus, because when they are having a good time picnic in the natural areas, sometimes families also bring food and so on, and this food, if not properly disposed of, can attract rodents that may carry the leptospira bacteria. And this can cause infection to humans through their urine. So that's why it's called penyakit kencing tikus. So what is leptospirosis, the bacteria that causes this disease? So it is called whale's disease. It is a spirochete. It's a gram-negative bacteria. Has a corkscrew shape, and it was discovered more than 100 years ago. Firstly, by Adolf Whale. That's why it is called whale's disease sometimes. However, the person who isolated the bacteria were these two doctors, Dr. Inada and also Dr. Ito from Japan. So in the world today, we have about annually 1 million cases of leptospira, leading up to 60,000 deaths if they do not receive proper treatment. And in Indonesia, I found out that in 2019, reported by the World Health Organization, there were about 920 cases and 122 deaths. In Malaysia also, we have quite a number of cases. So as you can see here, perhaps due to underreporting and official reporting, in Malaysia, leptospira is one of the diseases where you have to notify the Ministry of Health whenever you have a case of leptospira. So number of deaths is also quite high over many years, over about 10-year period. And periodically, we also still get reports in our local newspapers whenever there is a case of leptospira. So leptospira is transmitted by rat, as we have seen just now. It spreads either directly to human or to domestic animals. So if you have pets, you also have to be careful. And it can also affect cattle or those in the agricultural animals. And then once it is transferred to humans, whether through the water or through the rat's urine directly, it can cause many types of pain due to the internal organ being affected by the bacteria. And you have two phase, where at first you have very common sickness, which is similar to other types of disease, perhaps like dengue or even infectious diseases. But then you have what is known as the icteric phase, which is where a person has jaundice. So in terms of the pathogenesis, we can see here that the leptospira, once it enters the epidermis, for example, if someone has a cut in the skin, then the bacteria can enter through the breach in the skin, through the epidermis layer, and then it will slowly move on and attach to different parts of our cells, and then entering our blood vessels, and then get transported into different organs in the body. So these bacteria, because they have evolved over many years to be able to infect humans, they can enter the liver, the lung, the kidney, and also they are very smart in the sense that they can evade our immune system. So that is why leptospirosis can be very dangerous, because sometimes people think they have a normal infection, whether a flu, because they just have a fever in the beginning. But after that, due to the immune evasion, the leptospirosis can persist in our bodies. And this is from a talk that I gave some while ago in Japan, but I found this to be a very interesting and visceral image, where we can see the leptospira bacteria. So over here, they are actually in the liver cells. So it reaches the liver from the blood circulation, and then it can invade between the hepatocytes in the liver. it can destroy these liver cells. It can affect the bile, therefore leading to what is known as jaundice or the yellow color that we see because of the liver damage. So there are currently 10 species of Leptospira that has been identified. The main species that is responsible for most of the human infections is Leptospira interrogans. And then there are also intermediate or mildly pathogenic species, which are about 5 species of them. And then in the nature, in the environment, there are also non-pathogenic Leptospira. So these are less harmful to humans, but they may infect animals and other organisms in nature. So from a genomic study that was conducted in 2011, looking at the genome of Leptospira, researchers have found that there are about 655 unique proteins that may be potentially virulent. So there are 78% of them that has no function. So that is why we are particularly interested in trying to identify and find out more about the proteins of Leptospira, so we can perhaps treat and manage the disease more effectively in the future. So what are virulence factors? Virulence factors contribute to the pathogen's ability to cause disease. It is present in pathogenic species, for example, like this, but not in the non-pathogenic species. And virulence factors is important because to understand them, we can understand the pathogenesis or how the bacteria causes infection in the host, in human cells. We can also use them to identify targets for novel drugs or use them as biomarkers for diagnostics and design new vaccines. So using yeast, I'm going to show you in the next few minutes how we can use the yeast system to identify Leptospira interrogans virulence factors. So yeast is a model organism, just like the many other model organisms in biology and life science. As you can see here in Drosophila, the zebrafish, which is very popular. We also have Drosophila, the fruit fly. There is also the mouse and the mice, rat models, E. coli, and so on. So among them, yeast is a very simple microbial model, and we use Saccharomyces cerevisiae, which is the budding yeast, yeast that is used to make bread and so on, to make beer. But this is, of course, a laboratory strain, which is more suitable for research. So yeast is a very useful organism. And over the years, if you follow, for example, the Nobel Prize awards, then yeast research done in yeast has been instrumental to identify various cellular mechanisms such as apoptosis, autophagy, and so on. And even for cancer research, that has been awarded Nobel Prizes from discoveries made in yeast. And yeast is a very useful molecular biology model because we can do many things, many studies in it. So I'm not going to go through this, but we have published a paper on this last year, a review paper, and you may read more about it if you're interested in this area. So how does yeast work? So yeast is a simple system. It can be a good biosensor for bacterial virulence proteins. What we do is we clone the open reading frames or pathogenic genes from Leptospira or even any other pathogenic bacteria. And then these genes may present or express as a protein in yeast. So this is just a normal recombinant expression of proteins inside the yeast. And if the yeast is fine, they are not affected by these proteins being produced inside, then we can receive or we can observe a normal growth curve. However, if there is an effect on yeast, then we can see that the yeast is affected and the growth is also affected. So this is just a representation of a sick yeast, so that it's easier to understand. So we can use this difference where the growth is affected as an indicator of the bacterial virulence protein. So my student Lai many years ago, when he was carrying out his master's research, we started this project. And from about 3,700 open reading frames in Leptospira interrogans, we selected about 10%, which is the ORF, open reading frames of Leptospira, which potentially codes for those virulent Leptospira proteins. We were able to do a PCR to amplify these genes and then clone them so we were able to get about 230. We express them in yeast. We check the growth of each strain carrying the different plasmids. So in one well, we can have one plasmid being expressed. So we use a microplate reader to measure the absorbance, and we can get the growth profiles of the 230 plasmids and yeast strains that we were investigating. So these are some of the classes of proteins that were selected for this study, and these are all related to virulence or pathogenicity. So some of them are permeases, enzymes, some of them are related to signal transduction, to outer membranes, and they have different functions. So altogether, we selected 288, but were able to clone or obtain about 230 open reading frames into the plasmids. So this is what the result looks like after we've carried out the growth screen. So the screen, of course, are sigmoidal curves, but we converted them into a single point data so that it's easier to understand. So we can see here that those proteins that affected yeast has a lower growth compared to the control, which is somewhere over here. And then most of the proteins doesn't seem to have any effect on yeast, which is what is expected. So we identified about 10 proteins which were affecting yeast. So what we've done is that we wanted to confirm if these findings were true. So in yeast studies, we normally carried out what is known as a yeast spot assay. So what you do is just to take a little bit of the culture and then spot them on the agar. And this agar will tell you whether there is any growth or there is no growth. So for example, in this particular case, for the control, you can see that there is growth in both conditions. However, in the virulent strains, where the yeast is producing the virulence proteins, growth is actually affected, and we do not see any growth in some of the spot assays. So altogether, we've identified about 9 proteins which has effect on yeast, and some of these proteins have been studied or reported before, as we can see here. They are said to contribute to mammalian cell entry function. So it is called the MCE protein. Some of these proteins are also interesting in the sense that they have been reported to be possible antigens for leptospirosis, which means that we can use them as diagnostic antigens. But in our case, we are interested in finding novel proteins or proteins that have not been identified and have not been studied before. So we found this particular one protein that is possibly a putative virulence factor because they have a very strong effect on yeast. However, it has not been identified before. So that piqued our interest, and we wanted to know how these proteins affect yeast. So using molecular biology methods, we attach a GFP to this open reading frame, as you can see here. So we added a GFP tag so that now our protein can light up inside the yeast cell. So what it does is that it can tell us where this protein locates in the yeast and possibly pinpoint some of the interacting proteins. So how does it look like? So we express GFP either as a control, GFP only without any protein. And then here is connected to the Leptospira protein, which is in these codes. So we can see that for this particular protein, LIC10280, the novel protein that has not been identified previously, it seems to be located or targeting the nucleus. So that was very interesting for us. And then we also investigated the other proteins to see how they localize in the yeast cell. So some of them do not have a particular localization where they are very generally spread. But interestingly, we also observed that these pathogenic leptospira proteins, when they are expressed in the yeast cells, they also affect the yeast morphology. So the yeast cell is also affected in some way apart from its growth. So we look at all of the ones that we've identified. So after that, we focus on this particular protein called LIC10280 because that is the unknown protein and the other proteins have been described and or reported before. So we wanted to see if this protein can affect yeast if we change the concentration of the protein, meaning that we can control if we produce a small amount of protein, moderate amount of protein, or a high amount of protein. Does it affect yeast? But interestingly, we found that even at very, very low concentrations, this particular protein can affect yeast in a very detrimental way. And then we tried to inhibit the production of this protein. And we found that if we remove or we stop the production of this protein, then the yeast can recover its growth. So it means that this particular protein is the one that is affecting yeast directly. And this work was carried out by my PhD student Wong, who's done currently finishing his studies, and he's done a lot of work on this. So using Western blot, we also show that this particular protein is expressed and is present in yeast cells. So this is the protein which has about 120 amino acids, and it has a signal peptide as well. However, in the E. coli, when we express this protein is E. coli, it is not toxic to E. coli cells. So it means that this particular protein is only possibly toxic to mammalian cells, eukaryotic cells, because yeast is a simple eukaryotic organism. However, it doesn't affect E. coli when we express the same protein over here. But we found that when we express in E. coli, it seems that we obtain a smaller band, indicating that perhaps the signal peptide is removed in E. coli, as seen here. So we analyze this protein further. It is a 120 amino acid protein, which is relatively small when you talk about proteins in the cell, about 10 kilodalton. There's not much information available in the databases, but when we search, we found that it is a secreted protein, possibly an extracellular protein, and although it was predicted to be non-toxic in some of the toxin databases, but somehow it affected yeast. So we also studied using molecular biology methods because we are molecular biologists. So what we normally do is to try to chop up the protein to see which part of the protein is functional or affecting its function. So when we remove the signal peptide, as when you see here, the protein lost its function in inhibiting yeast. So over here, it can inhibit yeast, but when we remove the signal peptide, the function is lost. And then when we truncated from the C terminal of the protein, the activity is also lost as well. So we are not sure why exactly at this moment, but perhaps in the future we can understand more about this. And also we wanted to study the vitality of the cells and also viability. So what we mean by this is that we wanted to see if this particular protein is killing the yeast cells, whether it is causing cell death or it is just stopping the growth of the yeast. So when we conducted this experiment using the stains as shown in the previous slide, we used acetic acid as a control. So acetic acid can kill yeast cells, as you can see here. And then when there is no treatment, the cells can grow very normally. However, in our case, we found that there is only a slight reduction in the cell viability and also cell vitality. Therefore, it tells us that this particular cell, this particular protein doesn't cause cell death, and it only stops the growth of the yeast. So we call this as a growth arrest. So next, we investigated whether this protein is conserved and found in all of Leptospira. So another PhD student, Putri, investigated this by looking at the sequence, amino acid sequence and DNA sequence of this particular protein. And then we found that, again, interestingly, it is very much in the pathogenic species. So we can find this protein, all of pathogenic species, in the intermediate species, but it is absent in the non-pathogenic species. So maybe this gives us a clue that this protein may be involved in the infection or virulence of Leptospira. We also look at the amino acid sequence by comparing it with all the available species that we can obtain. And if you look at here, you can see that they cluster or align very conservatively among the pathogenic species, but among the intermediate species, the sequences has changed. So currently we are investigating this to see how this difference in sequence will affect the infection of Leptospira. And finally, what does this particular protein look like? So today, with the advancements of AlphaFold from Google, we can see that this protein has been predicted with high confidence to look something like this. So we are very excited about this. At least we know what this particular protein looks like. And right now, after looking at the structure of the protein by my colleague, Dr. Teh, we found that this protein has a kind of cavity. And these are some of the amino acid proteins, some of the amino acid residues, which may be involved with the activities of this protein. So we are currently investigating this. So in summary, we have identified a novel Leptospira virulence factor. We are still confirming with further studies how this particular protein may function during an infection. At least for now, it is very, very toxic and causes growth arrest in yeast. Even in low protein copies, the protein has a catalytic pocket, and we were trying to understand how this particular catalytic pocket can lead to its function. And hopefully we can understand more of this particular protein in yeast. So coming back to this particular figure, which I've shown you in the beginning, we know that the Leptospira protein, Leptospira bacteria causes infection by entering through the epidermis and then traveling through the blood vessels to go to the important organs in our body. And they can also evade the immune system in some ways. So therefore, understanding the pathogenesis of Leptospira and other bacterial pathogens is very important for disease management because we can now use this information whether for diagnostics or designing new drugs. And nowadays, with the many models of AI software, we can design novel proteins which may inhibit some of these particular proteins which causes infection.
I'm sorry, Dr. Eugene.
Yes.
Oh yeah, thank you. I'm sorry.
Yeah, so this is my last slide. I would just like to thank the committee again for giving me the opportunity to share our work today. And yeah, terima kasih and thank you so much.
Yeah, thank you so much, Dr. Eugene. Give applause for Dr. Eugene. Okay, thank you very much once again, Dr. Eugene, for your insight. Maybe I think we have similarity, Dr. Eugene. In Indonesia, have so many cases of leptospirosis. Maybe I hope we can join us for collaboration soon.
Yes.
Yeah. Okay. Now Q&A session. I'm sorry, I just have opened one questionnaire. Anyone?
Any question for Dr. Eugene?
If there are no questions, can I just share very briefly about our institute?
Oh, yeah.
Okay, because we have always wanted to tell you about our institute.
Oh,
I
yeah,
think,
sure.
yes,
Sure.
Airlanga's
Please.
Universitas Airlangga is quite popular in USM. We always hear about it.
Okay. Please,
Yes.
Dr. Eugene.
Yeah, okay. So our institute is called the Institute for Research in Molecular Medicine. It was founded more than 20 years ago, and we are commonly known as Inform in USM and also in Malaysia.
Yeah.
We are located in two campuses. So we have the main campus in Penang, which is about 3 hours away from Surabaya, very convenient by a direct flight. We also have another location, another building in the health campus of Kelantan, the east coast of Malaysia. Currently, I'm the deputy director, and these are our executive members, our director, Dr. Azia, and also my colleague, Dr. Venu. Currently, we have about 19 academic staff working on diagnostics and novel therapeutics and vaccine. And then we are currently one of the high institution of Center of Excellence in Malaysia, one of 20 institutions. And this is just about USM. I'm sure maybe you have known already. So our research areas are very much in diagnostics and infectious diseases, mechanisms of disease, as I've shown you just now. We are very much open to collaboration, so we welcome if you have any interest from your colleagues or yourself. We have developed some in vitro diagnostic kits that have been commercialized and are sold in the market in Malaysia and also in other parts of the world. We have many research collaborations with overseas universities all over the world. So we hope to welcome more collaborations with Universitas Airlangga. We only have 3 postgraduate programs at our institute. We have a PhD and Masters by Research and also Masters by Mixed Mode. So currently we have about 70 international and local students combined. These are some of the topics that are offered at our institute. We also have the mixed mode program, which is a coursework and combination of research. So in one semester, you can learn about molecular medicine, and then in the semester 2, you can learn more about either doing a business or doing more about research, and also conduct a small mini research project that will be in a dissertation. So this is quite a popular program, and we have many students from various parts of the world. So if anyone is interested, you are most welcome to join this program. Institute is also very actively involved in community engagement, going to schools for science outreach, and also some health programs. So yeah, that's it. We would like to very much encourage and welcome collaborations from yourself, whether it's through student exchange, whether your students would like to come for a few months. I think Universitas Airlangga students do come to USM for some short-term attachment, but perhaps not to our institute yet. And then we also welcome any other academic exchanges or joint collaborations. So thank you so much. Terima kasih again.
Thank you, Dr. Eugene. I hope we can collaboration soon. Maybe Prof. Teresia will say
Okay.
something.
Thank you very much, Dr. Eugene Ong.
Yeah, thank you.
Interest, about your insight, and thank you for your supporting for this program. And maybe in the future, we want to come in your campus.
Yeah, please visit us. Yeah, and I hope also I can visit your university sometime.
Okay. Yes, I have one question
Yeah.
about your presentation of
Yes.
the pathogenesis of Leptospira. In your last slide, you
Yes.
said that the pathogenesis is in the way, like attached to extracellular matrix and invade through host fibroblast endothelial,
Yes.
and then Leptospira enter the bloodstream, and facilitate immune evasion. And I want to ask about the invade through the host fibroblast endothelial and epithelial cells.
It means that the protein have homolog maybe with the fibroblasts or epithelial cells or endothelial cells like this, or maybe why the leptospira will insert by the cells.
Okay. So you would like to know more about the mechanisms of entry of the bacteria into either fibroblasts or into the cells. Is that correct?
Yes, yes,
Yeah,
yes.
mechanism of entry, right? Yeah. So I think to sort of maybe explain this, this is because of the unique shape of the bacteria. Oops. Unique shape of the bacteria. So Leptospira is a corkscrew type bacteria. So it has a spiral, and therefore I think it's due to this mechanism where it can have a spiral entry into host cells compared to other types of pathogens such as rod-shaped bacteria, where they need to have the cells perform some kind of phagocytosis to take up those cells. So a good example is actually Salmonella. Salmonella typhi can interfere with the cell and causes the cell to perform some kind of envelope to uptake the cells in the gut. But in the case of Leptospira, because it has a corkscrew shape, it is a spiral, so it is very effective in directly entering the cell and can bind to various extracellular matrix. So a few studies have reported that some proteins are involved in the binding to ECM material that allows the bacteria to penetrate the host cells.
Thank you. So it's very dangerous.
Yes, yes, yes.
Okay, okay.
Yeah.
Okay, thank you for your insight, Dr. Eugene.
Yeah, thank you, Prof.
Okay, thank you very much for Dr. Eugene. Once more again, give applause for Dr. Eugene
Thank you so much.
for appreciates our remarkable speaker, Dr. Eugene. We will give a digital certificate. We'll show in the screen. Oh yeah. We invite for Prof. Teresia for give digital certificate for Dr. Eugene. Please, Prof.
Thank you, Dr. Eugene. We will give you the digital certificate. Yeah. Thank
Yes,
you very
sure.
much.
Appreciate it.
Yeah, thank you very much once again. Thank you very much for Dr. Eugene. The topic, underfilling Leptospira virulence vector with ES. Okay, thank you, Doctor. And now, we will continue the next session, the next speaker. Okay, there is no announcement, door prize, Master of Ceremony. There's no. Okay, thank you. Okay, we will continue the next speaker. The next speaker is Dr. Arif. Dr. Arif Nur Muhammad Amsori. There is Dr. Arif in the Zoom.
Sure, I can hear.
Hello, Dr. Arif. How are you?
Hello, I'm good.
Hi, hope you can join with us soon. Hi, ketemu lagi.
Yeah.
Okay, thank you very much for your time, Dr. Arif. Now we will continue this discussion. The topic of Dr. Arif is hope in the pipeline, drug and vaccine innovation for Nipah virus. Right? Isn't right, Dr. Arif? Okay.
Yes,
But
correct.
before we continue our discussion, please share the curriculum vitae of Dr. Arif. Okay, Dr. Arif is now education from Dr. Arif is Kumamoto University, PhD in medical science, and now alumni. Yeah, Dr. Arif alumni from Universitas Erlangga, Doctor in Veterinary Science in 2023, and Bachelor of Science Biology pada tahun 2016. Next. Oh ya, mohon maaf, saya agak campur-campur. Nah, experience Dr. Arif is so many experience. Maybe you can just read it in the screen. Okay, next. Nah,
science achievement of Dr. Arif is so many. Next. Now, publication that written by Dr. Arif is so many, and expertise. I highlight the expertise of Dr. Arif is virology, bioinformatics, molecular biology, and immunology. So maybe you can join with Dr. Arif to collaboration research or maybe make a publication. Just notice that expertise of Dr. Arif is immunology too. Okay, next. Ya, Dr. Arif juga review editorial and review activity, editorial roles itu so many. Dr. Arif juga menjadi reviewer for academic journals. And next. Yes. Okay. Thank you very much. And now, because we have a limited time, okay, I'm so sorry, Dr. Arif. Now you can present your topic and your time, 30 minutes for presentation. Dr. Arif, time is yours.
Okay, thank you very much for introducing me. I'll just share my screen first. Could you see my screen?
Yeah, clearly.
Okay. Okay, good morning everyone. Yeah. Salam sehat tanpa obat for Indonesian people. And then thank you very much for having me in this activities in this seminar. And thank you for director of postgraduate school and head of the head of study program of immunology and all the committee of the conference. Today, I will talk about the hope in the pipeline drug and vaccine innovations for Nipah virus. I'm coming from Joint Research Center for Human Retrovirus Infection, Kumamoto University. Probably my research focusing on virology, and now I'm focusing on the retrovirus for especially like bovine leukemia virus, and then HTLV, HIV, and on the other hand, I also doing a research with my team and collaborators about zoonotic diseases like Nipah virus, Ebola, and so on. Okay, this is the table of contents for my presentation, overview of Nipah virus, zoonotic risk in Indonesia, current situation in Indonesia, therapeutics and drug development regarding the Nipah virus, and vaccine development landscape, challenges and opportunities ahead, and conclusion. Maybe vaccine development landscape have been demonstrated a lot by Professor Karin Naju. Okay, first, major infectious disease outbreak since 1918. So we can see here, Spanish influenza occurred in 1918, and how many deaths? Around 100 million. And we can see here Marburg hemorrhagic fever, Lassa fever, Ebola hemorrhagic fever, HIV/AIDS, Nipah fever caused by Nipah virus, SARS, MERS, Zika, and of course, COVID-19. So we can see here for distribution of Theropus bats, this kind of bats, and cases of Nipah virus worldwide. The color or red color introducing the Nipah virus occurring in Malaysia, Singapore, Philippines, India, and Bangladesh. How about Indonesia? In Indonesia, we don't have any cases in human, but some researchers already discovered these viruses. And the green color, it's about the Theropus distribution in Africa, South Africa, Madagascar, of course, in Southeast Asia, including some parts in Australia. So next, natural and experimental transmission of Nipah virus. We can see here Theropus bat in experimental infection. They can infect pig and other bats and then non-human primates, Syrian hamster, cat, ferret, mouse, guinea pig. For especially ferret, we can use ferret as animal trial in vaccine development and other researches regarding the upper infectious disease in our respiratory system. And then from Theropus bat, they are moving to pig and human. Like in Bangladesh cases, they are moving from pet directly to humans. In Philippines, we can see here from pet to horse and from horse to human and other animals. Yeah, for the details, we can see in this slide from saliva, urine, blood, or from pets, they can transmit the virus from horse to horse outbreaks in Philippines and pig to pig outbreaks in Malaysia, and then directly from pets to human in India or Bangladesh because of the rounded palm sap consumption. So that's why there are many cases directly from pets to human. This one about the colorized transmission electron microscope or micrograph. We can see here the yellow color and the red color, and this one regarding those virus. Okay, for how about the genome? So this one, the complete genome of Nipah virus with this accession code in GenBank, around 18,000 base pair, smaller if we compared with SARS-CoV-2. And we are focusing on F gene and G gene, or F protein and G protein. This kind of proteins can induce the immune response system. So that's why this kind of proteins very important to study it, and we can see many researchers publish the research in this kind of proteins. Okay, the genome and structure of Nipah virus. Nipah virus include in Paramyxoviridae. So this one, the other viruses in this family, Pula virus or Newcastle disease virus, and then Henipa virus, Hendra virus, and many things. And this one about the structure, we can see here the fusion protein or attachment glycoprotein and other structural proteins. And of course, for others, non-structural proteins helping the virus infect the host cells. This is the life cycle of Nipah virus binding to the host cells with Frid B2 and attachment glycoprotein, and then fusion and encoding release the viral single-stranded RNA, and then processing the transcription and translation like other viruses, and then assembly and budding process. After that, the new virulent particles comes up and infects other viruses, other cells. And then we can see here many vaccines and antivirals developments in against the proteins, like against the glycoprotein G, and then glycoprotein F, matrix protein, or also for antivirals, they targeting the RNA. This one about the Efrin B2 and G protein attachment. So we can see here from Bowden et al. And Su et al. And Efrin B3 with G protein. They already documented it in PDB database, and we can access it and we can use it for our research, especially in molecular dynamics, molecular docking, and other bioinformatics uses. Okay, amino acid alignment of ephrin B2 and ephrin B3 across species. This kind of research is so important because they can demonstrate how this virus can infect many species, not only human, not only the bats, or maybe not only the pigs, but almost all mammals can infected by this virus. If we check the B2 sequence and B3 sequence, only small changes here. That's why they can infect, rodent species based on Bosart et al. In virology. Quite old study, but this kind of research is so important. So, Solhering et al. 2018, 2016 stated about the Nipah virus replicates differently in human and porcine cells. Yeah, this one experimental studies have shown that Nipah virus replicates differently in human and porcine cells. The black color is porcine cells and the gray color is human cells. So human airway epithelial cells produce higher viral titers and exhibit faster spread through cell-to-cell fusion compared to primary porcine airway epithelial cells. We can see in this data also if interferon beta and other aspects. That's why this virus can infect the human. Symptoms and clinical sign of Nipah virus infection, we can see here in nose, eyes, lungs, of course, and spleen or lymph nodes, sera, blood, urine. In urine, we can see here viral RNA detected in urine. So maybe urine can be used as biomarkers for Nipah virus infection in the future. And kidneys, liver, heart, of course, brain or central nervous systems. So how about the taxonomy of Nipah virus? Based on the new updated taxonomy, including on order Mononegavirales and then family Paramyxoviridae and genus Henipavirus and the species name Henipavirus nipahens. There are other four species in Henipavirus like Enterovirus, Cheddar virus, Ghana virus, and the others. So this one about the phylogenetic tree of Paramyxoviridae. We can see here Paramyxoviridae and then Pornaviridae, Rhabdoviridae, Filoviridae. Filoviridae we can know like Ebola virus. And updated phylogenetic insight into Nipah virus, the blue color, Malaysia, and purple color, Thailand, green color, India, and the yellow color, Cambodia and Bangladesh. And we can see here the distribution of phylogenetic tree based on the regions, Bangladesh, Cambodia, and then Thailand, and so on. Indonesia is still not included because quite new research in Indonesia. Nipah virus specific diagnostic tools. Yeah, we can see here there are many tools used for diagnostic for infection like ELISA, detect antibodies, and viral antigen, and molecular aspects like cell-cell fusion assay, virus neutralization test, PCR, and sequencing, and tissue culture, and so on. So how about in Indonesia now? Based on Sendo et al. In 2010, Nipah virus in Kalimantan. Yeah, they published the results from Kalimantan, in West Kalimantan, of course, in Pontianak and Singkawang district. In Pontianak, they collected some samples that positive infected by Nipah virus in pets. And next, in Sumatra, same researcher, Sendo et al. In 2013 stated that Nipah virus in the fruit bat or Theropus vampirus in Sumatra, they collect samples from Deli Serdang and Medan and put in PCR tests. And then from the PCR test, they recorded many positive samples. After that, they performing a sequence, and of course, they have similarities with other viruses isolated from Bangladesh, India, Malaysia. The new research coming from Java, Putro et al. 2025, published this study in emerging infectious disease. So, Nipah virus detection in Theropus hippomelanus bats in Central Java. This one, the condition of their studies, and they collected the samples from bats. From this study, we can see here the samples they collect. They collect the samples from Magelang, and then they have similarities with Nipah virus Malaysia, other than Nipah virus Bangladesh. So it means that they have a close relation between virus in Indonesia and in Cambodia, Thailand, and Malaysia. That's interesting. So why bats become a unique? Because bats have unique immune defense as a viral reservoir. We can see here, paper published in Nature stated that bats have different immune system with other mammals. That's why if they infected with viruses, they are not response like human. So we can see here some aspects in bat are not produced and silently happens if we compare with immune system in human. That's why bats can be used as a natural reservoir. And what help for sustainable and healthy future is so important. You can see in this journal in Plus Pathogens. Prevention and control for Nipah virus infection are already stated by WHO Southeast Asia, but now Indonesia is not including in Southeast Asia member. Maybe there are some updated in the future. So bioinformatics and discovery and drug discovery for Nipah virus infections needed to reveal new drugs. This one super system for bioinformatics. We can see here like information technology, database, computational resource, and so on. And from 1,000 compounds, we can check and analyze until we get one compound only for candidate. Here is the details based on Saduangrat, Journal of Chemoinformatics. And marine natural products, again, in Nipah virus, already reported by Abdul Jalil et al. In computational biological chemistry. We can see here with bioinformatic tools, they can reveal some candidates. And also this one from Rababi and Nat, and they perform research in bioinformatics tools, starting from natural compounds, revealed, and toxicity analysis, molecular docking, principal component analysis, pharmacophore, and so on. And effect of remdesivir treatment on Nipah virus infection. So here, from David et al. And others, we can see in this slide that many drugs already tried to use for Nipah virus infection. And late of remdesivir have a good chance to be accepted as a drug in the future. And also, I developed a virtual research center for bioinformatics and biotechnology, and we are producing good systems and bioinformatic tools, including the molecular dynamics analysis. And you can see here for our research. Okay, we are moving to vaccine developments. Types of virus vaccines and adjuvants. We can see here many types of viral vector vaccine, DNA-based vaccines, and others already developed and described by previous speaker. And consideration for potential adjuvant, we can see here safety profile, of course. This one is important thing, and dose sparing effect, antigen compatibility, route of transmission, neutralizing antibody response, T-cell mediated immunity, and so on. This one, the preparation of subunit vaccine, starting from the virus genome analysis to subunit vaccine preparation and formulation. You can see here for virus-like particles vaccine, and I take this figure from Bahara et al. You can read all the details here. And this one, the diagrammatic workflow for DNA vaccine preparation and RNA vaccine, this one for DNA vaccine and this one for RNA vaccine. RNA vaccine, we can see for mRNA for the latest technology used in vaccine development. Okay, how about the computational biology and AI in vaccine development? We can reveal from the candidate prediction and then protein nature prediction and epitope prediction for B-cell epitope, T-cell epitope, and multi-epitope vaccine design, and then protein structure prediction, AlphaFold using and other tools, docking and simulation. For example, we can use ClassPro, HeadDoc, and Ember, GrowMax, and other tools, especially in AI. And then codon optimization to develop the plasmids and peptide isolation and purification. After that, adjuvant selection, and then preclinical and clinical evaluation. We can use mice, non-human primates, ferrets, and other species. And after that, clinical evaluation in human. How about the vaccine composition? So active ingredients, of course, like viral or bacterial antigens, adjuvants, antibiotics, trace components, preservatives, and stabilizers. That's so important for vaccine composition. And you can see here and read more in this reference. And this one about the vaccine adjuvants used in many times, like aluminum salts, xerosomes, and AS03, AS01, and so on. Indonesia have potential as a regional hub for vaccine R&D. We can see here, CEPI and Bio Farma have a partnership, and then of course Universitas Erlangga also developing Inavac vaccine in Vaccine Research Center and Development and Technology. And also this one, mRNA technology, we are join this consortium. Bio Farma represent Indonesia in this kind of event. And vaccine candidates, technology platform, phase, and disease. We know that recombinant protein have the most developed in vaccine development. And from this 22% of recombinant proteins, we can see here mostly in phase 1. And nucleic acid, inactivated, viral vector, and so on. And then how about the country leading in this development? USA, China, Western Europe, they are leading in vaccine development. And how about the virus or pathogens? SARS-CoV-2, influenza, HIV, malaria, and so on. Mostly, private or industry lead for this development, other than academic, corporation, or others. So Indonesia launched phase 3 trial for TB vaccine candidates. There are 6 candidates for TB vaccine, and Indonesia use 72 for clinical trials in phase 3. That's so important for eliminate TB in Indonesia. Yeah, you can read my article here in the conversation about the clinical trial phase 3, and don't believe in hoax. Okay. Design and immunogenicity testing of Nipah virus vaccine in mice. So in mice, we can see here, based on Kim et al., they developed preclinical trials in mice, and they collect the serum after 4 weeks and analyze which one better, which kind of peptides, and how about the dose, how about the adjuvant use. They analyze many things, including the route of transmission. Vaccine in development or to prevent Nipah virus infection. Yeah, many vaccines are already developed for Nipah virus infection. You can see here, and also the references and ongoing trials, clinical trials. Yeah, I already collected and checked for the slides, and there are 4 candidates in clinical trials ongoing right now. How about the new systems, conventional and novel vaccination routes? In the future, maybe we can develop microneedles. Microneedles prevent something not fun in vaccination process.
I'm sorry, Dr. Arif, you have 2 minutes
Yes.
left. Yes.
Yeah.
You.
And then unlocking structure using cryo-EM. And cryo-EM is so important and now established in Indonesia in BRIN. They have many types of cryo-EM technology. So it's not it's so important, and we can perform this study. And AlphaFold. Then what's lesson learned from COVID-19? And already described more in previous speakers. And this is X. We should ready in these diseases. And SEPI have target 100 days mission after the disease X identified. And in 100 days after that, vaccine and drug are already available. And SEPIs have animal model and clinical research partnerships, but Indonesia is not in these collaborations. And rise in high security pathogen labs worldwide is so important to eliminate Nipah virus or doing research in Nipah virus, because Nipah virus needs PSL4. So we can see here, not so many institutions have PSL4. In Indonesia, maybe we have BSL-3 and BSL-3 class, but not BSL-4. Okay, key takeaways and references. Nipah virus remains a serious threat in Southeast Asia, including Indonesia. Drug vaccine development is progressing, but no approved treatments yet. And then challenge limited infrastructure, early detection gaps, founding, of course, in founding, we have many problems. And One Health approach is crucial to effective promotion and control. And Indonesia can lead the stronger research collaboration and preparedness. So I have a research topic in frontiers in virology. You can submit your manuscript here, and I collaborate with my colleagues, including Russia, India, Japan, and others. And you can also apply for research internship in my research center in Kumamoto University, in Graduate School of Medical Science, and others. You can check here. I already placed some references. And this is my lab. We are focusing on the retrovirus research, HCRV, HIV, bovine leukemia virus, and other type of viruses like HPV, and others. Thank you very much. You can follow our lab Instagram if you need more information. Thank you very much, and back to the committee.
Okay, thank you very much, Dr. Arif. Give applause for our speaker, Dr. Arif. Okay, because of limited time, I just open one questioner. Any question? There is no question. It's mean the reason no wanted to door prize, right? No. Okay, any question? Yes, please. Please mention your name and affiliation. Thank you.
Good afternoon. My name is Septin. I'm graduated from the doctoral student of Erlangga University. Seeing your experience in the field of vaccine, Dr. Arif, it is interesting. And my question is, in developing vaccine itself, using the various methods, what is done to minimize the occurrence of antibody-dependent enhancement? Did you focus on that in the development of vaccine? Thank you, because you know that ADE is worsen the patient condition. Thank you.
Okay, yeah. Thank you for the question. ADE already reported in dengue vaccine previously, but now many researchers and one product already approved for dengue vaccine without any antibody enhancements problem. So depends on, if we see the overview of vaccine development, we should know that there are many process from the preclinical trials. In preclinical trials, there are many animals used, like mice, and then ferret, and then non-human primate. And we can see in this section that if there any problems with the safety of our vaccine candidates, is there any problems of the antibody enhancement, we can analyze more in this part of study in preclinical trials before we move to clinical trial. Otherwise, it will be more dangerous if we suddenly miss it and move to clinical trials, and something happen with human in clinical trials. That's so dangerous. So before in clinical trials, we should analyze and comprehensive assessments for preclinical trials, including antibody enhancement. But in bioinformatics, maybe there are many limitations. So that's why we cannot produce the vaccine with bioinformatics tools. Bioinformatics tools helps a lot in pre-development. We can analyze the protein, we can analyze the genome, we can analyze many things regarding the B-cell epitope mapping, T-cell epitope mapping, and other suggestions. But in antibody enhancement, we should know using the animal trials. In animal trials, we can analyze many things like how about the peptides impacts the organs of animals, how about the safety, how about many things. And then after we assess it and we can think that everything is okay, everything is clear and no problem, we can move to the clinical trials. And in preclinical trials, we don't use only one animal, not only mice, but sometimes we use mice, rats, and then non-human primates like macaques and other types of animals like ferrets. Ferrets, especially ferrets, that's so expensive, and we can import it from European countries to Indonesia, and they have special passport like human. That's so interesting. In my previous study, we developed many types of preclinical trials using ferrets in influenza and COVID-19 vaccine development. And we test for safety assessment of this kind of vaccine candidates. Okay, maybe enough, or you have still not clear? Feel free to ask.
Thank you, Dr. Arif, for your explanation. Okay, thank you very much, Dr. Arif. I'll give applause once again for Dr. Arif. Okay, for appreciates our remarkable speaker, Dr. Arif, we will give a digital certificate. We'll show the screen. And we invite Prof. Teresia for give a digital certificate for Dr. Arif. Please, Prof. Tera.
Terima kasih, Dr. Arif. Ya, bahasa Indonesia aja ya. Nah, karena ini asli Indonesia tapi suka tinggal di luar negeri ini. Ya, terima kasih udah memberikan support pada Imunologi Day kali ini ya. Semoga setelah pulang dari Kumamoto kembali ke Indonesia segera bergabung dengan kami. Gitu ya, Dr. Arif. Nah,
Terima kasih, Prof. Terima kasih banyak.
sertifikat saya berikan secara digital. Terima kasih banyak, Dr. Arif.
Terima kasih, Prof. Tere.
Ya, terima kasih Dr. Arif sekali lagi. Okay, this is the end of this session. Thank you very much for your attention. I'm Vivian Ervina. Thank you, and this is your time, Mr. Nabil.
All right, thank you so much, Dr. Vivin, for your time for moderating this session. Ladies and gentlemen, it is now time for the next agenda, the agenda that we have all been waiting for. It is now time for break time. And also for the poster presentation. So we invite all the jury and participants of the poster presentation to please prepare yourselves. And the presentation will take place in the hall outside of the conference room. So when you exit the conference room, please make a left turn, and that is where the poster presentation will take place. For the other participants, you are now welcome to enjoy the break time. The break time will last until 1 p.m. and we ask you to please kindly be back in the conference room before or by 1 p.m. Thank you and enjoy the break.
Indonesia memiliki mimpi besar menjadi Indonesia maju di tahun 2045. Sekolah Pascasarjana Universitas Airlangga memiliki 8 program studi multidisiplin, hadir menjadi institusi berkelas dunia dengan kemampuan untuk mengembangkan pemimpin-pemimpin terhormat yang berdampak bagi dunia. Selamat datang Sekolah Pascasarjana Universitas Airlangga. Dan inilah program studi yang kami kelola.
Saya Profesor Dr. Teresia Indah Budi sebagai Koordinator Program Studi Magister Immunologi Sekolah Pasca Sarjana Universitas Airlangga. Prodi Immunologi merupakan satu-satunya program studi Magister Immunologi di Indonesia. Prodi kami ini adalah multidisiplin, jadi bisa dari berbagai keilmuan. Sekolah-sekolah yang berkaitan dengan imunologi bisa mengikuti program studi imunologi di Universitas Airlangga. Kami punya kompetensi yaitu bahwa mahasiswa-mahasiswa yang mengikuti program studi imunologi ini bisa langsung terjun di dalam penelitian-penelitian yang mengikuti program dari penelitian dosen-dosen yang ada. Sehingga mahasiswa semakin mempunyai wawasan yang semakin banyak di bidang imunologi. Dan juga kami juga menyelenggarakan salah satu seminar yang kita sebut sebagai World Immunology Day, merupakan satu-satunya juga seminar yang kita selenggarakan baik itu tingkat nasional maupun internasional.
Kita disupport dengan adanya penelitian berpayung, pendanaan yang support luar biasa dari Pasca Sarjana Universitas Airlangga. Kita juga sebagai mahasiswa mendapat support untuk publikasi dan juga melalui pelatihan-pelatihan berkelas Scopus dari Q1 sampai Q4.
Karena imunologi itu banyak bertanya, itu PR-nya mahasiswa online harus lebih aktif dibandingkan kalau mahasiswa offline, dosen ada di depan kita dan kita tahu, oh seperti ini, oh seperti ini.
Magister Immunologi Pasca Sarjana Universitas Airlangga. Hebat.
Please we kindly ask you to eat in the room that has been provided outside, as in this conference room there is a no food and drink policy. Thank you, and thank you for your understanding. You may enjoy your meal outside in the room that has been provided.
Sibuk amat kakak cantik. Lagi ngerjain apa sih, Neng? Lagi ngerjain tugas nih. Kamu dari mana? Dari rumah. Daripada gak ada kuliah, lebih baik main di kampus. Tahu nggak? Ternyata autoimun itu sedang tren loh di anak muda. Katanya gara-gara gaya hidup. Oh iya? Iya, bener. Katanya makanan kekinian itu penyumbang utama terjadinya autoimun. Oh ya? Oh ya doang dari tadi juga dikasih tahu. Nah, selesai. Kenapa? Sehat. Sambut kali. Ayo, ikut aku. Mau mana? Udah, ikut aja. Di sini? Iya, di sini. Mau ngapain? Udah, daripada katanya sosmed, mending ikut kuliah tamu imunologi. Yuk.
Baik, hari ini kita akan bahas topik tentang autoimun. Kasus kanker itu juga merupakan salah satu dari autoimun, ya. Nah, dan apa penyebabnya dari autoimun? Dari apa yang sudah saya jelaskan tadi, ada yang mau ditanyakan kira-kira?
Ini kan membahas tentang autoimun. Nah, kalau misalnya tentang depresi, ada tidak hubungannya dengan autoimun? Makasih, Prof.
Cocok sekali ya, kebetulan di kurikulum kami ada kurikulum tentang psikoneuroimunologi. Jadi keterkaitan antara sistem imun dengan psikis.
Izin Prof bertanya, nama saya Astika Junansari. Apa benar Prof, makanan itu penyebab autoimun? Terima kasih.
Makanan itu apakah bisa merupakan salah satu penyebab autoimun ya? Bisa. Ya kenapa? Karena saat ini makanan-makanan modern sekarang ya, itu banyak yang artifisial.
Seru juga ya belajar imunologi. Bener banget, masuk imunologi aja. Lagian karena ini multidisiplin ilmu, bisa dari beragam keilmuan kok, nggak harus melulu kesehatan.
Bisa jadi bidang kesehatan bukan passion saya, Tapi dunia imunologi begitu menggoda. Tidak hanya tentang keilmuan yang luar biasa atau karena multidisiplinnya, tapi dengan imunologi, aku, kamu, dan kita bisa berbuat yang terbaik untuk orang-orang tercinta.
Ini dia alat yang bisa bikin kulit kamu makin sehat dan cantik. Namanya Dr. Firms Hydro Nano. Alat yang bisa mengeluarkan air dalam ukuran nano berbentuk spray. Ini adalah yang pertama di Indonesia karena terdapat 3 batuan mineral yang berfungsi sebagai antiinflamasi dan antibakteri. Bukan cuma air biasa, tapi ini air hidrogen yang mampu menyerap ke dalam kulit sehingga membuat kulit jadi terasa kencang dan elastis. Produk ini dapat digunakan untuk seluruh bagian wajah, tubuh, kulit kepala, dan bagian perawatan tubuh yang lain. Manfaat Dr. Firms Hydro Nano ini bisa untuk mencerahkan wajah dan membantu mencegah penuaan dini, membantu menghilangkan jerawat dan menghilangkan flek pada wajah, dan juga meningkatkan elastisitas kulit. Selain untuk menyehatkan dan membuat kulit jadi cerah, alat ini bisa jadi powerbank juga loh. Menarik kan? Buat kamu yang mau kulitmu cantik dan sehat, yuk langsung aja tanya ke Instagram @drwaterofficial.id.
Biotechnology is located in the beautiful and fast-developing Wuhan. ELK is a high-tech biological company specializing in immune and molecular biology research. We now own 1,600 square meter laboratory, having independent cell culture room and SPF animal room. We have 4 major platforms: pathology, immunology, molecular biology, cell biology. Our research focuses on immunochemistry, tumor research, neurobiology, cell cycle, signal transduction. We have 8,000 plus ELISA keys, 60 plus molecular biology reagents, 11,000 plus antibodies. We have certificated with ISO and CE mark. Each product has complete R&D, production and QC course, experienced R&D technical team, complete quality inspection support department, strict inspection process, providing customers with reliable quality scientific research products. We have fast delivery chain by FedEx or DHL. Since the company started overseas markets in 2018, we have reached stable cooperation with distributors coming from more than 50 countries and 5 continents. Our goal is to provide scientists with the most cutting-edge and cost-effective research tools. Our mission is to provide the best solution and the most reliable partners for researchers all over the world. We will always implement the concept of environmental protection in the production process.
Perlu kamu ketahui bahwa radikal bebas dan polusi udara saat ini sulit untuk bisa dihindari. Nah, dampaknya bisa membuat kulit kering, kusam, dan penuaan dini. Nah, supaya tubuh kamu tetap fit, kamu butuh antioksidan dan anti-aging untuk meremajakan kulit sel. Dan juga meningkatkan kualitas kesehatan tubuh dan kulit kamu. Nah, solusinya adalah dengan Dr. Water Hydrogen yang mampu merubah air biasa menjadi air hidrogen. Nah, katanya nih super antioksidan bisa 176 kali lebih banyak dari vitamin C karena bisa membantu kamu untuk melancarkan peredaran darah, menurunkan risiko penyakit jantung, stroke, terapi diabetes, dan juga kanker. Dan dapat juga digunakan sebagai inhaler untuk proses regenerasi sel dan terapi yang lebih cepat. Oh iya, paling penting nih, aman dikonsumsi semua kalangan. Produksi ini dimiliki dan juga didistribusikan oleh PT Ultra Multisukses. Dan saya ingin mengajak kamu semua untuk menggunakan Dr. Water Hydrogen sebagai solusi kesehatan tubuh kamu. Nah, untuk info lebih lanjut bisa langsung cek di website berikut ini.
Indonesia memiliki mimpi besar menjadi Indonesia maju di tahun 2045. Sekolah Pascasarjana Universitas Airlangga memiliki 8 program studi multidisiplin, hadir menjadi institusi berkelas dunia dengan kemampuan untuk mengembangkan pemimpin-pemimpin transformatif yang berdampak bagi dunia. Selamat datang di Sekolah Pascasarjana Universitas Airlangga, dan inilah program studi yang kami kelola.
Saya Profesor Dr. Teresia Indabudi sebagai Koordinator Program Studi Magister Immunologi Sekolah Pascasarjana Universitas Airlangga. Prodi Immunologi merupakan satu-satunya program studi Magister Immunologi di Indonesia. Prodi kami ini adalah multidisiplin, jadi bisa dari berbagai keilmuan. Sekolah-sekolah yang berkaitan dengan imunologi bisa mengikuti program studi imunologi di Universitas Airlangga. Kami punya kompetensi yaitu bahwa mahasiswa-mahasiswa yang mengikuti program studi imunologi ini bisa langsung terjun di dalam penelitian-penelitian yang mengikuti program dari penelitian dosen-dosen yang ada. Sehingga mahasiswa semakin mempunyai wawasan yang semakin banyak di bidang imunologi. Dan juga kami juga menyelenggarakan salah satu seminar yang kita sebut sebagai World Immunology Day, merupakan satu-satunya juga seminar yang kita selenggarakan baik itu tingkat nasional maupun internasional.
Kita disupport dengan adanya penelitian berpayung, pendanaan yang support luar biasa dari Pascasarjana Universitas Airlangga. Kita juga sebagai mahasiswa mendapat support untuk publikasi dan juga melalui pelatihan-pelatihan berkelas Scopus dari Q1 sampai Q4.
Karena imunologi itu banyak bertanya, itu PR-nya mahasiswa online harus lebih aktif dibandingkan kalau mahasiswa offline, dosen ada di depan kita dan kita tahu, oh seperti ini, oh seperti ini.
Magister Immunologi Pascasarjana Universitas Airlangga, hebat!
Sibuk amat kakak cantik. Lagi ngerjain apa sih, Neng? Lagi ngerjain tugas nih. Kamu dari mana? Dari rumah. Daripada gak ada kuliah, lebih baik main di kampus. Tahu nggak? Ternyata autoimun itu sedang tren loh di anak muda. Katanya gara-gara gaya hidup. Oh iya? Iya, bener. Katanya makanan kekinian itu penyumbang utama terjadinya autoimun. Oh iya? Oh ya doang dari tadi juga dikasih tahu. Nah, selesai. Kenapa? Sehat. Sambut kali. Ayo, ikut aku. Mau mana? Udah, ikut aja. Di sini? Iya, di sini. Mau ngapain? Udah, daripada katanya sosmed, mending ikut kuliah tamu imunologi. Yuk.
Baik, hari ini kita akan bahas topik tentang autoimun. Kasus kanker itu juga merupakan salah satu dari autoimun, ya. Nah, dan apa penyebabnya dari autoimun? Dari apa yang sudah saya jelaskan tadi, ada yang mau ditanyakan kira-kira?
Ini kan membahas tentang autoimun. Nah, kalau misalnya tentang depresi, ada tidak hubungannya dengan autoimun? Makasih, Prof.
Cocok sekali ya, kebetulan di kurikulum kami ada kurikulum tentang psikoneuroimunologi. Jadi keterkaitan antara sistem imun dengan psikis.
Izin Prof bertanya, nama saya Astika Junansari. Apa benar Prof, makanan itu penyebab autoimun? Terima kasih.
Makanan itu apakah bisa merupakan salah satu penyebab autoimun ya? Bisa. Ya kenapa? Karena saat ini makanan-makanan modern sekarang ya, itu banyak yang artifisial.
Seru juga ya belajar imunologi. Bener banget, masuk imunologi aja. Lagian karena ini multidisiplin ilmu, bisa dari beragam keilmuan kok, nggak harus melulu kesehatan.
Bisa jadi bidang kesehatan bukan passion saya, tapi dunia imunologi begitu menggoda. Tidak hanya tentang keilmuan yang luar biasa atau karena multidisiplinnya, tapi dengan imunologi, aku, kamu, dan kita bisa berbuat yang terbaik untuk orang-orang tercinta.
Ini dia alat yang bisa bikin kulit kamu makin sehat dan cantik. Namanya Dr. Firms Hydro Nano. Alat yang bisa mengeluarkan air dalam ukuran nano berbentuk spray. Ini adalah yang pertama di Indonesia karena terdapat 3 batuan mineral yang berfungsi sebagai antiinflamasi dan antibakteri. Bukan cuma air biasa, tapi ini air hidrogen yang mampu menyerap ke dalam kulit sehingga membuat kulit jadi terasa kencang dan elastis. Produk ini dapat digunakan untuk seluruh bagian wajah, tubuh, kulit kepala, dan bagian perawatan tubuh yang lain. Manfaat Dr. Firms Hydro Nano ini bisa untuk mencerahkan wajah dan membantu mencegah penuaan dini, membantu menghilangkan jerawat dan menghilangkan flek pada wajah, dan juga meningkatkan elastisitas kulit. Selain untuk menyehatkan dan membuat kulit jadi cerah, alat ini bisa jadi powerbank juga loh. Menarik kan? Buat kamu yang mau kulitmu cantik dan sehat, yuk langsung aja tanya ke Instagram @drwaterofficial.id.
Biotechnology is located in the beautiful and fast-developing Wuhan. ELK is a high-tech biological company specializing in immune and molecular biology research. We now own 1,600 square meter laboratory, having independent cell culture room and SPF animal room. We have 4 major platforms: pathology, immunology, molecular biology, cell biology. Our research focuses on immunochemistry, tumor research, neurobiology, cell cycle, signal transduction. We have 8,000 plus ELISA keys, 60 plus molecular biology reagents, 11,000 plus antibodies. We have certificated with ISO and CE mark. Each product has complete R&D, production and QC course, experienced R&D technical team, complete quality inspection support department, strict inspection process, providing customers with reliable quality scientific research products. We have fast delivery chain by FedEx or DHL. Since the company started overseas markets in 2018, we have reached stable cooperation with distributors coming from more than 50 countries and 5 continents. Our goal is to provide scientists with the most cutting-edge and cost-effective research tools. Our mission is to provide the best solution and the most reliable partners for researchers all over the world. We will always implement the concept of environmental protection in the production process.
Hai friends, perlu kamu ketahui bahwa radikal bebas dan polusi udara saat ini sulit untuk bisa dihindari. Nah, dampaknya bisa membuat kulit kering, kusam, dan penuaan dini. Nah, supaya tubuh kamu tetap fit, kamu butuh antioksidan dan anti-aging untuk meremajakan kulit sel. Dan juga meningkatkan kualitas kesehatan tubuh dan kulit kamu. Nah, solusinya adalah dengan Dr. Water Hydrogen yang mampu merubah air biasa menjadi air hidrogen. Nah, katanya nih super antioksidan bisa 176 kali lebih banyak dari vitamin C. Karena bisa membantu kamu untuk melancarkan peredaran darah, menurunkan resiko penyakit jantung, stroke, terapi diabetes, dan juga kanker. Dan dapat juga digunakan sebagai inhaler untuk proses regenerasi sel dan terapi yang lebih cepat. Oh iya, paling penting nih, aman dikonsumsi semua kalangan. Produksi ini dimiliki dan juga didistribusikan oleh PT Ultra Multisukses. Dan saya ingin mengajak kamu semua untuk menggunakan Dr. Water Hydrogen sebagai solusi kesehatan tubuh kamu. Nah, untuk info lebih lanjut bisa langsung cek di website berikut ini.
Indonesia memiliki mimpi besar menjadi Indonesia maju di tahun 2045. Sekolah Pascasarjana Universitas Airlangga memiliki 8 program studi multidisiplin, hadir menjadi institusi berkelas dunia dengan kemampuan untuk mengembangkan pemimpin-pemimpin transformatif yang berdampak bagi dunia. Selamat datang Sekolah Pascasarjana Universitas Airlangga, dan inilah program studi yang kami kelola.
Saya Profesor Dr. Teresia Indabudi sebagai Koordinator Program Studi Magister Immunologi Sekolah Pasca Sarjana Universitas Airlangga. Prodi Immunologi merupakan satu-satunya program studi Magister Immunologi di Indonesia. Prodi kami ini adalah multidisiplin, jadi bisa dari berbagai keilmuan. Sekolah-sekolah yang berkaitan dengan imunologi bisa mengikuti program studi imunologi di Universitas Airlangga. Kami punya kompetensi yaitu bahwa mahasiswa-mahasiswa yang mengikuti program studi imunologi ini bisa langsung terjun di dalam penelitian-penelitian yang mengikuti program dari penelitian dosen-dosen yang ada. Sehingga mahasiswa semakin mempunyai wawasan yang semakin banyak di bidang imunologi. Dan juga kami juga menyelenggarakan salah satu seminar yang kita sebut sebagai World Immunology Day, merupakan satu-satunya juga seminar yang kita selenggarakan, baik itu tingkat nasional maupun internasional.
Kita disupport dengan adanya penelitian berpayung, pendanaan yang support luar biasa dari Pasca Sarjana Universitas Airlangga. Kita juga sebagai mahasiswa mendapat support untuk publikasi dan juga melalui pelatihan-pelatihan berkelas Scopus dari Q1 sampai Q4.
Karena imunologi itu banyak bertanya, itu PR-nya mahasiswa online harus lebih aktif dibandingkan kalau mahasiswa offline, dosen ada di depan kita dan kita tahu, oh seperti ini, oh seperti ini.
Magister Imunologi Pasca Sarjana Universitas Airlangga, hebat!
Sibuk amat kakak cantik. Lagi ngerjain apa sih, Neng? Lagi ngerjain tugas nih. Kamu dari mana? Dari rumah. Daripada gak ada kuliah, lebih baik main di kampus. Tahu nggak? Ternyata autoimun itu sedang tren loh di anak muda. Katanya gara-gara gaya hidup. Oh iya? Iya, bener. Katanya makanan kekinian itu penyumbang utama terjadinya autoimun. Oh iya? Oh ya doang dari tadi juga dikasih tahu. Nah, selesai. Kenapa? Sehat. Sambut kali. Ayo, ikut aku. Mau mana? Udah, ikut aja. Di sini? Iya, di sini. Mau ngapain? Udah, daripada katanya sosmed, mending ikut kuliah tamu imunologi. Yuk.
Baik, hari ini kita akan bahas topik tentang autoimun. Kasus kanker itu juga merupakan salah satu dari autoimun, ya. Nah, dan apa penyebabnya dari autoimun? Dari apa yang sudah saya jelaskan tadi, ada yang mau ditanyakan kira-kira?
Ini kan membahas tentang autoimun. Nah, kalau misalnya tentang depresi, ada tidak hubungannya dengan autoimun? Makasih, Prof.
Cocok sekali ya, kebetulan di kurikulum kami ada kurikulum tentang psikoneuroimunologi. Jadi keterkaitan antara sistem imun dengan psikis.
Izin Prof bertanya, nama saya Astika Juniansari. Apa benar Prof, makanan itu penyebab autoimun? Terima kasih.
Makanan itu apakah bisa merupakan salah satu penyebab autoimun ya? Bisa. Ya kenapa? Karena saat ini makanan-makanan modern sekarang ya, itu banyak yang artifisial.
Seru juga ya belajar imunologi. Bener banget, masuk imunologi aja. Lagian karena ini multidisiplin ilmu, bisa dari beragam keilmuan kok, nggak harus melulu kesehatan.
Bisa jadi bidang kesehatan bukan passion saya, tapi dunia imunologi begitu menggoda. Tidak hanya tentang keilmuan yang luar biasa atau karena multidisiplinnya, tapi dengan imunologi, aku, kamu, dan kita bisa berbuat yang terbaik untuk orang-orang tercinta.
Dia alat yang bisa bikin kulit kamu makin sehat dan cantik. Namanya Dr. Firms Hydro Nano, alat yang bisa mengeluarkan air dalam ukuran nano berbentuk spray. Ini adalah yang pertama di Indonesia karena terdapat 3 batuan mineral yang berfungsi sebagai antiinflamasi dan antibakteri. Bukan cuma air biasa, tapi ini air hidrogen yang mampu menyerap ke dalam kulit sehingga membuat kulit jadi terasa kencang dan elastis. Produk ini dapat digunakan untuk seluruh bagian wajah, tubuh, kulit kepala, dan bagian perawatan tubuh yang lain. Manfaat Dr. Firms Hydro Nano ini bisa untuk mencerahkan wajah dan membantu mencegah penuaan dini, membantu menghilangkan jerawat dan menghilangkan flek pada wajah, dan juga meningkatkan elastisitas kulit. Selain untuk menyehatkan dan membuat kulit jadi cerah, alat ini bisa jadi powerbank juga loh. Menarik kan? Buat kamu yang mau kulitmu cantik dan sehat, yuk langsung aja tanya ke Instagram @drwaterofficial.id.
Biotechnology is located in the beautiful and fast-developing Wuhan. ELK is a high-tech biological company specializing in immune and molecular biology research. We now own 1,600 square meter laboratory, having independent cell culture room and SPF animal room. We have 4 major platforms: pathology, immunology, molecular biology, cell biology. Our research focuses on immunochemistry, tumor research, neurobiology, cell cycle, signal transduction. We have 8,000 plus ELISA keys, 60 plus molecular biology reagents, 11,000 plus antibodies. We have certificated with ISO and CE mark. Each product has complete R&D, production and QC course, experienced R&D technical team, complete quality inspection support department, strict inspection process, providing customers with reliable quality scientific research products. We have fast delivery chain by FedEx or DHL. Since the company started overseas markets in 2018, we have reached stable cooperation with distributors coming from more than 50 countries and 5 continents. Our goal is to provide scientists with the most cutting-edge and cost-effective research tools. Our mission is to provide the best solution and the most reliable partners for researchers all over the world. We will always implement the concept of environmental protection in the production process.
Hai friends, perlu kamu ketahui bahwa radikal bebas dan polusi udara saat ini sulit untuk bisa dihindari. Nah, dampaknya bisa membuat kulit kering, kusam, dan penuaan dini. Nah, supaya tubuh kamu tetap fit, kamu butuh antioksidan dan anti-aging untuk meremajakan kulit sel. Dan juga meningkatkan kualitas kesehatan tubuh dan kulit kamu. Nah, solusinya adalah dengan Dr. Water Hydrogen yang mampu merubah air biasa menjadi air hidrogen. Nah, katanya nih super antioksidan bisa 176 kali lebih banyak dari vitamin C. Karena bisa membantu kamu untuk melancarkan peredaran darah, menurunkan resiko penyakit jantung, stroke, terapi diabetes, dan juga kanker. Dan dapat juga digunakan sebagai inhaler untuk proses regenerasi sel dan terapi yang lebih cepat. Oh iya, paling penting nih, aman dikonsumsi semua kalangan. Produksi ini dimiliki dan juga didistribusikan oleh PT Ultra Multisukses. Dan saya ingin mengajak kamu semua untuk menggunakan Dr. Water Hydrogen sebagai solusi kesehatan tubuh kamu. Nah, untuk info lebih lanjut bisa langsung cek di website berikut ini.
Indonesia memiliki mimpi besar menjadi Indonesia maju di tahun 2045. Sekolah Pascasarjana Universitas Airlangga memiliki 8 program studi multidisiplin, hadir menjadi institusi berkelas dunia dengan kemampuan untuk mengembangkan pemimpin-pemimpin transformatif yang berdampak bagi dunia. Selamat datang di Sekolah Pascasarjana Universitas Airlangga, dan inilah program studi yang kami kelola.
Saya Profesor Dr. Teresia Indabudi sebagai Koordinator Program Studi Magister Immunologi Sekolah Pascasarjana Universitas Airlangga. Prodi Immunologi merupakan satu-satunya program studi Magister Immunologi di Indonesia. Prodi kami ini adalah multidisiplin, jadi bisa dari berbagai keilmuan. Sekolah-sekolah yang berkaitan dengan imunologi bisa mengikuti program studi imunologi di Universitas Airlangga. Kami punya kompetensi, yaitu bahwa mahasiswa-mahasiswa yang mengikuti program studi imunologi ini bisa langsung terjun di dalam penelitian-penelitian yang mengikuti program dari penelitian dosen-dosen yang ada, sehingga mahasiswa semakin mempunyai wawasan yang semakin banyak di bidang imunologi. Dan juga kami juga menyelenggarakan salah satu seminar yang kita sebut sebagai World Immunology Day, merupakan satu-satunya juga seminar yang kita selenggarakan, baik itu tingkat nasional maupun internasional.
Kita disupport dengan adanya penelitian berpayung, pendanaan yang support luar biasa dari Pascasarjana Universitas Airlangga. Kita juga sebagai mahasiswa mendapat support untuk publikasi dan juga melalui pelatihan-pelatihan berkelas Scopus dari Q1 sampai Q4.
Karena imunologi itu banyak bertanya, itu PR-nya mahasiswa online harus lebih aktif dibandingkan kalau mahasiswa offline, dosen ada di depan kita dan kita tahu, oh seperti ini, oh seperti ini.
Magister Immunologi Pascasarjana Universitas Airlangga, hebat!
Sibuk amat kakak cantik. Lagi ngerjain apa sih, Neng? Lagi ngerjain tugas nih. Kamu dari mana? Dari rumah. Daripada gak ada kuliah, lebih baik main di kampus. Tahu nggak? Ternyata autoimun itu sedang tren loh di anak muda. Katanya gara-gara gaya hidup. Oh iya? Iya, bener. Katanya makanan kekinian itu penyumbang utama terjadinya autoimun. Oh iya? Oh oya-oya doang dari tadi juga dikasih tahu. Nah, selesai. Kenapa? Sehat. Sambut kali. Ayo, ikut aku. Mau mana? Udah, ikut aja. Di sini? Iya, di sini. Mau ngapain? Udah, daripada katanya sosmed, mending ikut kuliah tamu imunologi. Yuk.
Baik, hari ini kita akan bahas topik tentang autoimun. Kasus kanker itu juga merupakan salah satu dari autoimun, ya. Nah, dan apa penyebabnya dari autoimun? Dari apa yang sudah saya jelaskan tadi, ada yang mau ditanyakan kira-kira?
Ini kan membahas tentang autoimun. Nah, kalau misalnya tentang depresi itu ada tidak hubungannya dengan autoimun? Makasih, Prof.
Cocok sekali ya, kebetulan di kurikulum kami ada kurikulum tentang psikoneuroimunologi. Jadi keterkaitan antara sistem imun dengan psikis.
Izin Prof bertanya, nama saya Astika Junansari. Apa benar Prof, makanan itu penyebab autoimun? Terima kasih.
Makanan itu apakah bisa merupakan salah satu penyebab autoimun ya? Bisa. Ya kenapa? Karena saat ini makanan-makanan modern sekarang ya, itu banyak yang artifisial.
Seru juga ya belajar imunologi. Bener banget, masuk imunologi aja. Lagian karena ini multidisiplin ilmu, bisa dari beragam keilmuan kok, nggak harus melulu kesehatan.
Bisa jadi bidang kesehatan bukan passion saya, tapi dunia imunologi begitu menggoda. Tidak hanya tentang keilmuan yang luar biasa atau karena multidisiplinnya, tapi dengan imunologi, aku, kamu, dan kita bisa berbuat yang terbaik untuk orang-orang tercinta.
Ini dia alat yang bisa bikin kulit kamu makin sehat dan cantik. Namanya Dr. Farms Hydro Nano. Alat yang bisa mengeluarkan air dalam ukuran nano berbentuk spray. Ini adalah yang pertama di Indonesia karena terdapat 3 batuan mineral yang berfungsi sebagai antiinflamasi dan antibakteri. Bukan cuma air biasa, tapi ini air hidrogen yang mampu menyerap ke dalam kulit sehingga membuat kulit jadi terasa kencang dan elastis. Produk ini dapat digunakan untuk seluruh bagian wajah, tubuh, kulit kepala, dan bagian perawatan tubuh yang lain. Manfaat Dr. Farms Hydro Nano ini bisa untuk mencerahkan wajah dan membantu mencegah penuaan dini, membantu menghilangkan jerawat dan menghilangkan flek pada wajah, dan juga meningkatkan elastisitas kulit. Selain untuk menyehatkan dan membuat kulit jadi cerah, alat ini bisa jadi powerbank juga loh. Menarik kan? Buat kamu yang mau kulitmu cantik dan sehat, yuk langsung aja tanya ke Instagram @drwaterofficial.id.
Biotechnology is located in the beautiful and fast-developing Wuhan. ELK is a high-tech biological company specializing in immune and molecular biology research. We now own 1,600 square meter laboratory, having independent cell culture room and SPF animal room. We have 4 major platforms: pathology, immunology, molecular biology, cell biology. Our research focuses on immunochemistry, tumor research, neurobiology, cell cycle, signal transduction. We have 8,000 plus ELISA keys, 60 plus molecular biology reagents, 11,000 plus antibodies. We have certificated with ISO and CE mark. Each product has complete R&D, production and QC course, experienced R&D technical team, complete quality inspection support department, strict inspection process, providing customers with reliable quality scientific research products. We have fast delivery chain by FedEx or DHL. Since the company started overseas markets in 2018, we have reached stable cooperation with distributors coming from more than 50 countries and 5 continents. Our goal is to provide scientists with the most cutting-edge and cost-effective research tools. Our mission is to provide the best solution and the most reliable partners for researchers all over the world. We will always implement the concept of environmental protection in the production process.
Koordinator Program Studi Magister Immunologi Sekolah Pasca Sarjana Universitas Airlangga. Prodi Immunologi merupakan satu-satunya program studi Magister Immunologi di Indonesia. Prodi kami ini adalah multidisiplin, jadi bisa dari berbagai keilmuan. Sekolah-sekolah yang berkaitan dengan imunologi bisa mengikuti program studi imunologi di Universitas Airlangga. Kami punya kompetensi, yaitu bahwa mahasiswa-mahasiswa yang mengikuti program studi imunologi ini bisa langsung terjun di dalam penelitian-penelitian yang mengikuti program dari penelitian dosen-dosen yang ada, sehingga mahasiswa semakin mempunyai wawasan yang semakin banyak di bidang imunologi. Dan juga kami juga menyelenggarakan salah satu seminar yang kita sebut sebagai World Immunology Day, merupakan satu-satunya juga seminar yang kita selenggarakan, baik itu tingkat nasional maupun internasional.
Kita disupport dengan adanya penelitian berpayung, pendanaan yang support luar biasa dari Pasca Sarjana Universitas Airlangga. Kita juga sebagai mahasiswa mendapat support untuk publikasi dan juga melalui pelatihan-pelatihan berkelas Scopus dari Q1 sampai Q4.
Karena imunologi itu banyak bertanya, itu PR-nya mahasiswa online harus lebih aktif dibandingkan kalau mahasiswa offline, dosen ada di depan kita dan kita tahu, oh seperti ini, oh seperti ini.
Magister Immunologi Pasca Sarjana Universitas Airlangga, hebat!
Sibuk amat kakak cantik. Lagi ngerjain apa sih, Neng? Lagi ngerjain tugas nih. Kamu dari mana? Dari rumah. Daripada gak ada kuliah, lebih baik main di kampus. Tahu nggak? Ternyata autoimun itu sedang tren loh di anak muda. Katanya gara-gara gaya hidup. Oh iya? Iya, bener. Katanya makanan kekinian itu penyumbang utama terjadinya autoimun. Oh iya? Oh ya doang dari tadi juga dikasih tahu. Nah, selesai. Kenapa? Sehat. Sambut kali. Ayo, ikut aku. Mau mana? Udah, ikut aja. Di sini? Iya, di sini. Mau ngapain? Udah, daripada katanya sosmed, mending ikut kuliah tamu imunologi. Yuk.
Baik, hari ini kita akan bahas topik tentang autoimun. Kasus kanker itu juga merupakan salah satu dari autoimun, ya. Nah, dan apa penyebabnya dari autoimun? Dari apa yang sudah saya jelaskan tadi, ada yang mau ditanyakan kira-kira?
Ini kan membahas tentang autoimun. Nah, kalau misalnya tentang depresi, ada tidak hubungannya dengan autoimun? Makasih, Prof.
Cocok sekali ya, kebetulan di kurikulum kami ada kurikulum tentang psikoneuroimunologi. Jadi keterkaitan antara sistem imun dengan psikis.
Izin Prof bertanya, nama saya Astika Junansari. Apa benar Prof, makanan itu penyebab autoimun? Terima kasih.
Makanan itu apakah bisa merupakan salah satu penyebab autoimun ya? Bisa. Ya kenapa? Karena saat ini makanan-makanan modern sekarang ya, itu banyak yang artifisial.
Seru juga ya belajar imunologi. Bener banget, masuk imunologi aja. Lagian karena ini multidisiplin ilmu, bisa dari beragam keilmuan kok, nggak harus melulu kesehatan.
Bisa jadi bidang kesehatan bukan passion saya, tapi dunia imunologi begitu menggoda. Tidak hanya tentang keilmuan yang luar biasa atau karena multidisiplinnya, tapi dengan imunologi, aku, kamu, dan kita bisa berbuat yang terbaik untuk orang-orang tercinta.
bisa bikin kulit kamu makin sehat dan cantik. Namanya Dr. Firms Hydro Nano, alat yang bisa mengeluarkan air dalam ukuran nano berbentuk spray. Ini adalah yang pertama di Indonesia karena terdapat 3 batuan mineral yang berfungsi sebagai antiinflamasi dan antibakteri. Bukan cuma air biasa, tapi ini air hidrogen yang mampu menyerap ke dalam kulit sehingga membuat kulit jadi terasa kencang dan elastis. Produk ini dapat digunakan untuk seluruh bagian wajah, tubuh, kulit kepala, dan bagian perawatan tubuh yang lain. Manfaat Dr. Firms Hydro Nano ini bisa untuk mencerahkan wajah dan membantu mencegah penuaan dini, membantu menghilangkan jerawat dan menghilangkan flek pada wajah, dan juga meningkatkan elastisitas kulit. Selain untuk menyehatkan dan membuat kulit jadi cerah, alat ini bisa jadi powerbank juga loh. Menarik kan? Buat kamu yang mau kulitmu cantik dan sehat, yuk langsung aja tanya ke Instagram @drwaterofficial.id.
Biotechnology is located in the beautiful and fast-developing Wuhan. ELK is a high-tech biological company specializing in immune and molecular biology research. We now own 1,600 square meter laboratory, having independent cell culture room and SPF animal room. We have 4 major platforms: pathology, immunology, molecular biology, cell biology. Our research focuses on immunochemistry, tumor research, neurobiology, cell cycle, signal transduction. We have 8,000 plus ELISA keys, 60 plus molecular biology reagents, 11,000 plus antibodies. We have certificated with ISO and CE mark. Each product has complete R&D, production and QC course, experienced R&D technical team, complete quality inspection support department, strict inspection process, providing customers with reliable quality scientific research products. We have fast delivery chain by FedEx or DHL. Since the company started overseas markets in 2018, we have reached stable cooperation with distributors coming from more than 50 countries and 5 continents. Our goal is to provide scientists with the most cutting-edge and cost-effective research tools. Our mission is to provide the best solution and the most reliable partners for researchers all over the world. We will always implement the concept of environmental protection in the production process.
ladies and gentlemen, to the conference of the World Immunology Day 2025. I hope that the lunch break was very nice and the food provided was also delicious. I ate the same thing as you guys, and wow, the food was delicious. So kudos to the consumption team of the committee. All right, now without further ado, without wasting any more time, let us go right into the fifth and final lecture of today's conference, which will once again be moderated by our wonderful moderator, Dr. Vivin. I'm sure that you all guys already know her and still remember her. So, Dr. Vivin, the stage is now yours.
Okay, thank you very much, Mr. Nabil, Master of Ceremony. Okay, now we will continue our discuss for this international conference World Immunology Day, and the next speaker is Dr. Pramila Rijal, PhD. There is Dr. Pramila in the Zoom.
Hello.
Okay. Hello, Dr. Pramila. How are you? Hope you're fine.
Yeah, I'm great. Thank you.
Okay. Yeah, thank you very much for attending this World Immunology Day, Dr. Pramila. Okay, before we continue our discuss, let me I read curriculum vitae, Dr. Pramila. Can you share screen? Okay. Yes. Dr. Pramila Rijal, MRC with the Institute of Molecular Medicine, and Dr. Pramila Experience and Education, COI Career Development Fellow at the University of Oxford, and Dr. Pramila also postdoctoral science in University of Oxford, and doctoral philosophy medical science at the University of Oxford, and then bachelor science, life science, cum laude. Give applause for Dr. Pramila at the Han University, the Netherlands. Next. Okay, Dr. Pramila have a key research achievement. Neuraminidase influenza vaccine, development influenza neuraminidase nanoparticle vaccine and improve DNA production via loop grafting, and so many patents from Dr. Pramila. And then next. Okay, Dr. Pramila also have a selected skill, virology and immunology, and then molecular biology, data analysis and visualization, leadership and collaboration. It's a very match with us immunological and immunology magister program in postgraduate school Surabaya. Also learning about leadership, Dr. Pramila. And then Dr. Pramila teaching and supervision, supervising PhD and postdoctoral researcher, and then mentored MSc undergraduate and A-level student. Next. Okay, so many grant awards and publication from Dr. Pramila Rijal. You can read it is so many. Next. Okay. Okay. Yeah, thank you very much. Okay, Dr. Pramila, maybe you have 30 minutes for presentation. And now, after presentation, we continue with the Q&A discussion. Okay, now time is your, Dr. Pramila.
First of all, thank you very much for inviting me to present my research at the 4th World Immunology Day 2025 conference at the Universitas Airlangga. Thank you to the organizing committee and Professor Theresia. So today I'll be presenting on the neuraminidase influenza. So influenza is quite common all over the world. So I'll take you through some of the introduction and then into my research. So influenza has a wide range of hosts. So there are 4 influenza types. I'm just gonna, yeah. So there are 4 influenza types, and human epidemics are mainly caused by influenza A and B. There are influenza C and D as well. However, they do not cause severe infections in humans, and it's so the C is very mild. But then the influenza A viruses have quite a wide range of host immunity. So to introduce the influenza virus first, there are two. So it's a single-stranded RNA genome. It's a negative sense, and unlike the other RNA viruses, it's a single-stranded, and there are 8 segments. The gene is not a single, but then there are 8 segmented genes. So there are 2 major surface glycoproteins, which are HA and NA, we call them. So the HA is the most dominant, and it has about 80% on the surface, and the neuraminidase is only 20%. Yeah. So so there are influenza A and B viruses which cause human epidemics or pandemics. And in terms of influenza A, there are hemagglutinins H1 to H18 subtypes, and neuraminidase are from N1 to N11 subtypes. So based on the HA and NA the virus contain, the virus gets the name, the nomenclature. For example, H1N1. So there could be number of combinations. However, in humans, there are only 3 viruses which do co-circulate. Besides influenza A, there are also influenza B, but then influenza B it does not cause pandemic because it only causes seasonal or epidemics in humans. It doesn't have a wild birds reservoir. That's why. So influenza virus, because it's a RNA virus, and it can mutate, and it can mutate in 2 ways. So the one is called antigenic drift, which just means that the accumulation of the genetic mutations, and because of those genetic mutations, the HA and NA protein can be different, and then the antibodies would not recognize the new mutated viruses, and that is called antigenic drift. And then the second one is called antigenic shift. So the antigenic shift, because as I mentioned before, the gene is 8 segmented, so the genetic reassortment of these genes can happen in an intermediate host, or within the avian viruses as well, and they can either directly jump into the humans zoonotic viruses, or they can reassort in pigs, which acts as a mixing vessel between birds and humans. And that, because the humans are not immunologically targeted by these previous viruses, and it can cause pandemic. So that is called antigenic shift. So in last century, there were 3 pandemics. So the most significant one was the H1N1, which came in 1918, and it caused about 50 million deaths. And the other pandemics were H2N2 and H1N1. And recently, the H1N1, which started in 1977, and H3N2 from 1968, and so these can continue once they come from a zoonotic transmission. They can establish themselves, and then they will gain mutations, and then they can continue as seasonal viruses once they come as a pandemic. So there's always a risk for new pandemics. So we don't know what's going to be the next one. So would it be H5N1, H7N1, or any unknown ones? So H7N9, it caused number of human infections in 2013 in China, but because of the early interventions, it didn't cause pandemic. So recently, there are 3 viruses which co-circulate in humans as seasonal viruses, H1N1, H3N2, and B viruses. So for the B, there are B vector, two lineages of B influenza B viruses. We call them Victoria or Yamagata lineages. So far, the burden from the seasonal influenza is that it causes about 1 billion cases globally and 3 to 5 million severe infections and up to 650,000 deaths annually. There is a vaccine which the vaccine effectiveness can vary every year based on different factors. So, but then the vaccine effectiveness for H3N2 is particularly low. However, for H1N1 and B, it is good. So, what's the vaccine? What's in the vaccine, right? So the recent way of vaccination is we match the circulating hemagglutinin, which means that every year the WHO decides which vaccine strain, and then the manufacturers 6 months in advance before the flu season starts, manufacturers start preparing the vaccine, and the vaccine primarily is made in chicken eggs. The virus is infected, the eggs are infected with viruses, and then the grown viruses is inactivated, and the HA protein is purified from these viruses. So we call them split virion vaccines. But then there are problems with the way these viruses are made in eggs, because they can get these adaptation mutations which might alter the antibody response and then can cause mismatch of the vaccine and the circulating viruses. So recently there are also other ways of manufacturing, like the cell-based or the recombinant HA. So the HA is the main component of the vaccine. So this is the structure of hemagglutinin. It's a trimeric protein. So hemagglutinin is a trimeric protein. So we call this, the top part it's called head, and then the lower part is called stem or the stalk. So majority of the antibodies are against head domain. However, this is the most variable reason of hemagglutinin. It can gain mutations. That is one reason why we need to update vaccine every year. And there are antibodies to the stalk domain, which is more conserved, and then they can be cross-reactive between different strains, vaccine strains as well. However, the potency-wise, the head antibodies are much more potent than the stem antibodies. So the vaccine effectiveness can be improved. So I'll go first over how these antibodies to hemagglutinin and the neuraminidase work. So the function of the hemagglutinin is it facilitates virus binding and entry. So the hemagglutinin has this receptor binding site. So it attaches itself to the lung epithelial cells, and then helps get internalized inside the cells. And once the virus is internalized, it makes its RNA components and the viral proteins by hijacking the host system, and then they bud out on the surface where the neuraminidase comes into play. So the neuraminidase is essential for release of the viruses, because they can cleave off these receptors, which we call sialic acid receptors. So HA, as I mentioned, is the key antigen for vaccines, for seasonal as well as pandemic vaccines. So, but for NA, it is only present in unregulated and non-standardized form, and the immunity is not explored much in humans. So the HA is highly variable, that's why we need the regular vaccine update, whereas NA, it has a lower antigenic drift than hemagglutinin. So that's the protein that I'm studying, neuraminidase. So I will present the research in two parts. So I'll first explain the antibodies that we isolated against neuraminidase, and then the second part will be on the vaccine development. So just to take you through the literature on neuraminidase immunity, there is incredible literature on how the NA antibodies can be protective. So in 1988 paper, the first Webster et al. Showed that the neuraminidase antibodies can protect against lethal influenza. And in 1995, there was a purified neuraminidase vaccine was given to humans to show that it is immunogenic and non-toxic in humans. And then following up literature, it also showed that the neuraminidase antibodies can actually cross-protect, and this was in animal studies. And then the recent studies from 2015 and 16 showed that anti-NA antibodies are independent correlate of protection in humans, which means that they are independent of HA antibodies. They themselves can protect against virus severity and infections. So the recent study, the most prominent one is the correlate with protection in human challenge studies. So in these humans, antibodies were measured to NA, and then the humans were infected with the viruses, and the virus titers, the nose in the nose and lungs were measured, and along with other symptoms. And the anti-NA antibodies correlated very well with the prevention of viruses, virus titers, and as well as the symptom severity. So the monoclonal antibodies to neuraminidase. So we isolated human monoclonal antibodies using a single B-cell cloning technique, which was developed around 2009-2010. So how it works is that once the person gets immunized or infection, then you have within a week you have these B cells called plasmablast B cells, which express antibodies at very high level, and we isolate the PBMC and plasma from these humans. We with the FACS analysis, we sort for these single B cells in a 9-6 well plate, and then do the PCR of the heavy chain, the variable region of the heavy chain and the light chain of the antibody. We sequence them, and then we clone them in the expression vectors, and then we co-express them in cell lines to express the antibodies, and then which we validate using ELISA and other virus neutralization assays. So using this technology, we isolated from these 4 donors, we isolated 8 antibodies to neuraminidase. So this work was done in collaboration with Professor Arthur Huang in Taiwan. So some of the donors you will see actually were infected with H7N9 in 2013-14, and one donor here is received the trivalent influenza vaccine. So once we have the antibodies isolated, then we do various tests to assess their function and binding ability, and then whether they can protect against the virus. So one of the assay for the NA is specifically the inhibition of the NA activity, because I haven't gone through the NA yet, but NA is an enzyme and it cleaves off the sialic acids. So one of the tests would be to see if the function is inhibited. So I'm going to present the 3 antibodies out of this study. So this is interesting data because the antibodies to HA do not last. They might last for maybe 3 to 5 years, and then the virus mutates and they are not reactive anymore. However, these antibodies that we isolated, we tested them against N1 neuraminidase from 1918 to recent ones, and we found that actually these 3 antibodies inhibited the NA function for all these N1 viruses which have evolved for over 100 years. However, you will notice that there are some gaps. Some of the years there, the virus neuraminidase might have gained the mutation, and that would abrogate the antibody potency or the function. However, overall, there is a huge amount of cross-reactivity, but this is within subtype cross-reactive. But then there are neuraminidase, for example, N1 to N9, and some of the antibodies recently reported can be cross-protective across the groups, not just within N1, but then N1, N9, or N2, for example. So these antibodies inhibited the N1 from that has evolved in humans. However, H5N1 is a pandemic threat, and and wild birds are the main reservoirs of H5N1 virus, and sometimes they transmit into humans. And they can cause deaths. The case fatality rate of H5N1 is about 50%. So the first human infection from H5N1 was in 1996, and since then there have been several cases, random cases of H5N1 jumping from wild birds or other animals, intermediate host, into humans. So we wanted to test the potency of these antibodies against H5N1 neuraminidase. So these are so we expressed the antibodies and then we did the inhibition assays. And here are the clade 1 to numbers of clade that has been until 2015. So here you will see that these are the name of the 3 antibodies. So particularly this AG7C antibody, it inhibited the N1 from H5N1 against all clades. Whereas you will notice that this is the CYP serum generated against this particular neuraminidase, and the CYP, the polyclonal serum, are not as cross-reactive as these monoclonal antibodies. So the next experiment we do to test these antibodies is to give the antibodies to mice and then challenge these mice with viruses to see if they can protect against infection. And one of the indicator of protection is the prevention of weight loss, because once you give the viruses, then mouse start losing weight. So we can go up to 20% weight loss depending on the animal license. So here we give the antibody first as a prophylactic, and then we come with viruses 24 hours later, and then we assess the weight until 2 weeks. And we use irrevalent antibody as or the buffer, just PBS, as a control, as a negative control. So in this particular experiment, we used the H1N1 1934 virus, and here you will see that the antibody, this particular antibody called AG7C, whether it was given intraperitoneal or intranasal, it prevented these mice from weight loss, whereas the controls animals had to be culled after losing up to 20% weight loss within day 6 to 7. So this shows that the NA antibodies are protective against viruses. So because of these interesting antibodies, our interest got into developing vaccines using neuraminidase proteins. So, and also because of the H5N1, which was causing human infections. So there is a huge discussion on whether H5N1 will be a next pandemic, because I briefly mentioned about H5N1 insurgence in humans. However, recently in the US, there were a huge amount of cows which were infected with H5N1 viruses, and there was reduced milk supply. The virus was isolated from milk and wastewater, and also there was a direct transmission from animals to humans of these viruses. However, there is no human to human transmission because there are host restriction factors and other factors which stop the zoonotic transmission rate. However, these H5N1 viruses has also been isolated from lots of other animals, for example, wild birds as well as poultry, chickens, and also marine mammals and land mammals. Um, so there is a risk for if the human to human transmission can happen, then that is a main risk for pandemic, causing pandemic. So we selected one of the strain from the H5N1. So the strain we selected was from the recent clade 2, it's called 2344B, which was causing recent infections, H1 infections, and majority of human infections are caused by this virus within this clade. And this is also the virus which have been spreading widely in birds and mammals, and also it was found from mink farms in Spain, and caused a huge amount of deaths of the minks, and also dairy farm cattle and domestic cats in the US. So this is how the neuraminidase looks. So neuraminidase is a mushroom-shaped protein, and it has 4 identical subunits, tetramers, and it is on the surface by stalk, and this is called head domain, and there is a cytoplasmic tail and transmembrane domain. So the neuraminidase head will have these beta sheets which form the scaffold of the protein, and then the loops which comes on the top. And the neuraminidase has this it forms like a cavity where the sialic acid receptor binds, and that's the substrate by which the Na enzyme acts upon. And in terms of therapeutics, drugs, there are neuraminidase inhibiting drugs such as Tamiflu or oseltamivir and Janamivir, and that's also how these drugs work. However, in terms of drugs, they are only efficient if they are given within the very short window of treatment. They have to be given early after symptoms onset, otherwise they do not work very well. So this is our vaccine development workflow. So how we do not want to go through the egg viruses route. So we wanted to do the recombinant protein production. So for that, we designed a plasmid which contains the gene for expression. So the construct has a signal sequence, and then we put the other purification and other tags for the purification of the protein. And then we replace the stop domain of the neuraminidase with tetramerization domain. And this has and then comes the neuraminidase, only the head domain at the C-terminus domain. And we do the transient transfection of the XP2 or XP293 cells, which are known for high yield and also human-like glycosylation and post-translational modifications. So we purify the protein, and then we do a variety of tests to find out the purity and stability of the protein, and also if it has retained the good conformation, because the 3D shape or the structure of the protein is very important to make good antibodies. Further, we instead of giving the free protein, we multimerize them on the virus-like particles, because then you have like 60 Na molecules on the virus-like particles, and that will be more immunogenic. And we tested this vaccine in mice. The vaccine, it's tested as a prime boost, and we give it intramuscular with the adjuvant, and then 3 weeks after boost we do the challenge. And again, similar as we test the antibodies, we do the virus challenge and then look for the weight loss and clinical signs. So here I will show you an example of the H5N1 neuraminidase. Here we call it MSN1, and then the H1N1 2009 neuraminidase. And as controls, we do have NA antibody given here at this point as a positive control for protection, and then the negative will be non-flu protein. So the functional assay for testing the antibodies is the whether the NA activity. So this is the protein with sugars and sialic acids on the 96 well plate, and the neuraminidase will cleave off the sialic acids this top on the top, and if there are anti-NA antibodies, they will prevent this cleavage, and that's what we measure the activity of the NA. And the inhibiting antibodies means that there will be no or low activity of the neuraminidase. So in this assay here, you can see that for the control, which is Ebola protein here, and there is 100% activity. However, with these specific serum, NA and NAVLP, they give a good amount of antibodies which can inhibit the neuraminidase activity, and antibody to neuraminidase 1G1 is a control over here. And then in terms of protection, so because this sera also inhibited the H1N1 from 1934 as well as 2009 viruses, so we did the challenge with this 2009 virus. And here you will see, as shown for the antibody, the N109, which is a matched neuraminidase, it showed a protection with some weight loss because it gives you partial protection because the neuraminidase is not sterilizing immunity. And then the MSN1 vaccine from the clade 2344B, it also prevented from mice from gaining the up to 20% weight loss. So we also did prime with one and the boost with another, and then that also protected, whereas the control didn't. So that shows that the anti-NA antibodies can be protective also in the polyclonal sera. However, the main challenge of the vaccine production with the neuraminidase is the yield, because in recombinant expression, the yield is really low, only the maximum up to 30 mg per liter. So we expressed various number of proteins in our system, and we found that actually this protein 2009 expressed very low. However, this protein expressed really well. Although there are not many mutations within these two, they do match sequence-wise. So we're thinking of how to improve the yield and stability of the protein. So for that, we have to go through the NA protein in, anatomy. So this is a tetramer, and then as I mentioned, the NA has a scaffold and the loops, and then these loops are the main part where the antibodies bind to. So here I'm showing a monomer. And yeah, as I mentioned, these are the antibody binding surface, and then this is just forms the scaffold of the protein which retains the stability. So we thought of transferring these loops from the low expressing neuraminidase to the scaffold of the high expressing NA, and we made these NA hybrid molecules. Here I'm only showing a monomer, but then it will express as a tetramer. So I'm not going into the detail. It's recently published in eLife with peer reviews. But then I'll just give you a brief overview of this research here. So we found that by making these hybrid proteins, the yield was increased by 5 to 20 fold higher. And then the proteins we obtained were less prone to degradation and aggregation, so they had high thermostability. And also we tested against various antibodies, and we found that they were in the shape that we want them to be. So all the conformational epitopes were maintained. And the structure, we have the structure of this protein, crystal structures of this protein, and then they retained the native structure. And then further we tested these hybrid molecules in mouse vaccination, and then they match the immune response, antibody response with the wild type NA. And then further we showed that these hybrid molecules provided loop specific protection. So the antibodies which contributed to protection were against the loop. Please go to this paper if you'd like to go into details, but because of the time constraints, I'll keep it to the summary only. So now in the finals, almost the final slide. So to summarize, antibody-based immunity to neuraminidase is much broader compared to hemagglutinin. And the H5N1 NA protein is a good vaccine target for eliciting protective antibodies. And if it could be used combined with HA, then the vaccine effectiveness may go higher. And then some seasonal neuraminidase do not express well, and we have methods to enhance the yield by transferring loops to avian N1 framework. And going forward, we want to explore the scope of recombinant HA plus NA vaccines for seasonal and pandemic. And also we are also working on the feasibility of these loop grafting methods and other NA subtypes. In the paper, we only did for N1. So with that, I finished my talk, but I would like to acknowledge all the people who contributed to this study. So the key person was Alan Townsend, who was my previous supervisor, and I worked closely with him. And Leanne was a PhD student who did some of the experiments, and Ekta is a current postdoc in the lab. And there are other people who helped within the lab, Jack, Lisa, Diana, and Arthur Huang is a professor at the and group leader at the National University of Taiwan, who had been isolating antibodies, and we have this long-term collaboration with them. And there is a structural biology group in Oxford who did the structures of these proteins, Thomas Bowden, David Stewart, and Guido. And then there is a WHO Influenza Center at the Francis Crick Institute, and John Macaulay and Rod Daniels. And thank you everyone, Mark, Tao, and Yan Wu, and everyone, and the funders. So yeah, thank you very much for listening to my talk. So I'm happy to take questions now.
Okay. Thank you very much, Dr. Pramila, for your insight. Give a plus for Dr. Pramila. Okay, now we will continue in a Q&A session. I'm sorry, I just open one questioner. Okay, of course. Please, Dr. Ilham.
Yeah.
Hello, Dr. Pramila. It's been a while.
Yeah, nice to meet you, Ilham.
Yeah, nice to meet you too. Okay, yeah, thank you for sharing your knowledge with us. And maybe during your introduction, you in your slide, you show us the antigenic drift and antigenic shift of the virus, and you say that the antigenic shift is only it's happen in the vector or in the reservoir. Is it not happen in a human when is it infected human? I mean the virus not going to have the antigenic shift. And my second question is like the vaccine effectiveness you show us in the beginning, there are three types of vaccines. The highest one is only 61%, I believe. Is it I mean this value is not until like 80 or 90%. Is it because of the antigenic driven shift? Is that the case?
Yeah.
And yeah, the third one is when you told us the method of the cell lines for expression. Um, yeah, because me in here we have a dengue lab. Actually we have a HALT project, which is producing antibodies using SPIMAC as the cells to combine the B cell to produce the antibody. Maybe you have some tips which cell for the best expression for the antibodies. Thank you very much.
Thank you. These are very good questions. So I'll go through the first one. So the first one was whether the antigenic shift happens in humans. Do they always need reservoirs, right? So most of the cases, I should read the literature thoroughly as well, but I'm based on my knowledge. So the H1N1, which came in 2009, was mixed. The pig act as a mixing vessel. However, you always do not need antigen for mixing vessel reservoirs for antigenic shift. They might be able to adapt in humans as well. However, the chances are quite rare because of all the host restriction factors. And the preference for the binding of the sialic acids in humans is very different from chicken eggs. Yeah, so the best chances is actually reascertained for the viruses to it's a chance mutation. So the second question, I'll go through the third because I have to remember the second one. There was a so for the expression, so we use the CHO cells and 293 cells. So these are mammalian cells. So we specifically use XP-CHO. So these are proprietary cells from Thermo Fisher, which give very high yield because these are these cells were selected for high expression of the protein, and then they are high density cells which can grow at like 40 million per ml culture once they are at the peak, and make a good amount of protein. So also for antibodies we have been using these cells and they give us very good antibodies. But in terms of expression, there are now lots of methods actually, like the improved transfection reagents, improved media, which can make difference in the expression of the proteins. So could you repeat the second question again, just briefly?
The second question is about the vaccines.
Vaccine effectiveness, that's right. Yeah, yes, got it.
So the highest
So the vaccine, you're right, because the vaccine effectiveness of the COVID vaccine is around 95% for the mRNA and others. So comparatively to that, the vaccine effectiveness for flu, we call it suboptimal. So this is average taken from number of years. So, and then it can vary based on the seasons and various factors as well. The reason why the vaccine effectiveness low is also because the mismatch because of the egg adaptation mutations. But the Moderna is now actually working on the mRNA vaccines for influenza as well. They have done up to phase 2 and the phase 3 is in progress. So we might have mRNA vaccines in the market soon, and that will be interesting to see if the mRNA now improves the vaccine effectiveness also of the flu vaccines. Yeah, thank you.
Okay, thank you very much, Dr. Pramila. And I hope it's not the last time we keep in touch. Maybe we can keep in touch with immunology program here, and maybe in the future we can have a collaboration with the Oxford University.
Yeah, that sounds great. Yeah.
We can, or maybe Prof. Terry have something to say. Okay, thank you.
Okay.
Thank you very much.
Thank you very much, Dr. Pramila. Yeah, we hope we can collaboration with Dr. Pramila also. Okay, Dr. Pramila, we have another question in chat box by Zoom from Amana. Maybe I can read this. Thank you for insightful Prof. Pramila. Hopefully we can collaborate and discuss. The question is, antigenic drift in influenza driven by an endogenous or exogenous pathway? And how does it contribute the virus ability to evade the immune system? Additionally, why must influenza vaccine be updated annually? And is this primarily due to antigenic drift or the exogenous process of antigenic shift? Thank you, Dr. Pramila.
Thank you very much. That's very good fundamental questions. So the antigenic drift in influenza is driven mainly by how the virus mutates, because the genome is RNA. So unlike DNA, RNA does not have the proofreading mechanism of the virus polymerase, and then it makes mistakes, and those mistakes are not corrected, and that's how it gains random mutations. However, these random mutations can cause the change in the hemagglutinin protein structure. Now, for example, the lysine in one of the region can change to asparagine or some other residues, and these mutations can cause, like, if you have been vaccinated with 2014 HA, for example, that had been circulating, and then you made the antibodies against 2014. However, when it comes to 2016, now the virus has gained all these mutations in the HA, and then the HA shape had changed, and then these antibodies from 2014 will no longer recognize this 2016 virus. So that is the antigenic drift, and that's how the the virus can evade the host immune response. That's why that is the reason why we need to update the vaccines, and we need the annual vaccines to match with the virus evolution. So we have to evolve our B cell response as well. So I think I've answered how does it contribute to evade the immune system. So I haven't talked about the other innate immunity. There are lots of literature on how the virus can evade the immune system as well. And I've only answered in terms of antibodies, but there are viral proteins which can actually modulate the host immune response as well, and that's another mechanism how the virus is able to evade the immune system. Um, and yes, the reason for influenza vaccine update annually is also because of the antigenic drift, and then the antibodies previously generated no longer being active to the circulating strain. So it's not because of the antigenic shift. So the antigenic shift is slightly different. So for example, the antigenic shift, to give an example, so in 1957, there was H2N2 virus, which jumped from animals' zoonotic regions to humans, and then it spread widely. So the humans were immunologically naive against this hemagglutinin, and that's the reason why it widely spread as a pandemic, because of the naive population not being immunized before, or not seeing the HA or NA before. So that's the reason for the antigenic shift and the pandemic. Thank you.
Okay, thank you for the answer, Dr. Pramila Rijal. Because of the limited time, this session is end. Thank you very much for our remarkable speaker, Dr. Pramila. Give applause once again for Dr. Pramila. And then for appreciate our remarkable speaker, Dr. Pramila, we will give a digital certificate, and then we'll show in the screen. We invite Prof. Tere for give a digital certificate, please.
Okay, thank you, Dr. Pramila, for your insight and very interesting presentation and topic today. And we hope we can collaborate in the future, maybe in joint research or supervision. Yeah. Okay.
Yeah,
Thank you
great,
very
yeah.
much, Dr. Pramila. Yeah.
Thank you very much, Professor Teresa,
Yeah.
and thank you for inviting me and giving this opportunity.
Thank you. Bye.
Thank you very much. Yeah, bye-bye.
Okay, thank you very much. I think this is the end of our decision. And now this is your time, Mr. Nabil.
All right, thank you so much, Dr. Vivin, for moderating this session. Everyone, please give once again another warm round of applause for Dr. Vivin for moderating this session. All right, just before I announce the next agenda, it is time that I give a short intermezzo once again. That is to announce the second group of people who will receive the door prizes. Will the committee in charge please share the screen? Process. Yes. So what do you guys think is the next agenda after this? Eating maybe? Another break time? Is it something serious maybe? It looks like everyone has serious faces here. Relax a little, guys. It's a Saturday, you know? Wow, what kind of door prize? We actually have a lot of door prizes to give out. In fact, again, just repeating what I said before, one of the door prizes is actually a voucher that is given by a dental clinic. For the treatment that the voucher is valid for, it is for teeth whitening, if I'm not mistaken. Yes, so that is one of the door prizes. In fact, the discount is actually pretty big, to be honest, right, Prof? Yes, it's actually a very big discount. So, here they are. The people who will receive the next batch of their prizes goes to Septian and Ilham and Amana. Is that two people or is that one person? I forgot. Congratulations to the people who have won the second batch of door prizes. You may contact the committee to receive your door prize towards the end of the conference. All right, now without wasting any more time, it is now time for the next agenda. The agenda that we are all waiting for, and that is the oral presentation. Wow. So, to all the participants of the oral presentation, you may now mobilize to your respected rooms based on the information that has been previously given by the committee. So, the oral presentation will be happening here in the main room, room 1, right in front of the conference room, and then room 2 and 3, which is on the side of room 1. There will be a sign on the door that will indicate which room that is. So, people, friends, you may now prepare yourselves and mobilize yourselves to your respected rooms. Those who are presenting in the main room here, you may stay in your seats. All right. We wish you the best of luck, everybody. Good luck. Okay, so everyone who is presenting in this room, kindly move forwards so that you can be closer to the screen and so that we can be involved in the discussion. So please move forward to any seat that is available in front to the offline participants. participants in the main room. We will now be beginning the oral presentation. So once again, my name is Nabil Ahmad Bafiuddin, and I will be the moderator for today's oral presentation in the main room. So those participants who are here offline, who should be here for the offline participants are participants under the name of Dr. Larasita Prameswari and Miss Dwi Wahyuning Asi. Are you already present? Oh, okay. For Miss Dwi Wahyuning, are you present already? All right. For now, we will just continue with the oral presentation. So we will now be starting off with the, before we actually start, I would like to remind all participants that the total time will be, that is given is 8 minutes, where you have 5 minutes to present your topic, and you have a 3-minute Q&A session with the judge, which in this case will be Professor Teresia. And for the presentation time, we will give a tolerance of 5 seconds. So if you go over 5 seconds, over 5 minutes, we will stop you, regardless if it's if you are presenting offline or via online. All right. So for the committee, are you ready for the first presenter? Yeah. All right, so we will begin with Dr. Larasita Prameswari, who will give a presentation on the topic of tissue biomarkers for predicting response to immune checkpoint inhibitor therapy in cancer. To Dr. Larasita, you now have 5 minutes to present your topic, and we'll begin when you say the first word.
Good afternoon, Professor and everyone. My name is Larissa Prameswari from Immunology program of Airlangga University. Today I'll be presenting my topic about tissue biomarkers for predicting response to immune checkpoint inhibitor therapy in cancers. In this topic, I focus on how biomarkers can help us to select patients who will truly can advantage of immune checkpoint inhibitor therapy. Next. For the first, so immune checkpoints like CTLA-4 and PD-L1 are regulatory proteins on T cells and tumors. These checkpoints make tumor cells take advantage to escape for the immune destructions. So the immune checkpoint inhibitors block these proteins and restore T cell mediated to anti-tumor response. But inhibitory immune checkpoint inhibitory therapy, only minority of patients take the advantage of this therapy. So this highlight the importance for predictive biomarkers to improve the response of immune checkpoint inhibitors therapy. For the first, I want to discuss about the CTLA-4. Next. CTLA-4 is expressed on T cells and play a key role in downregulating immune response. By blocking the CTLA-4, the therapy can activate T cells, but may cause severe immune-related side effects. And then CTLA-4 expression in tumor infiltrating lymphocyte is associated with better outcome in advanced melanoma. So in this therapy, only used in advanced melanoma. And this protein also often co-expressed with PD-1. So that indicating complex immunity of cancer environment. And this can be suggest potential for combination ICI therapy. Next. For PD-L1 immunohistochemistry, this protein can express on tumor and immune cells by binding PD-1 and with T cell activity. The tumor cells can affect the immune system. And most study biomarkers for anti-PD-1 and PD-L1 therapy explain that the therapy can block this, so that that can be restore T cells mediated for anti-tumor response. And for the measure the PD-L1 expressions by using two scoring system for the first tumor progression score and combine positive scores. And many studies suggest that higher PD-L1 is often linked to the better response, but not always predictive biomarker. So PD-L1 is an imperfect predictor. But despite limitation, PD-L1 immunohistochemistry is the most accessible biomarker in clinical use. Next. So this is the picture illustrated the how tumor cells and other cells releasing cytokines. This is the illustrated the how tumor cells can escape the immune system by express CTL by express CTLA4 that binds the B7 or more than CD28 in the dendritic cell. So it can inhibit the T cells to
All right,
activate
the
it.
time is now up. I apologize for that. Okay, we will now move on to the Q&A session that will go for 3 minutes. To Prof. Theresia, the time is now yours.
Okay, thank you. Thank you, Dr. Laras. I want to ask about your checkpoint inhibitor. Can you explain about the checkpoint inhibitors? What do you mean? And where the checkpoint will inhibit it? And where the checkpoint the checkpoint will inhibited by CTL4 or maybe can you explain about it?
Checkpoint inhibitor is a group of protein that izin Prof kalau misalkan pakai bahasa ini. Jadi kelompok checkpoint inhibitor ini adalah sebuah kelompok protein yang dapat diekspresikan oleh baik sel T maupun sel tumor yang berfungsi untuk yang mempengaruhi aktivasi dari sel T dalam melawan atau meregulasi dari sistem imun sendiri. Jadi checkpoint inhibitor ini lebih sering berguna dalam men-down regulation pada kasus autoimun, tapi karena pada kasus tumor cells mereka dapat mengekspresikan protein checkpoint yang dapat membuat mereka tuh bisa lolos dari sistem ini.
All right, thank you, Prof. Theresia, for the Q&A session, and also thank you to Dr. Lara Sita for your oral presentation. We will now move on to the next oral presentation given by Miss Dwi Wahyuning Asih, are you now present? Yes. Okay, you will be presenting on the topic of the identification of highly immunogenic types, antibodies, and cytotoxic T lymphocyte CTL on SARS-CoV-2 new variants. You have 5 minutes, starting as soon as you talk.
Okay, good afternoon, everyone. I'm Dwi Wahyuning. On this day, I want to show you about my little project about pre-in silico study with entitled Identification of Highly Immunogenic Epitope Antibodies and CTL from New SARS-CoV-2 variant. Next. Next. Yes. This is, we all know about the agent pathogen. This is a SARS-CoV-2. The this pathogen is cause COVID-19 pandemic and in 2020, and SARS-CoV-2 has a positive single strain RNA genome, and the genome is with and the genome have encodes 29 proteins, and the structural protein is apa namely envelope membrane nuclear capsid, and the main structural protein is spike. Next. And this is a number of mapping about the number of confirmed COVID-19 case report over the last 20 days as of 5 January 2025. And this the right side is the total number of COVID-19 case in Indonesia about for January 2025. And the three provinces with the highest report is Jakarta, East Java, and West Java. Next. And maybe next. And this is a prevalence of SARS-CoV-2 variant of interest and variant under monitoring as one as January of 2025. The most prevalent of variant is JN1 in global, and this is also applies in Indonesia. Report from Indonesia Ministry of Health until May 2024. Next. Nah, in Indonesia, vaccine development is used Sinovac, Pfizer, AstraZeneca, and Moderna, and this vaccine can cause trigger the emergence of new variant or mutation, and further studies to predict highly immunogenic epitope from new SARS-CoV-2 variant is needed, and the objective is to identify highly immunogenic epitope for antibodies epitope antibodies and CTL. Next. And this method with I use a server of ABC prediction from predict antibody epitope, and I use while CTL epitopes I use IEDB server, and then I use server Vaxigen for antigen for find antigenicity values. And next. Maybe next. Nah, this result about the antibody epitope identification. I use iPhone to highest score to highest peptide sequence with with score of ABC prediction is I with rectangle with rectangle. Terus and next, and this is result of antibody epitope from GN1 and apa new SARS-CoV-2 variant. And maybe next.
All right, that time is now up. I apologize for that. It is now time for the Q&A session that will be given by the jury. For that, the time is now yours, Professor.
Okay, thank you, Dr. Dwi Wahyuningsih. Yes, okay. In your track, in your PPT, you got the highest immunogenicity, yeah, in nucleocapsid. Yes, the highest, yeah, the highest in nucleocapsids of virus, yeah, SARS-CoV-2. And what's your opinion about this? After you got this antigen, that in virus the highest is in nucleocapsid. Yes.
Prof, mohon izin saya dengan bahasa Indonesia. Ya. Menurut saya mengapa diperoleh di nukleokapsid itu menurut penelitian, jadi studi saya ini sesuai hasilnya sesuai dengan penelitian oleh Chan et al 2020. Prof menyatakan bahwa the highest immunogenic epitope in antibodies is nucleocapsid. Jadi karena nucleocapsid merupakan protein yang jumlahnya abundant atau banyak sekali di virus SARS-CoV-2, Prof.
Yeah, okay, thank you.
Thank you.
Okay, thank you for your oral presentation and thank you, Prof, for the Q&A discussion. We will now move on to the presentation of the next participant, which will be given online via a video. For the participant whose name is Ali Zainal Abidin, who will be presenting on the topic of leronlimab as a promising precision therapy targeting CCR5 in metastatic triple-negative breast cancer, a narrative review. For the committee in charge, please kindly prepare the video. If the duration of the video is more than 5 minutes, kindly cut the video at 5 minutes, 5 seconds.
Good morning, ladies and gentlemen, of the judges and committee of our Immunology Day for Erlangga Immunology International Conference. My name is Alizan Abidin, and I'm honored to stand my team to present our narrative review with the title "The Potency of Leronlimab as a Precision Therapy in Targeting CCR5 for Metastatic Triple-Negative Breast Cancer." TNBC is the most aggressive breast cancer subtypes characterized by the absence of estrogen receptor, progesterone receptor, and HER2 expression, higher recurrence rates, early metastasis, and poorer prognosis compared to other subtypes. The case is about 15 to 25% of all breast cancer cases worldwide. Metastatic TNBC or mTNBC present a major clinical challenge due to the lack of targeted receptors. Standard hormonal and targeted therapies are ineffective and limited in TNBC. Moreover, chemotherapy remains the mainstay of systemic treatment, but often result in short-lived responses and drug resistance. So, the urgent need for novel, effective targeted therapies has led to increased interest in precision medicine approaches. CCR5 is a G protein-coupled receptor that plays a crucial role in regulating immune cell migration and inflammatory responses. CCR5 normally helps direct immune cells to site of infection or injury by guiding their movement, a process called chemotaxis. However, in cancer, especially TNBC, CCR5 is often overexpressed. This overexpression is linked to more aggressive tumor behavior and worse patient outcomes. CCR5 signaling also active enzymes like matrix metalloproteinase, which break down the extracellular matrix and promotes metastasis. In TNBC, the CCL5-CCR5 axis play a key role in metastasis. CCR5 responds to CCL5 signals in the tumor microenvironments, promoting cancer cell migration, infection, and angiogenesis. This chemokine receptor interaction helps guide tumor cells to distant organs like lungs, liver, and bones, leading to poor clinical outcomes. Therefore, CCR5 is considered a promising target for therapy in mTNBC. Larolimab is a humanized monoclonal antibody that targets CCR5. By blocking CCR5-CCL5 interaction, it disrupts signaling pathways that drive tumor cell migration and infections. In preclinical and early clinical studies, larolimab has shown potential to reduce metastasis and enhance immune response, especially in CCR5-positive mTNBC. Ongoing trials, including combinations with carboplatin, support its role as a promising precision therapy for patients with limited options. Based on study result by Joe et al. 2021, showed that larolimab significantly reduced CCL5-induced calcium influx compared to controls, showing it effectively blocks CCR5 signaling, thereby suppressing cancer cell migration and metastasis. Moreover, this study was also conducted in vivo using model mouse. In a mouse model, larolimab significantly reduced breast cancer metastasis to the lungs, as shown by lower bioluminescence signals, and this suggests that larolimab effectively blocks CCR5-CCL5 signaling, supporting its potential as precision therapy for mTNBC. Larolimab offers several key advantages. It targets CCR5 specifically, a key driver of metastasis in TNBC, with minimal off-target effects and low toxicity profiles. It shows strong synergy with carboplatin, effectively inhibit metastasis and help overcome therapy resistance. Larolimab also enhances anti-tumor immunity and fits well within precision medicine by targeting patients with CCR5-positive tumors. Despite its promise, larolimab still faces several challenges. Some patients report side effects, including fatigue and, more rarely, blood-related toxicities. Determining the right dose and identifying patients with the high CCR5 expression is essential. Its use with other therapies also requires careful evaluation. Lastly, cost and limited accessibility may hinder widespread adoption. Moving forward, larolimab feature includes better patient selecting using CCR5 biomarkers, exploring combination within chemotherapy or immunotherapy agents, and validating results through larger clinical trials. Real-world studies and improved drug formulation are also key to increasing access and long-term success. In conclusions, larolimab shows strong promise as a precision therapy for mTNBC by blocking CCR5. While challenges like safety, dosing, and access remains, future efforts should focus on biomarker-guided use, effective combination, and larger clinical. Thank you for your attention. I would like to be pleased to address any question or engage in future discussion at this time.
All right, thank you for the oral presentation. To Mr. Ali, are you in the Zoom and can you hear us? Mr. Ali, can you hear us?
Yeah.
Yes, you can hear us. All right. It is now time for the Q&A session for 3 minutes, which will be conducted by the judge. To Professor Teresia, the time is yours.
Thank you. Mr. Ali, yeah, I interested with your research. Yeah. CCR5 is a chemokine for recruitment the pro-inflammatory cells to the site of inflammation. And what's the related with the cancer, especially with the metastasis?
Thank you for the questions. CCR5 is a chemokine receptor that plays a critical role in cancer cells, including in infection, migration, and metastasis, particularly in immunosuppressive subtypes like TNBC. It is frequently overexpressed in TNBC cells and facilitates the recruitment of immunosuppressive cells such as Tregs and MDSGs that can promoting a tumor permissive microenvironment. Targeting CCR5 disrupts the CCR5 signaling axis that drives metastatic spread.
Okay, thank you.
Thank you.
All right, thank you, Mr. Ali, for your presentation and Professor for your question. We will now move on to the next video presentation, which will be given by which will be given by Venanzi Victari on the topic of the potency of P95HER2-TCB as a promising new therapy for advanced HER2 plus breast cancer, a narrative review. For the committee in charge, please play the video.
Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen, all of the judges and the committee of the 4th Erlangga Immunology International Conference. My name is Venus Victoria and I am pleased to present my team's topic of the potency of P95HER2 TCB as a promising new therapy for advanced HER2-positive breast cancer, a narrative review. HER2-positive breast cancer is characterized by aggressive proliferation and poorer prognosis, which accounts for 15 to 20% of all breast cancer cases. HER2 protein drives tumorigenesis and cancer projection via intracellular signaling pathways activation such as PI3K-AKT and MAPK. There are several therapies applied to HER2-positive breast cancers, of which trastuzumab and pertuzumab resulted in improved clinical outcomes. However, advanced HER2-positive breast cancer remains a challenge due to its resistance. Two of the resistance mechanisms involve alteration in the pathway of PI3K-AKT mTOR and the presence of P95HER2. P95HER2 is a truncated form of HER2 receptor that is constitutively active, therefore serves as a promising and novel precision immunotherapy target for advanced HER2-positive breast cancers. Currently developing immunotherapy is T-cell bispecific antibodies, which could simultaneously bind to T-cells and P95HER2, redirecting the T-cells to specifically destroy cancer cells with overexpression of P95HER2. In vitro and in vivo studies which have been done showed a promising result, confirming the efficacy of P95HER2 TCB, which has a potent anti-tumor effect. These existing preclinical data pushed for two non-phase 1 clinical trials. Previous preclinical data by AP Biosciences showed that their P95HER2 TCB resulted in no adverse side effects in monkeys at the highest dose, with T-half of 108 hours in the dosage of 25 to 100 mg per kilogram body weight. In addition, preclinical data of technitomy showed evident tumor regression with no observed toxicity. To our knowledge, its phase 1 clinical study is currently in the approval process. Despite its superiority, P95HER2 TCB still possesses several limitations and challenges. P95HER2 expression remains heterogeneous and there are still difficulties in its detection, risking in a false negative or false positive result. Moreover, this P95HER2 TCB immunotherapy has no effect on cells with low HER2 expression or even on HER2 negative. There are many HER2-positive therapies either applied or in development, but P95HER2 TCB immunotherapy still take the spotlight, particularly when compared to therapies utilizing trastuzumab or tyrosine kinase inhibitors. Unlike trastuzumab, which is unable to cross blood-brain barrier, P95HER2 TCB can do the opposite, making it an ideal therapy for patients with breast cancer which has metastasized to the brain. Moreover, systemic side effects which could occur from the usage of tyrosine kinase inhibitors is not yet observed in P95HER2 TCB due to its high specificity. In conclusion, P95HER2 TCB is a promising new therapy for advanced HER2-positive breast cancers, particularly those with overexpression of P95HER2. And although this immunotherapy still needs further study, existing preclinical data have shown its high specificity as well as its ability to cross blood-brain barrier, making it more preferable than the previous applied therapies. Thank you, and we appreciate the opportunity to further explore.
All right, thank you for the oral presentation. To Miss Finanzi, are you in the Zoom? If you are in the Zoom, can you reach out to us?
Yes, I'm here.
Okay, thank you for your response. Okay, we will now have a Q&A session for 3 minutes by Professor Teresia. Prof, your time is yours.
Okay, thank you. Dr. Fenansi. Okay. Yeah, thank you for your innovation. Yeah, I want to ask about your HER2. Is it HER2 is especially for breast cancer, or it is possible to others? And what's the function of HER2?
All right, thank you, Professor, for the question. I would like to answer. So on this review article, we do focusing on HER2 expressed in breast cancers, either metastatic or not. And HER2 is as a receptor that regulates the signaling pathway by the MAPK or PI3K, and it's a very important receptor which is commonly found overexpressed in breast cancer patients, and it's a very important target if one want to develop a medication against breast cancers.
Okay, thank you.
Okay, thank you, Professor, for your Q&A session. We will now be moving on to the next presentation given by Ms. Putari Zawalia Munarwi, who will be giving a presentation on the topic of P16 and Ki67 immunohistochemistry assay for early detection of cervical cancer. To the committee in charge, can you please share the video? Thank you.
Assalamualaikum warahmatullahi wabarakatuh. Thank you for the opportunity to present my study today. My name is Putri Zawalia Munarwi. I will present about P16 and Ki67 immunohistochemistry assay for early detection of cervical cancer. As we know, cervical cancer remains a major global health challenge. It is the second most common cancer affecting women worldwide. Its main cause is infection by the human papillomavirus or HPV. Although HPV is the main cause, not all HPV types lead to cancer. Only certain high-risk types, such as HPV 16 and 18, have oncogenic potential. The P16 protein is an important biomarker found in cells infected with high-risk HPV. It is not expressed in low-risk HPV infections, but is highly expressed in high-risk infections. To understand why P16 is overexpressed, let's look at the molecular mechanism that occurs in high-risk HPV infections. High-risk HPV produces the E7 protein, which directly inactivates the PRB protein normally responsible for preventing uncontrolled cell division. When PRB is inactivated, E2F remains active and cells continue dividing. In response, the body increases P16 production to try to stop this. Unfortunately, the increase in P16 cannot stop cells proliferation because PRB has already been inactivated by the virus. As a result, excessive P16 expressions occur in infected tissues. This is why strong and diffuse P16 staining serves as a surrogate marker to detect oncogenic transformation caused by high-risk HPV. Additionally, high-risk HPV-infected cells show a more proliferative index than low-risk HPV. Ki67 is a protein that is expressed in proliferating cells. Therefore, the Ki67 proliferation index can serve as a marker for higher proliferating cells that are commonly found in high-risk HPV-infected cells. This is why the combination of P16 and Ki67 provides a highly effective approach for early detection of cervical cancer. Using P16 and Ki67 staining together gives a clearer picture of cells that have undergone oncogenic changes. This combination has proven to be beneficial as an early marker of cervical cancer. In conclusion, immunohistochemical staining of P16 and Ki67 can be used as a strong indicator of HPV-driven oncogenic transformation. Applying P16 and Ki67 immunostaining on cervical smears will have benefits over HPV DNA detection, since HPV DNA detects all types of HPV infections while P16 and Ki67 immunostaining only show positive result in high-risk HPV infected cells. This approach complements current screening method by not only identifying the virus, but also assessing the biological response of affected cells. This is particularly valuable for strengthening biomarker-based cervical cancer screening strategies. There are some of the reference I used in this study. Thank you for your attention. I hope this presentation has provided useful insights into how P16 and Ki67 can enhance early detection of cervical cancer. Wassalamualaikum warahmatullahi wabarakatuh.
All right, thank you for your presentation. Miss Putri Zawalia, are you in the Zoom?
Yes, I am.
All right, thank you. We will now have a Q&A session for 3 minutes to Professor Theresia. The time is now yours.
Thank you, Putri. Yeah. As we know that Ki67 is a protein for divided for cells. And would you explain, is it Ki67 is found in the HPV infection or in cervical cancer by infect HPV? You know, I mean?
Yes.
Okay.
Okay, thank you for your question. So K67 is not produced by the HPV virus itself, but it is a cellular marker that becomes highly expressed in cells undergoing abnormal proliferation due to high-risk HPV infection. When high-risk HPV infects cervical cell, the viral oncoprotein A6 and E7 disrupt normal cell cycle control, leading to uncontrolled cell division. This abnormal proliferation results in elevated Ki67 expression, particularly in cervical intraepithelial neoplasia, in CIN2 or CIN3 and cervical cancer. So Ki67 is not found in the virus itself, but in the host cells affected by the oncogenic effects of HPV infections. Thank you.
Okay, thank you.
All right, thank you, Professor, for your question. We will now be moving on to the next participant for who will whose name is Agustin Wulansuci Darmayanti, who will give a presentation on the topic of systematic literature network analysis, oral microbiome, and focal infection. For the committee in charge, please kindly share the screen. Thank you.
Assalamualaikum warahmatullahi wabarakatuh. I'm going to present our systematic literature network analysis about oral microbiome and focal infection. This is the content of our presentation. The oral microbiome comprises over 700 distinct microbes, particularly bacteria, which form complex biofilm essential for maintaining oral and systemic health. The disturbance of oral microbiota balance can lead to oral infectious disease that called dysbiosis. Oral dysbiosis induce oral chronic inflammation associated with various systemic disease. This study used SLA approach to explore more deeply the relationship between the oral microbiome and focal infection by reviewing articles from the Scopus database over the past decade. SLA is an effective method to evaluate research development in this theme. The data was taken online using the advanced of the Scopus page using this keyword and that published in 2016 to 2025 and original research. The data analyzed by Viewer and published on Paris and was conducted by Prisma. Annual publications have experienced a continuous increasing trend, except in 2018. The trend in 2025 cannot be seen because it's still in current year. The most published in 3 main journals. United States, Japan, China, Germany published this topic more than the others. Based on the field, medicine is more related to this theme than the others. Iwauchi et al. 2019 had a total 68 citation or the most citation. Bibliometric analysis showed for cluster, you can scan the barcode to know the item of the cluster, its topic in its cluster related with the others. The main topic in cluster 3 or blue is oral microbiome related to inflammation both in oral and systemic. The figure shows several oral microbiota are still related to focal infection. This indicated that has not been explored in this topic and become novelty. The visualization showed mold flora, microflora, inflammation, periodontitis, and dysbiosis are more discussed deeply than the other topics. This is the Prisma chart. This is the result of review. You can scan the barcode. Most of the studies was the observational that discuss about the oral infection to systemic disease, especially Alzheimer. Interestingly, 4 of 31 articles link that oral dysbiosis is related to gut dysbiosis that can cause infection in several organs. This table of main finding, you can scan the barcode, and the main finding found that oral microbiome not influenced by ill condition and change, but the level of inflammation was higher in this sick group. Both gram-positive and gram-negative cause a focal infection which triggers systemic inflammation and translocated to other organ by damaging epithelial integrity. The oral microbiome plays a vital role in oral and overall of body health. Oral microbiome interact the two complex ways in the body cell and can influence the variety health condition, especially in dysbiosis condition. Focal infection occur when oral microbiota spread to other part of the body and trigger an inflammatory reaction through inflammation pathway. The microbiome can escape and break down the epithelial integrity, enter to bloodstream, spread, and translocate to other organ. Briefly, the oral microbiome is a potential agent that can trigger focal infection in various organs. Thank you.
All right, thank you for your oral presentation. To Ms. Agustin, you are in the Zoom? Okay, yes, thank you. To Professor Teresia, the time is now yours.
Okay, thank you, Agustin. Yeah. I interested with your opinion. Yeah.
Thank you.
Yeah. Microbiome and how big the microbiome to change into the focal infection. How big and how and why in change into the focal infection.
Okay, thank you, Prof, for the question. I will try to answer. Oral microbiome normally is flora normal that have ability to be pathogen when the microenvironment oral microenvironment there is a imbalance, especially in pH and acidity that produced by oral biofilm, especially in subgingiva and that inhabitant of anaerobic bacteria that influence the environment around the subgingiva, and it will be to be dysbiosis. So there is imbalance between pathogen and also the flora normal. And then the factor virulence of bacteria, both in positive and negative bacteria, it will be distract the integrity of endothelial and epithelial, and then it will make dissemination and also make a systemic inflammation, and it will be translocated to the other organ. Especially in this review, most of bacteria in oral microbiome can be found in brain and also in gut, make cause Alzheimer disease and also uh, uh, disorders in gut. Thank you.
Okay, thank you.
Right. Thank you, Professor Theresia, for your question. We will now be moving on to the next oral presenter. Before we do, I would like to confirm, Ms. Calixte Kiyuzuzu, are you in the Zoom? Miss Calixte Kuyuzuzu, are you there? Oh, okay. Okay,
Yes, good afternoon.
all right. Just to confirm. So, okay, we will start off with your oral presentation video that you have sent us. For the committee in charge, you can share the video now. Thank you.
Okay, perfect.
All right. You can start your presentation. You have 5 minutes, and that timer will start as soon as you talk. To Mr. Calixte Kyuzuzu, are you there?
Thank you. Sorry, I was muted and I couldn't unmute myself. Can you hear me now?
Yes, we can hear you. Thanks so much. You can start as soon as you talk.
Okay, perfect. Thank you very much. So, good afternoon, everyone. I am Calixte Chususo. I graduated from Alanga University in 2021. And today I'm presenting my research that aimed to characterize and compare humoral immune responses in mice immunized with crude antigen of Plasmodium berghei schizon-rich antigen and Plasmodium berghei with all stages. Next. So I'm currently affiliated with the Center for Population Health at the University of Global Health Equity, Kigali, Rwanda, but also Immunology Program Postgraduate School, Erlanga University, where I graduated from. Next. So basically, currently, malaria is a big global health issue that has to be dealt with, and especially in the lower and middle income countries, including Rwanda, where you reside currently. And so far, there is no approved vaccine for malaria, apart from the RTSS, which is R21, which has been rolled out in different 19 countries in Africa. However, there is no effective vaccine that has been approved to date. Next. With that, as I said, there is a need to conduct studies around malaria to find if there will be a way of finding effective vaccine. So this study specifically aimed to compare the IgG levels in mice immunized with crude antigen and to characterize the proteins in which those antibodies were recognized. Next. So basically, this is how we prepared the crude antigen. We had to inject phenylhydrazine chloride to the mice prior infection with Plasmodium bedgae, infected blood. And then we had to add to collect blood, and then you have to freeze them at negative temperature. And then we had to wash blood using PBS and saponin. And we also added protein inhibitors, and then we sonicated 3 times for 5 minutes. This is the procedure of preparing the antigen. But for the antigen, which contained all stages of Plasmodium, we did not inject mice with phenylhydrazine chloride prior to the infection. Next. So after getting the crude antigen, we had to check the concentration of those antigen using nanodrop for spectrophotometer, where the Plasmodium bergaceous rich antigen had a concentration of 4.75 milligram per milliliter, and the Plasmodium bergaceous ring trophozoite schizont gametocyte antigen had this concentration of 4.95. And then this had to be emulsified into the complete and incomplete friends adjuvant prior to the immunization. And then you had to titrate the antibodies using ELISA and to characterize the proteins using the SDS page and Western blot. Next. So this is how we created different groups of mice. Group 1 that was injected with PBS-AG, which is an antigen rich in schizont stage, and the group 2, which was immunized with antigen rich in all stages. Next. And we did the immunization, of course, the first immunization with antigens that were emulsified in complete friends adjuvant, and for the boosting immunization, after considering the interval of 5 days, we were immunizing with antigen that were emulsified in with incomplete friends adjuvant. Next. So the analysis was done using SPSS. ANOVA was considered to do the analysis, and then the p-values of less than 0.05 was considered statistically significant. Next. So the results, the Plasmodium bergaceous antigen, rich antigen, demonstrated higher antibody titers compared to the Plasmodium ring trophozoite gametocyte antigen. And comparing the two groups, there was a statistical significant difference in the antibody titers observed, but also the difference between the group that was immunized with PBS-AG and the group that was immunized with PBRTSG-AG and the control group. There was a statistical significant difference in antibodies titer that were observed. So the antibody titer from the Plasmodium bergaceous rich antigen
The time
varied
is
from
now
0.
up. Yes, I'm sorry, the time is now up. Can you go straight to the conclusion?
Okay, perfect, no problem. Next slide. So as we were immunizing, the antibody titer were increasing. Next.
Strict
then
conclusion.
and from there, we concluded that the Plasmodium or Bergay schizont-rich antigen is a promising vaccine candidate, as it recognized the molecular weight of 66, and it can be further explored for the purpose of developing a vaccine for malaria. Thank you.
All right, thank you for your oral presentation. We will now be moving on to a Q&A session for 3 minutes by Professor Teresia. The time is now yours.
Okay, thank you, Sisi. How are you?
I'm good. How are you, Prof?
Yeah. Okay, Sisi. I want to know about your research. Yeah. IgG. It means that you have found the IgG for malaria. And how do you sure that this antigen can to be a vaccine? Yeah. Have you done about the immunogenic?
So basically, we did not test the immunogenicity of it, but of course, by inducing the production of polyclonal antibodies against the antigen, it was an indication that it can be a potential vaccine candidate, but also considering the molecular weight of that antigen that we prepared, and the fact that it was recognized by both antibodies, either antibodies against all stages and antibody against the schizont stage. So considering its foreignness, its molecular weight, and its antigenicity and ability to induce the production of antibodies, polyclonal antibodies, of course, yeah, it can be considered as one of the potential vaccine candidate, considering that it's a protein and it's high molecular weight.
Okay, I see. That you found that the IgG, this is immunogenic with antibody. Yeah.
Yeah, basically the protein was immunogenic. That's why it induced the production of antibodies. And again, IgG, it has a protective function in the body. So yeah, as the high titer of antibody was produced, it is promising. However, further exploration and further research should be conducted to make sure that the protein is inducing the high titer of antibody, yes, but again, we have to explore further.
Okay, thank you, Sisi. See you. Bye.
All right, thank you, Professor Teresia, for your question. We will now be moving on to the next oral presentation that will be given by Ms. Amana, who will be giving a presentation on the topic of immunoglobulin G and E profiles in children with intellectual disabilities, insights into mucosal immune responses, and oral systemic health. For the committee in charge, please kindly share the video. Thank you.
For the first Assalamualaikum warahmatullahi wabarakatuh. Thank you everyone, and thank you to the committee for giving me the opportunity to present today at the World Immunology Day event. My name is Amana. I'm the researcher at Medical Faculty Swadaya Gunung Jati Cirebon. I'm so excited to share my insight on IgG and IgA profile in children with intellectual disability, insight into mucosal immune response and oral systemic health, and how it's relevant to immunology area. For the first slide, it's talk about the background on this research. On this research, we are focused with oral mucosa. The oral mucosa initiating the innate and adaptive immune response. With the marker of this research, it's IgG and IgA. IgG and IgA is secreted by immune cells with the salivary glands and mucosal associated lymphoid tissue, or we call it MALT. While IgA is associated with allergic response and inflammation triggered by allergen or the parasitic infection. This immunoglobulin or IgG are reliable biomarker for assessing mucosal immune profile and their implication for the oral systemic health. For the data research before, they show that the children with the intellectual disability and the children with autism have a poor oral hygiene, increasing the mucosal inflammation and opportunistic infection. For this elevated, the IgG level in saliva have been correlated with the pro-inflammatory cytokine and oral inflammation with a potential marker of chronic immune activation and salivary IgA level with the reflex dysregulation T helper mediated allergic response. So this research, we are non-invasive methods such as salivary analysis are particularly valuable for accessing immune function in children with intellectual disability and given the challenge associated with the invasive procedure like a blood sampling. So the method on this research is from the saliva from the respondent. The respondent is from the children with intellectual disability, and then the control group from the SLB Budi Utama in Cirebon. The sample were collected in the morning following a nighttime fasting, and then the saliva sample were stored in sterile container and then examined using ELISA procedure. The ELISA procedure, we have analysis with the IgG. So the result from the data distribution show that the most common gender in control group with 60% and the children with intellectual disability with 53%, and from the age criteria, so the control group most common it's 12 years with 41-38%, and the intellectual disability with the same criteria, the most common it's 10-12 years from the 44.5%. And then we continue the analysis of increasing in IgG level and IgA level. We are using the Mann-Whitney, and then the results show the significant difference in IgG and IgA levels between the control group and the children with intellectual disability. So we continue the result. The data show the significant difference in the control group in terms of IgA and IgG. The data show the significant difference in IgA and IgG level between the control group and the other group, but the highest IgG level was identified with the children with intellectual disability with a maximum value with mean in 46, and the highest IgA level was observed in the control group with a maximum value is 704 with a level of IgA. So from this discussion, the study revealed the significant difference in salivary immune level between the children with intellectual disability and healthy control. So from that data indicating the distinct immune profile, the children with intellectual disability had a higher average salivary IgG compared the control group. With a maximum IgG level, it's more in children with intellectual disability.
Right, the time is now up.
Data suggesting the
Yes, thank you. We will now move on to the Q&A session. For Ms. Amana, are you in the Zoom?
Yes.
All right, welcome. We will now proceed to the Q&A session to Prof.
Okay, thank you.
Yeah, thank you, Ms. Amana. Your research, this is your research. This is your research. Yes, okay. And what's your opinion Yeah, with your research, with your research about the immune system in the children with intellectual disability. What your opinion
Thank
after
you
you
for
got result the research?
Okay, thank you for the question, Professor Teresia. The research conclusion, I suggest this research have the impact for the children with intellectual disability. The impact for the marker IgG and IgA is the have immunological marker with intellectual disability is have apa namanya? Maaf, sorry. They have a response can influence health outcomes that may indirectly affect the neurodevelopment or quality of life in these children. And then they have consist of below IgA and IgG might relate to children with intellectual disability, considering of immune dysregulation, inflammation, and associated condition.
It means that the intellectual is it related with the immune system in IgG and IgA? Can you explain?
Yeah. The children with IgG and IgA, the children with intellectual disability have a neuroinflammation condition with not have a chronic driven by IgA may contribute the neuroinflammatory process, potentially maybe worsening cognitive outcomes for the children with intellectual disability. Maybe the marker for IgA and IgG have a clinical relevance with allergic condition. And then the monitoring IgG, it's have monitoring clinical relevance with the inflammation like.
Okay, thank you.
Okay, thank you, Professor Teresia.
All right, thank you for your presentation and for the Q&A session. We will now be moving on to the final presentation given by Mr. Muhammad Alwino Bayo Firdausi, who will be giving an oral presentation on the topic of applications in oral surgeries of demineralized dentine membrane in promoting osteogenesis via plasma cell differentiation, an experimental study. To the committee in charge, please kindly share the video. Thank you.
Demineralized dentine membrane promotes osteogenesis via plasma cell differentiation of craniofacial critical size defect. Oral and maxillofacial surgeon frequently encounter mandibular abnormalities. Congenital deformities, cancer, trauma, and infection are all examples of pathological processes that cause these abnormalities. Critical size defects are incapable of self-healing or self-recognition of more than 10% of the lost bone. To restore form, masticatory function, and aesthetics, the optimal mandibular construction is required. Infection, surgical procedure, tumors, trauma, and congenital defect can all hard bone, which is one of the body's tissue. Oral bone defect can be caused by surgical procedure such as tooth extraction, periodontal disease, trauma, and infection. Small bone defect can be treated by bone graft, substitute materials, growth factor, or injection of blood derived from bone marrow. Many studies are currently being conducted on the administration of stem cells for the treatment of the bone defect. In the case of massive bone, surgery is required to reconstruct the missing bone so that the line can function. Treatment of extensive bone defect is different into osteogenic cell and contribute to the formation of osteoid. A network of bone, the act as sites for bone deposition, is created by osteoid mineralization. The guide bone regeneration membrane must go through 7 stages, including cell exclusion, tenting, scaffolding, stabilization, and framework in order to achieve the best result. $60 blister rats weighing 250 until 300 grams, 2 until 3 months old, and it's good health with no disability were housed in cage for 7 days to adjust to their new surrounding. Rats are kept in animal cage place in isolated and locked room with good ventilation and natural lighting. The material was taken from the Surabaya City Uptown, processed at the Dr. Sutomo Surabaya Hospital Network Bank. DDMM is a developmental material that is processed directly from fresh bovine dentin. The ethical boards approved the experimental staining. Following that, plasma cells were counted using a 400 magnification microscope with 9 field of view. The study characteristics are present in table. Histological result based on graphic 1. The highest number of plasma cells in the bone defect was obtained on the 3rd day in the defect treatment group implant with PPCM membrane and bone graft, while on the 7th day in the defect treatment group implant DDMM membrane and bone graft. The HB of plasma cell can be seen in figure 2. The treatment group with the defect was implanted with bovine pericardium collagen membrane and graft. The historical findings from this study were examined to determine the impact of various membrane type and bone graft on plasma cell response in the therapy of bone defect. The findings show that the highest number of plasma cells in the bone defect was observed on the 3rd day in the group treated with PPCM and a bone graft. In contrast, the group treated with DDMM and bone graft exhibit the highest number of plasma cells on the 7th day. The difference in plasma cell count between the DDMM membrane and the negative control group without membrane implantation was significantly higher. It can be concluded that the DDMM membrane can increase plasma cell to help fight pathogens so that bone repair can occur. According to the findings, both the PPCM membrane and the GBM membrane, when used in conjunction with bone graft, can affect plasma cell activity and perhaps improve bone healing when treating bone defect.
For the committee, can you check if there is any other part of the presentation? Okay, Mr. Alvino, are you in the Zoom? Or Mr. Alvino, are you in the Zoom? He is not in the Zoom. Final call, Mr. Muhammad Alvino, are you in the Zoom? Mr. Alvino? Mr. Muhammad Alwino Bayo Firdausi. He's not in the Zoom, okay. All right. This is to be noted that Mr. Alwino is not in the Zoom. So we will now finish today's oral presentation. We thank all of the participants for participating in today's oral presentation, whether you are participating offline or online. We wish you the best of results. Prof. Tere, would you like to say some words? Oh, no, that's it. All right. So as we wait for, we will now wait for the other participants to come back into the main room as they also finish their oral presentation. We hope that you receive the best result. Thank you.
Indonesia memiliki mimpi besar menjadi Indonesia maju di tahun 2045. Sekolah Pasca Sarjana Universitas Airlangga memiliki 8 program studi multidisiplin, hadir menjadi institusi berkelas dunia dengan kemampuan untuk mengembangkan pemimpin-pemimpin transformatif yang berdampak bagi dunia. Selamat datang di Sekolah Pasca Sarjana Universitas Airlangga, dan inilah program studi yang kami kelola.
Saya Profesor Dr. Teresia Indabudi sebagai Koordinator Program Studi Magister Immunologi Sekolah Pasca Sarjana Universitas Airlangga. Prodi Immunologi merupakan satu-satunya program studi Magister Immunologi di Indonesia. Prodi kami ini adalah multidisiplin, jadi bisa dari berbagai keilmuan. Sekolah-sekolah yang berkaitan dengan imunologi bisa mengikuti program studi imunologi di Universitas Airlangga. Kami punya kompetensi yaitu bahwa mahasiswa-mahasiswa yang mengikuti program studi imunologi ini bisa langsung terjun di dalam penelitian-penelitian yang mengikuti program dari penelitian dosen-dosen yang ada. Sehingga mahasiswa semakin mempunyai wawasan yang semakin banyak di bidang imunologi. Dan juga kami juga menyelenggarakan salah satu seminar yang kita sebut sebagai World Immunology Day, merupakan satu-satunya juga seminar yang kita selenggarakan baik itu tingkat nasional maupun internasional.
Kita disupport dengan adanya penelitian berpayung, pendanaan yang support luar biasa dari Pasca Sarjana Universitas Airlangga. Kita juga sebagai mahasiswa mendapat support untuk publikasi dan juga melalui pelatihan-pelatihan berkelas Scopus dari Q1 sampai Q4.
Karena imunologi itu banyak bertanya, itu PR-nya mahasiswa online harus lebih aktif dibandingkan kalau mahasiswa offline, dosen ada di depan kita dan kita tahu, oh seperti ini, oh seperti ini.
Magister Immunologi Pasca Sarjana Universitas Airlangga, hebat!
Ini dia alat yang bisa bikin kulit kamu makin sehat dan cantik. Namanya Dr. Forms Hydro Nano. Alat yang bisa mengeluarkan air dalam ukuran nano berbentuk spray. Ini adalah yang pertama di Indonesia karena terdapat 3 batuan mineral yang berfungsi sebagai antiinflamasi dan antibakteri. Bukan cuma air biasa, tapi ini air hidrogen yang mampu menyerap ke dalam kulit sehingga membuat kulit jadi terasa kencang dan elastis. Produk ini dapat digunakan untuk seluruh bagian wajah, tubuh, kulit kepala, dan bagian perawatan tubuh yang lain. Manfaat Dr. Form Hydro Nano ini bisa untuk mencerahkan wajah dan membantu mencegah penuaan dini, membantu menghilangkan jerawat dan menghilangkan flek pada wajah, dan juga meningkatkan elastisitas kulit. Selain untuk menyehatkan dan membuat kulit jadi cerah, alat ini bisa jadi powerbank juga loh. Menarik kan? Buat kamu yang mau kulitmu cantik dan sehat, yuk langsung aja tanya ke Instagram @drwaterofficial.id.
Sibuk amat kakak cantik. Lagi ngerjain apa sih, Neng? Lagi ngerjain tugas nih. Kamu dari mana? Dari rumah. Daripada gak ada kuliah, lebih baik main di kampus. Tahu nggak? Ternyata autoimun itu sedang tren loh di anak muda. Katanya gara-gara gaya hidup. Oh iya? Iya, bener. Katanya makanan kekinian itu penyumbang utama terjadinya autoimun. Oh iya? Oh ya doang dari tadi juga dikasih tahu. Nah, selesai. Kenapa? Sehat. Sambut kali. Ayo, ikut aku. Mau mana? Udah, ikut aja. Di sini? Iya, di sini. Mau ngapain?
Udah daripada katanya sosmed, mending ikut kuliah tamu imunologi. Yuk.
Baik, hari ini kita akan bahas topik tentang autoimun. Kasus kanker itu juga merupakan salah satu dari autoimun, ya. Nah, dan apa penyebabnya dari autoimun? Dari apa yang sudah saya jelaskan tadi, ada yang mau ditanyakan kira-kira?
Ini kan membahas tentang autoimun. Nah, kalau misalnya tentang depresi itu ada tidak hubungannya dengan autoimun? Makasih, Prof.
Cocok sekali ya, kebetulan di kurikulum kami ada kurikulum tentang psikoneuroimunologi. Jadi keterkaitan antara sistem imun dengan psikis.
Izin Prof bertanya, nama saya Astika Jinansari. Apa benar Prof, makanan itu penyebab autoimun? Terima kasih.
Makanan itu apakah bisa merupakan salah satu penyebab autoimun ya? Bisa. Ya kenapa? Karena saat ini makanan-makanan modern sekarang ya, itu banyak yang artifisial.
Seru juga ya belajar imunologi. Bener banget, masuk imunologi aja. Lagian karena ini multidisiplin ilmu, bisa dari beragam keilmuan kok, nggak harus melulu kesehatan.
Bisa jadi bidang kesehatan bukan passion saya, tapi dunia imunologi begitu menggoda. Tidak hanya tentang keilmuan yang luar biasa atau karena multidisiplinnya, tapi dengan imunologi, aku, kamu, dan kita bisa berbuat yang terbaik untuk orang-orang tercinta.
Indonesia memiliki mimpi besar menjadi Indonesia maju di tahun 2045. Sekolah Pasca Sarjana Universitas Airlangga memiliki 8 program studi multidisiplin, hadir menjadi institusi berkelas dunia dengan kemampuan untuk mengembangkan pemimpin-pemimpin transformatif yang berdampak bagi dunia. Selamat datang di Sekolah Pasca Sarjana Universitas Airlangga, dan inilah program studi yang kami kelola.
Saya Profesor Dr. Teresia Indah Budi sebagai Koordinator Program Studi Magister Immunologi Sekolah Pasca Sarjana Universitas Airlangga. Prodi Immunologi merupakan satu-satunya program studi Magister Immunologi di Indonesia. Prodi kami ini adalah multidisiplin, jadi bisa dari berbagai keilmuan. Sekolah-sekolah yang berkaitan dengan imunologi bisa mengikuti program studi imunologi di Universitas Airlangga. Kami punya kompetensi yaitu bahwa mahasiswa-mahasiswa yang mengikuti program studi imunologi ini bisa langsung terjun di dalam penelitian-penelitian yang mengikuti program dari penelitian dosen-dosen yang ada. Sehingga mahasiswa semakin mempunyai wawasan yang semakin banyak di bidang imunologi. Dan juga kami juga menyelenggarakan salah satu seminar yang kita sebut sebagai World Immunology Day, merupakan satu-satunya juga seminar yang kita selenggarakan, baik itu tingkat nasional maupun internasional.
Kita disupport dengan adanya penelitian berpayung, pendanaan yang support luar biasa dari Pasca Sarjana Universitas Airlangga. Kita juga sebagai mahasiswa mendapat support untuk publikasi dan juga melalui pelatihan-pelatihan berkelas Scopus dari Q1 sampai Q4.
Karena imunologi itu banyak bertanya, itu PR-nya mahasiswa online harus lebih aktif. Dibandingkan kalau mahasiswa offline, dosen ada di depan kita dan kita tahu, oh seperti ini, oh seperti ini.
Magister Immunologi Pasca Sarjana Universitas Airlangga, hebat!
Elk Biotechnology is located in the beautiful and fast-developing Wuhan. Elk is a high-tech biological company specializing in immune and molecular biology research. We now own 1,600 square meter laboratory, having independent cell culture room and SPF animal room. We have 4 major platforms: pathology, immunology, molecular biology, cell biology. Our research focuses on immunochemistry, tumor research, neurobiology, cell cycle, signal transduction. We have 8,000 plus ELISA keys, 60 plus molecular biology reagents, 11,000 plus antibodies. We have certificated with ISO and CE mark. Each product has complete R&D, production and QC course, experienced R&D technical team, complete quality inspection support department, strict inspection process, providing customers with reliable quality scientific research products. We have fast delivery chain by FedEx or DHL. Since the company started overseas markets in 2018, we have reached stable cooperation with distributors coming from more than 50 countries and 5 continents. Our goal is to provide scientists with the most cutting-edge and cost-effective research tools. Our mission is to provide the best solution and the most reliable partners for researchers all over the world. We will always implement the concept of environmental protection in the production process.
Hai friends, perlu kamu ketahui bahwa radikal bebas dan polusi udara saat ini sulit untuk bisa dihindari. Nah, dampaknya bisa membuat kulit kering, kusam, dan penuaan dini. Nah, supaya tubuh kamu tetap fit, kamu butuh antioksidan dan anti-aging untuk meremajakan kulit sel. Dan juga meningkatkan kualitas kesehatan tubuh dan kulit kamu. Nah, solusinya adalah dengan Dr. Water Hydrogen yang mampu merubah air biasa menjadi air hidrogen. Nah, katanya nih super antioksidan bisa 176 kali lebih banyak dari vitamin C. Karena bisa membantu kamu untuk melancarkan peredaran darah, menurunkan risiko penyakit jantung, stroke, terapi diabetes, dan juga kanker. Dan dapat juga digunakan sebagai inhaler untuk proses regenerasi sel dan terapi yang lebih cepat. Oh iya, paling penting nih, aman dikonsumsi semua kalangan. Produksi ini dimiliki dan juga didistribusikan oleh PT Ultra Multisukses. Dan saya ingin mengajak kamu semua untuk menggunakan Dr. Water Hydrogen sebagai solusi kesehatan tubuh kamu. Nah, untuk info lebih lanjut bisa langsung cek di website berikut ini.
Indonesia memiliki mimpi besar menjadi Indonesia maju di tahun 2045. Sekolah Pascasarjana Universitas Airlangga memiliki 8 program studi multidisiplin, hadir menjadi institusi berkelas dunia dengan kemampuan untuk mengembangkan pemimpin-pemimpin transformatif yang berdampak bagi dunia. Selamat datang di Sekolah Pascasarjana Universitas Airlangga, dan inilah program studi yang kami kelola.
Saya Profesor Dr. Teresia Indabudi sebagai Koordinator Program Studi Magister Immunologi Sekolah Pascasarjana Universitas Airlangga. Prodi Immunologi merupakan satu-satunya program studi Magister Immunologi di Indonesia. Prodi kami ini adalah multidisiplin, jadi bisa dari berbagai keilmuan. Sekolah-sekolah yang berkaitan dengan imunologi bisa mengikuti program studi imunologi di Universitas Airlangga. Kami punya kompetensi, yaitu bahwa mahasiswa-mahasiswa yang mengikuti program studi imunologi ini bisa langsung terjun di dalam penelitian-penelitian yang mengikuti program dari penelitian dosen-dosen yang ada, sehingga mahasiswa semakin mempunyai wawasan yang semakin banyak di bidang imunologi. Dan juga kami juga menyelenggarakan salah satu seminar yang kita sebut sebagai World Immunology Day, merupakan satu-satunya juga seminar yang kita selenggarakan, baik itu tingkat nasional maupun internasional.
Kita disupport dengan adanya penelitian berpayung, pendanaan yang support luar biasa dari Pascasarjana Universitas Airlangga. Kita juga sebagai mahasiswa mendapat support untuk publikasi dan juga melalui pelatihan-pelatihan berkelas fokus dari Q1 sampai Q4.
Karena imunologi itu banyak bertanya, itu PR-nya mahasiswa online harus lebih aktif dibandingkan kalau mahasiswa offline, dosen ada di depan kita dan kita tahu, oh seperti ini, oh seperti ini.
Magister Immunologi Pascasarjana Universitas Airlangga, hebat!
Ini dia alat yang bisa bikin kulit kamu makin sehat dan cantik. Namanya Dr. Firms Hydro Nano. Alat yang bisa mengeluarkan air dalam ukuran nano berbentuk spray. Ini adalah yang pertama di Indonesia karena terdapat 3 batuan mineral yang berfungsi sebagai antiinflamasi dan antibakteri. Bukan cuma air biasa, tapi ini air hidrogen yang mampu menyerap ke dalam kulit sehingga membuat kulit jadi terasa kencang dan elastis. Produk ini dapat digunakan untuk seluruh bagian wajah, tubuh, kulit kepala, dan bagian perawatan tubuh yang lain. Manfaat Dr. Firms Hydro Nano ini bisa untuk mencerahkan wajah dan membantu mencegah penuaan dini, membantu menghilangkan jerawat dan menghilangkan flek pada wajah, dan juga meningkatkan elastisitas kulit. Selain untuk menyehatkan dan membuat kulit jadi cerah, alat ini bisa jadi powerbank juga loh. Menarik kan? Buat kamu yang mau kulitmu cantik dan sehat, yuk langsung aja tanya ke Instagram @drwaterofficial.id.
Sibuk amat kakak cantik. Lagi ngerjain apa sih, Neng? Lagi ngerjain tugas nih. Kamu dari mana? Dari rumah. Daripada gak ada kuliah, lebih baik main di kampus. Tahu nggak? Ternyata autoimun itu sedang tren loh di anak muda. Katanya gara-gara gaya hidup. Oh iya? Iya, bener. Katanya makanan kekinian itu penyumbang utama terjadinya autoimun. Oh iya? Oh ya doang dari tadi juga dikasih tahu. Nah, selesai. Ih, kenapa? Sehat. Sambut kali. Ayo, ikut aku. Mau mana? Udah, ikut aja. Di sini? Iya, di sini. Mau ngapain? Udah, daripada katanya sosmed, mending ikut kuliah tamu imunologi. Yuk.
Baik, hari ini kita akan bahas topik tentang autoimun. Kasus kanker itu juga merupakan salah satu dari autoimun, ya. Nah, dan apa penyebabnya dari autoimun? Dari apa yang sudah saya jelaskan tadi, ada yang mau ditanyakan kira-kira?
Ini kan membahas tentang autoimun. Nah, kalau misalnya tentang depresi itu ada tidak hubungannya dengan autoimun? Makasih, Prof.
Cocok sekali ya, kebetulan di kurikulum kami ada kurikulum tentang psikoneuroimunologi. Jadi keterkaitan antara sistem imun dengan psikis.
Izin Prof bertanya, nama saya Astika Juniansari. Apa benar Prof, makanan itu penyebab autoimun? Terima kasih.
Makanan itu apakah bisa merupakan salah satu penyebab autoimun ya? Bisa. Ya kenapa? Karena saat ini makanan-makanan modern sekarang ya, itu banyak yang artifisial.
Seru juga ya belajar imunologi. Bener banget, masuk imunologi aja. Lagian karena ini multidisiplin ilmu, bisa dari beragam keilmuan kok, nggak harus melulu kesehatan.
Bisa jadi bidang kesehatan bukan passion saya, tapi dunia imunologi begitu menggoda. Tidak hanya tentang keilmuan yang luar biasa atau karena multidisiplinnya, tapi dengan imunologi, aku, kamu, dan kita bisa berbuat yang terbaik untuk orang-orang tercinta.
Indonesia memiliki mimpi besar menjadi Indonesia maju di tahun 2045. Sekolah Pasca Sarjana Universitas Airlangga memiliki 8 program studi multidisiplin, hadir menjadi institusi berkelas dunia dengan kemampuan untuk mengembangkan pemimpin-pemimpin transformatif yang berdampak bagi dunia. Selamat datang di Sekolah Pasca Sarjana Universitas Airlangga, dan inilah program studi yang kami kelola.
Saya Profesor Dr. Teresia Indabudi sebagai Koordinator Program Studi Magister Immunologi Sekolah Pasca Sarjana Universitas Airlangga. Prodi Immunologi merupakan satu-satunya program studi Magister Immunologi di Indonesia. Prodi kami ini adalah multidisiplin, jadi bisa dari berbagai keilmuan. Sekolah-sekolah yang berkaitan dengan imunologi bisa mengikuti program studi imunologi di Universitas Airlangga. Kami punya kompetensi yaitu bahwa mahasiswa-mahasiswa yang mengikuti program studi imunologi ini bisa langsung terjun di dalam penelitian-penelitian yang mengikuti program dari penelitian dosen-dosen yang ada. Sehingga mahasiswa semakin mempunyai wawasan yang semakin banyak di bidang imunologi. Dan juga kami juga menyelenggarakan salah satu seminar yang kita sebut sebagai World Immunology Day, merupakan satu-satunya juga seminar yang kita selenggarakan, baik itu tingkat nasional maupun internasional.
Kita disupport dengan adanya penelitian berpayung, pendanaan yang support luar biasa dari Pasca Sarjana Universitas Airlangga. Kita juga sebagai mahasiswa mendapat support untuk publikasi dan juga melalui pelatihan-pelatihan berkelas Scopus dari Q1 sampai Q4.
Karena imunologi itu banyak bertanya, itu PR-nya mahasiswa online harus lebih aktif. Dibandingkan kalau mahasiswa offline, dosen ada di depan kita dan kita tahu, oh seperti ini, oh seperti ini.
Magister Imunologi Pasca Sarjana Universitas Airlangga. Hebat!
Biotechnology is located in the beautiful and fast-developing Wuhan. ELK is a high-tech biological company specializing in immune and molecular biology research. We now own a 1,600 square meter laboratory having independent cell culture room and SPF animal room. We have 4 major platforms: pathology, immunology, molecular biology, cell biology. Our research focuses on immunochemistry, tumor research, neurobiology, cell cycle, signal transduction. We have 8,000 plus ELISA keys, 60 plus molecular biology reagents, 11,000 plus antibodies. We have certificated with ISO and CE mark. Each product has complete R&D, production and QC course, experienced R&D technical team, complete quality inspection support department, strict inspection process, providing customers with reliable quality scientific research products. We have fast delivery chain by FedEx or DHL. Since the company started overseas markets in 2018, we have reached stable cooperation with distributors coming from more than 50 countries and 5 continents. Our goal is to provide scientists with the most cutting-edge and cost-effective research tools. Our mission is to provide the best solution and the most reliable partners for researchers all over the world. We will always implement the concept of environmental protection in the production process.
perlu kamu ketahui bahwa radikal bebas dan polusi udara saat ini sulit untuk bisa dihindari. Nah, dampaknya bisa membuat kulit kering, kusam, dan penuaan dini. Nah, supaya tubuh kamu tetap fit, kamu butuh antioksidan dan anti-aging untuk meremajakan kulit sel dan juga meningkatkan kualitas kesehatan tubuh dan kulit kamu. Nah, solusinya adalah dengan Dr. Water Hydrogen yang mampu merubah air biasa menjadi air hidrogen. Nah, katanya nih super antioksidan bisa 176 kali lebih banyak dari vitamin C karena bisa membantu kamu untuk melancarkan peredaran darah, menurunkan risiko penyakit jantung, stroke, terapi diabetes, dan juga kanker. Dan dapat juga digunakan sebagai inhaler untuk proses regenerasi sel dan terapi yang lebih cepat. Oh iya, paling penting nih, aman dikonsumsi semua kalangan. Produksi ini dimiliki dan juga didistribusikan oleh PT Ultra Multi Sukses. Dan saya ingin mengajak kamu semua untuk menggunakan Dr. Water Hydrogen sebagai solusi kesehatan tubuh kamu. Nah, untuk info lebih lanjut bisa langsung cek di website berikut ini.
Indonesia memiliki mimpi besar menjadi Indonesia maju di tahun 2045. Sekolah Pasca Sarjana Universitas Airlangga memiliki 8 program studi multidisiplin, hadir menjadi institusi berkelas dunia dengan kemampuan untuk mengembangkan pemimpin-pemimpin transformatif yang berdampak bagi dunia. Selamat datang di Sekolah Pasca Sarjana Universitas Airlangga, dan inilah program studi yang kami kelola.
Saya Profesor Dr. Teresia Indabudi sebagai Koordinator Program Studi Magister Immunologi Sekolah Pasca Sarjana Universitas Airlangga. Prodi Immunologi merupakan satu-satunya program studi Magister Immunologi di Indonesia. Prodi kami ini adalah multidisiplin, jadi bisa dari berbagai keilmuan. Sekolah-sekolah yang berkaitan dengan imunologi bisa mengikuti program studi imunologi di Universitas Airlangga. Kami punya kompetensi yaitu bahwa mahasiswa-mahasiswa yang mengikuti program studi imunologi ini bisa langsung terjun di dalam penelitian-penelitian yang mengikuti program dari penelitian dosen-dosen yang ada. Sehingga mahasiswa semakin mempunyai wawasan yang semakin banyak di bidang imunologi. Dan juga kami juga menyelenggarakan salah satu seminar yang kita sebut sebagai World Immunology Day, merupakan satu-satunya juga seminar yang kita selenggarakan baik itu tingkat nasional maupun internasional.
Kita disupport dengan adanya penelitian berpayung, pendanaan yang support luar biasa dari Pasca Sarjana Universitas Airlangga. Kita juga sebagai mahasiswa mendapat support untuk publikasi dan juga melalui pelatihan-pelatihan berkelas Scopus dari Q1 sampai Q4.
Karena imunologi itu banyak bertanya, itu PR-nya mahasiswa online harus lebih aktif. Dibandingkan kalau mahasiswa offline, dosen ada di depan kita dan kita tahu, oh seperti ini, oh seperti ini.
Magister Immunologi Pasca Sarjana Universitas Airlangga, hebat!
Ini dia alat yang bisa bikin kulit kamu makin sehat dan cantik. Namanya Dr. Firms Hydro Nano. Alat yang bisa mengeluarkan air dalam ukuran nano berbentuk spray. Ini adalah yang pertama di Indonesia karena terdapat 3 batuan mineral yang berfungsi sebagai antiinflamasi dan antibakteri. Bukan cuma air biasa, tapi ini air hidrogen yang mampu menyerap ke dalam kulit sehingga membuat kulit jadi terasa kencang dan elastis. Produk ini dapat digunakan untuk seluruh bagian wajah, tubuh, kulit kepala, dan bagian perawatan tubuh yang lain. Manfaat Dr. Firms Hydro Nano ini bisa untuk mencerahkan wajah dan membantu mencegah penuaan dini, membantu menghilangkan jerawat dan menghilangkan flek pada wajah, dan juga juga meningkatkan elastisitas kulit. Selain untuk menyehatkan dan membuat kulit jadi cerah, alat ini bisa jadi powerbank juga loh. Menarik kan? Buat kamu yang mau kulitmu cantik dan sehat, yuk langsung aja tanya ke Instagram @drwaterofficial.id.
Sibuk amat kakak cantik. Lagi ngerjain apa sih, Neng? Lagi ngerjain tugas nih. Kamu dari mana? Dari rumah. Daripada gak ada kuliah, lebih baik main di kampus. Tahu nggak? Ternyata autoimun itu sedang tren loh di anak muda. Katanya gara-gara gaya hidup. Oh iya? Iya, bener. Katanya makanan kekinian itu penyumbang utama terjadinya autoimun. Oh iya? Oh ya doang dari tadi juga dikasih tahu. Nah, selesai. Kenapa? Sehat. Sambut kali. Ayo, ikut aku. Mau mana? Udah, ikut aja. Di sini? Iya, di sini. Mau ngapain? Udah, daripada katanya sosmed, mending ikut kuliah tamu imunologi. Yuk.
Baik, hari ini kita akan bahas topik tentang autoimun. Kasus kanker itu juga merupakan salah satu dari autoimun, ya. Nah, dan apa penyebabnya dari autoimun? Dari apa yang sudah saya jelaskan tadi, ada yang mau ditanyakan kira-kira?
Ini kan membahas tentang autoimun. Nah, kalau misalnya tentang depresi, ada tidak hubungannya dengan autoimun? Makasih, Prof.
Cocok sekali ya, kebetulan di kurikulum kami ada kurikulum tentang psikoneuroimunologi. Jadi keterkaitan antara sistem imun dengan psikis.
Izin Prof bertanya, nama saya Astika Jinansari. Apa benar Prof, makanan itu penyebab autoimun? Terima kasih.
Makanan itu apakah bisa merupakan salah satu penyebab autoimun ya? Bisa. Ya kenapa? Karena saat ini makanan-makanan modern sekarang ya, itu banyak yang artifisial.
Seru juga ya belajar imunologi. Bener banget, masuk imunologi aja. Lagian karena ini multidisiplin ilmu, bisa dari beragam keilmuan kok, nggak harus melulu kesehatan.
Bisa jadi bidang kesehatan bukan passion saya, tapi dunia imunologi begitu menggoda. Tidak hanya tentang keilmuan yang luar biasa atau karena multidisiplinnya, tapi dengan imunologi, aku, kamu, dan kita bisa berbuat yang terbaik untuk orang-orang tercinta.
Indonesia memiliki mimpi besar menjadi Indonesia maju di tahun 2045. Sekolah Pasca Sarjana Universitas Airlangga memiliki 8 program studi multidisiplin, hadir menjadi institusi berkelas dunia dengan kemampuan untuk mengembangkan pemimpin-pemimpin transformatif yang berdampak bagi dunia. Selamat datang di Sekolah Pasca Sarjana Universitas Airlangga, dan inilah program studi yang kami kelola.
Saya Profesor Dr. Teresia Indabudi sebagai Koordinator Program Studi Magister Immunologi Sekolah Pasca Sarjana Universitas Airlangga. Prodi Immunologi merupakan satu-satunya program studi Magister Immunologi di Indonesia. Prodi kami ini adalah multidisiplin, jadi bisa dari berbagai keilmuan. Sekolah-sekolah yang berkaitan dengan imunologi bisa mengikuti program studi imunologi di Universitas Airlangga. Kami punya kompetensi yaitu bahwa mahasiswa-mahasiswa yang mengikuti program studi imunologi ini bisa langsung terjun di dalam penelitian-penelitian yang mengikuti program dari penelitian dosen-dosen yang ada. Sehingga mahasiswa semakin mempunyai wawasan yang semakin banyak di bidang imunologi. Dan juga kami juga menyelenggarakan salah satu seminar yang kita sebut sebagai World Immunology Day, merupakan satu-satunya juga seminar yang kita selenggarakan, baik itu tingkat nasional maupun internasional.
Kita disupport dengan adanya penelitian berpayung, pendanaan yang support luar biasa dari Pasca Sarjana Universitas Airlangga. Kita juga sebagai mahasiswa mendapat support untuk publikasi dan juga melalui pelatihan-pelatihan berkelas fokus dari Q1 sampai Q4.
Karena imunologi itu banyak bertanya, itu PR-nya mahasiswa online harus lebih aktif. Dibandingkan kalau mahasiswa offline, dosen ada di depan kita dan kita tahu, oh seperti ini, oh seperti ini.
Magister Imunologi Pasca Sarjana Universitas Airlangga, hebat!
Biotechnology is located in the beautiful and fast-developing Wuhan. ELK is a high-tech biological company specializing in immune and molecular biology research. We now own 1,600 square meter laboratory, having independent cell culture room and SPF animal room. We have 4 major platforms: pathology, immunology, molecular biology, cell biology. Our research focuses on immunochemistry, tumor research, neurobiology, cell cycle, signal transduction. We have 8,000 plus ELISA keys, 60 plus molecular biology reagents, 11,000 plus antibodies. We have certificated with ISO and CE mark. Each product has complete R&D, production and QC course, experienced R&D technical team, complete quality inspection support department, strict inspection process, providing customers with reliable quality scientific research products. We have fast delivery chain by FedEx or DHL. Since the company started overseas markets in 2018, we have reached a stable cooperation with distributors coming from more than 50 countries and 5 continents. Our goal is to provide scientists with the most cutting-edge and cost-effective research tools. Our mission is to provide the best solution and the most reliable partners for researchers all over the world. We will always implement the concept of environmental protection in the production process.
Hai friends, perlu kamu ketahui bahwa radikal bebas dan polusi udara saat ini sulit untuk bisa dihindari. Nah, dampaknya bisa membuat kulit kering, kusam, dan penuaan dini. Nah, supaya tubuh kamu tetap fit, kamu butuh antioksidan dan anti-aging untuk meremajakan kulit sel. Dan juga meningkatkan kualitas kesehatan tubuh dan kulit kamu. Nah, solusinya adalah dengan Dr. Water Hydrogen yang mampu merubah air biasa menjadi air hidrogen. Nah, katanya nih super antioksidan bisa 176 kali lebih banyak dari vitamin C. Karena bisa membantu kamu untuk melancarkan peredaran darah, menurunkan risiko penyakit jantung, stroke, terapi diabetes, dan juga kanker. Dan dapat juga digunakan sebagai inhaler untuk proses regenerasi sel dan terapi yang lebih cepat. Oh iya, paling penting nih, aman dikonsumsi semua kalangan. Produksi ini dimiliki dan juga didistribusikan oleh PT Ultra Multisukses. Dan saya ingin mengajak kamu semua untuk menggunakan Dr. Water Hydrogen sebagai solusi kesehatan tubuh kamu. Nah, untuk info lebih lanjut bisa langsung cek di website berikut ini.
Indonesia memiliki mimpi besar menjadi Indonesia maju di tahun 2045. Sekolah Pascasarjana Universitas Airlangga memiliki 8 program studi multidisiplin, hadir menjadi institusi berkelas dunia dengan kemampuan untuk mengembangkan pemimpin-pemimpin transformatif yang berdampak bagi dunia. Selamat datang di Sekolah Pascasarjana Universitas Airlangga, dan inilah program studi yang kami kelola.
Saya Profesor Dr. Teresia Indabudi sebagai Koordinator Program Studi Magister Immunologi Sekolah Pascasarjana Universitas Airlangga. Prodi Immunologi merupakan satu-satunya program studi Magister Immunologi di Indonesia. Prodi kami ini adalah multidisiplin, jadi bisa dari berbagai keilmuan. Sekolah-sekolah yang berkaitan dengan imunologi bisa mengikuti program studi imunologi di Universitas Airlangga. Kami punya kompetensi, yaitu bahwa mahasiswa-mahasiswa yang mengikuti program studi imunologi ini bisa langsung terjun di dalam penelitian-penelitian yang mengikuti program dari penelitian dosen-dosen yang ada. sehingga mahasiswa semakin mempunyai wawasan yang semakin banyak di bidang imunologi. Dan juga kami juga menyelenggarakan salah satu seminar yang kita sebut sebagai World Immunology Day, merupakan satu-satunya juga seminar yang kita selenggarakan, baik itu tingkat nasional maupun internasional.
Kita disupport dengan adanya penelitian berpayung, pendanaan yang support luar biasa dari Pasca Sarjana Universitas Airlangga. Kita juga sebagai mahasiswa mendapat support untuk publikasi dan juga melalui pelatihan-pelatihan berkelas Scopus dari Q1 sampai Q4.
Karena imunologi itu banyak bertanya, itu PR-nya mahasiswa online harus lebih aktif dibandingkan kalau mahasiswa offline, dosen ada di depan kita dan kita tahu, oh seperti ini, oh seperti ini.
Magister Imunologi Pasca Sarjana Universitas Airlangga, hebat!
Ini dia alat yang bisa bikin kulit kamu makin sehat dan cantik. Namanya Dr. Forms Hydro Nano. Alat yang bisa mengeluarkan air dalam ukuran nano berbentuk spray. Ini adalah yang pertama di Indonesia karena terdapat 3 batuan mineral yang berfungsi sebagai antiinflamasi dan antibakteri. Bukan cuma air biasa, tapi ini air hidrogen yang mampu menyerap ke dalam kulit sehingga membuat kulit jadi terasa kencang dan elastis. Produk ini dapat digunakan untuk seluruh bagian wajah, tubuh, kulit kepala, dan bagian perawatan tubuh yang lain. Manfaat Dr. Form Hydro Nano ini bisa untuk mencerahkan wajah dan membantu mencegah penuaan dini, membantu menghilangkan jerawat dan menghilangkan flek pada wajah, dan juga meningkatkan elastisitas kulit. Selain untuk menyehatkan dan membuat kulit jadi cerah, alat ini bisa jadi powerbank juga loh. Menarik kan? Buat kamu yang mau kulitmu cantik dan sehat, yuk langsung aja tanya ke Instagram @drwaterofficial.id.
Sibuk amat kakak cantik. Lagi ngerjain apa sih, Neng? Lagi ngerjain tugas nih. Kamu dari mana? Dari rumah. Daripada gak ada kuliah, lebih baik main di kampus. Tahu nggak? Ternyata autoimun itu sedang tren loh di anak muda. Katanya gara-gara gaya hidup. Oh iya? Iya, bener. Katanya makanan kekinian itu penyumbang utama terjadinya autoimun. Oh iya? Oh ya doang dari tadi juga dikasih tahu. Nah, selesai. Kenapa? Sehat. Sambut kali. Ayo, ikut aku. Mau mana? Udah, ikut aja. Di sini? Iya, di sini. Mau ngapain? Udah, daripada katanya sosmed, mending ikut kuliah tamu imunologi. Yuk.
Baik, hari ini kita akan bahas topik tentang autoimun. Kasus kanker itu juga merupakan salah satu dari autoimun, ya. Nah, dan apa penyebabnya dari autoimun? Dari apa yang sudah saya jelaskan tadi, ada yang mau ditanyakan kira-kira?
Ini kan membahas tentang autoimun. Nah, kalau misalnya tentang depresi, ada tidak hubungannya dengan autoimun? Makasih, Prof.
Cocok sekali ya, kebetulan di kurikulum kami ada kurikulum tentang psikoneuroimunologi. Jadi keterkaitan antara sistem imun dengan psikis.
Izin Prof bertanya,
Nama saya Astika Juniansari. Apa benar, Prof, makanan itu penyebab autoimun? Terima kasih.
Makanan itu apakah bisa merupakan salah satu penyebab autoimun ya? Bisa. Ya kenapa? Karena saat ini makanan-makanan modern sekarang ya, itu banyak yang artifisial.
Seru juga ya belajar imunologi. Bener banget, masuk imunologi aja. Lagian karena ini multidisiplin ilmu, bisa dari beragam keilmuan kok, nggak harus melulu kesehatan.
Bisa jadi bidang kesehatan bukan passion saya, tapi dunia imunologi begitu menggoda. Tidak hanya tentang keilmuan yang luar biasa atau karena multidisiplinnya, tapi dengan imunologi, aku, kamu, dan kita bisa berbuat yang terbaik untuk orang-orang tercinta.
Indonesia memiliki mimpi besar menjadi Indonesia maju di tahun 2045. Sekolah Pascasarjana Universitas Airlangga memiliki 8 program studi multidisiplin, hadir menjadi institusi berkelas dunia dengan kemampuan untuk mengembangkan pemimpin-pemimpin transformatif yang berdampak bagi dunia. Selamat datang di Sekolah Pascasarjana Universitas Airlangga, dan inilah program studi yang kami kelola.
Saya Profesor Dr. Teresia Indabudi sebagai Koordinator Program Studi Magister Immunologi Sekolah Pascasarjana Universitas Airlangga. Prodi Immunologi merupakan satu-satunya program studi Magister Immunologi di Indonesia. Prodi kami ini adalah multidisiplin, jadi bisa dari berbagai keilmuan. Sekolah-sekolah yang berkaitan dengan imunologi bisa mengikuti program studi imunologi di Universitas Airlangga. Kami punya kompetensi yaitu bahwa mahasiswa-mahasiswa yang mengikuti program studi imunologi ini bisa langsung terjun di dalam penelitian-penelitian yang mengikuti program dari penelitian dosen-dosen yang ada, sehingga mahasiswa semakin mempunyai wawasan yang semakin banyak di bidang imunologi. Dan juga kami juga menyelenggarakan salah satu seminar yang kita sebut sebagai World Immunology Day, merupakan satu-satunya juga seminar yang kita selenggarakan, baik itu tingkat nasional maupun internasional.
Kita disupport dengan adanya penelitian berpayung, pendanaan yang support luar biasa dari Pascasarjana Universitas Airlangga. Kita juga sebagai mahasiswa mendapat support untuk publikasi dan juga melalui pelatihan-pelatihan berkelas fokus dari Q1 sampai Q4.
Karena imunologi itu banyak bertanya, itu PR-nya mahasiswa online harus lebih aktif. Dibandingkan kalau mahasiswa offline, dosen ada di depan kita dan kita tahu, oh seperti ini, oh seperti ini.
Magister Immunologi Pascasarjana Universitas Airlangga, hebat!
Biotechnology is located in the beautiful and fast-developing Wuhan. ELK is a high-tech biological company specializing in immune and molecular biology research. We now own 1,600 square meter laboratory having independent cell culture room and SPF animal room. We have 4 major platforms. Pathology, immunology, molecular biology, cell biology. Our research focuses on immunochemistry, tumor research, neurobiology, cell cycle, signal transduction. We have 8,000 plus ELISA keys, 60 plus molecular biology reagents, 11,000 plus antibodies. We have certificated with ISO and CE mark. Each product has complete R&D, production and QC course, experienced R&D technical team, complete quality inspection support department, strict inspection process, providing customers with reliable quality scientific research products. We have fast delivery chain by FedEx or DHL. Since the company started overseas markets in 2018, we have reached stable cooperation with distributors coming from more than 50 countries and 5 continents. Our goal is to provide scientists with the most cutting-edge and cost-effective research tools. Our mission is to provide the best solution and the most reliable partners for researchers all over the world. We will always implement the concept of environmental protection in the production process.
perlu kamu ketahui bahwa radikal bebas dan polusi udara saat ini sulit untuk bisa dihindari. Nah, dampaknya bisa membuat kulit kering, kusam, dan penuaan dini. Nah, supaya tubuh kamu tetap fit, kamu butuh antioksidan dan anti-aging untuk meremajakan kulit sel. Dan juga meningkatkan kualitas kesehatan tubuh dan kulit kamu. Nah, solusinya adalah dengan Dr. Water Hydrogen yang mampu merubah air biasa menjadi air hidrogen. Nah, katanya nih super antioksidan bisa 176 kali lebih banyak dari vitamin C karena bisa membantu kamu untuk melancarkan peredaran darah, menurunkan risiko penyakit jantung, stroke, terapi diabetes, dan juga kanker. Dan dapat juga digunakan sebagai inhaler untuk proses regenerasi sel dan terapi yang lebih cepat. Oh iya, paling penting nih, aman dikonsumsi semua kalangan. Produksi ini dimiliki dan juga didistribusikan oleh PT Ultra Multi Sukses. Dan saya ingin mengajak kamu semua untuk menggunakan Dr. Water Hydrogen sebagai solusi kesehatan tubuh kamu. Nah, untuk info lebih lanjut bisa langsung cek di website berikut ini.
Indonesia memiliki mimpi besar menjadi Indonesia maju di tahun 2045. Sekolah Pascasarjana Universitas Airlangga memiliki 8 program studi multidisiplin, hadir menjadi institusi berkelas dunia dengan kemampuan untuk mengembangkan pemimpin-pemimpin transformatif yang berdampak bagi dunia. Selamat datang di Sekolah Pascasarjana Universitas Airlangga, dan inilah program studi yang kami kelola.
Saya Profesor Dr. Teresia Indabudi sebagai Koordinator Program Studi Magister Immunologi Sekolah Pascasarjana Universitas Airlangga. Prodi Immunologi merupakan satu-satunya program studi Magister Immunologi di Indonesia. Prodi kami ini adalah multidisiplin, jadi bisa dari berbagai keilmuan. Sekolah-sekolah yang berkaitan dengan imunologi bisa mengikuti program studi imunologi di Universitas Airlangga. Kami punya kompetensi, yaitu bahwa mahasiswa-mahasiswa yang mengikuti program studi imunologi ini bisa langsung terjun di dalam penelitian-penelitian yang mengikuti program dari penelitian dosen-dosen yang ada, sehingga mahasiswa semakin mempunyai wawasan yang semakin banyak di bidang imunologi. Dan juga kami juga menyelenggarakan salah satu seminar yang kita sebut sebagai World Immunology Day, merupakan satu-satunya juga seminar yang kita selenggarakan, baik itu tingkat nasional maupun internasional.
Kita disupport dengan adanya penelitian berpayung, pendanaan yang support luar biasa dari Pascasarjana Universitas Airlangga. Kita juga sebagai mahasiswa mendapat support untuk publikasi dan juga melalui pelatihan-pelatihan berkelas Scopus dari Q1 sampai Q4.
Karena imunologi itu banyak bertanya, itu PR-nya mahasiswa online harus lebih aktif. Dibandingkan kalau mahasiswa offline, dosen ada di depan kita dan kita tahu, oh seperti ini, oh seperti ini.
Magister Immunologi Pascasarjana Universitas Airlangga, hebat!
Ini dia alat yang bisa bikin kulit kamu makin sehat dan cantik. Namanya Dr. Firms Hydro Nano. Alat yang bisa mengeluarkan air dalam ukuran nano berbentuk spray. Ini adalah yang pertama di Indonesia karena terdapat 3 batuan mineral yang berfungsi sebagai antiinflamasi dan anti antibakteri. Bukan cuma air biasa, tapi ini air hidrogen yang mampu menyerap ke dalam kulit sehingga membuat kulit jadi terasa kencang dan elastis. Produk ini dapat digunakan untuk seluruh bagian wajah, tubuh, kulit kepala, dan bagian perawatan tubuh yang lain. Manfaat Dr. Form Hydro Nano ini bisa untuk mencerahkan wajah dan membantu mencegah penuaan dini, membantu menghilangkan jerawat dan menghilangkan flek pada wajah, dan juga meningkatkan elastisitas kulit. Selain untuk menyehatkan dan membuat kulit jadi cerah, alat ini bisa jadi powerbank juga loh. Menarik kan? Buat kamu yang mau kulitmu cantik dan sehat, yuk langsung aja tanya ke Instagram @drwaterofficial.id.
Sibuk amat kakak cantik. Lagi ngerjain apa sih, Neng? Lagi ngerjain tugas nih. Kamu dari mana? Dari rumah. Daripada gak ada kuliah, lebih baik main di kampus. Tahu nggak? Ternyata autoimun itu sedang tren loh di anak muda. Katanya gara-gara gaya hidup. Oh iya? Iya, bener. Katanya makanan kekinian itu penyumbang utama terjadinya autoimun. Oh iya? Oh ya doang dari tadi juga dikasih tahu. Nah, selesai. Ih, kenapa? Sehat. Sambut kali. Ayo, ikut aku. Mau mana? Udah, ikut aja. Di sini? Iya, di sini. Mau ngapain? Udah, daripada katanya sosmed, mending ikut kuliah tamu imunologi. Yuk.
Baik, hari ini kita akan bahas topik tentang autoimun. Kasus kanker itu juga merupakan salah satu dari autoimun, ya. Nah, dan apa penyebabnya dari autoimun? Dari apa yang sudah saya jelaskan tadi, ada yang mau ditanyakan kira-kira?
Ini kan membahas tentang autoimun. Nah, kalau misalnya tentang depresi itu ada tidak hubungannya dengan autoimun? Makasih, Prof.
Cocok sekali ya, kebetulan di kurikulum kami ada kurikulum tentang psikoneuroimunologi. Jadi keterkaitan antara sistem imun dengan psikis.
Izin Prof bertanya, nama saya Astika Juniansari. Apa benar Prof, makanan itu penyebab autoimun? Terima kasih.
Makanan itu apakah bisa merupakan salah satu penyebab autoimun ya? Bisa. Ya kenapa? Karena saat ini makanan-makanan modern sekarang ya, itu banyak yang artifisial.
Seru juga ya belajar imunologi. Bener banget, masuk imunologi aja. Lagian karena ini multidisiplin ilmu, bisa dari beragam keilmuan kok, nggak harus melulu kesehatan.
Bisa jadi bidang kesehatan bukan passion saya, tapi dunia imunologi begitu menggoda. Tidak hanya tentang keilmuan yang luar biasa atau karena multidisiplinnya, tapi dengan imunologi, aku, kamu, dan kita bisa berbuat yang terbaik untuk orang-orang tercinta.
Indonesia memiliki mimpi besar menjadi Indonesia maju di tahun 2045. Sekolah Pascasarjana Universitas Airlangga memiliki 8 program studi multidisiplin hadir menjadi institusi berkelas dunia dengan kemampuan untuk mengembangkan pemimpin-pemimpin transformatif yang berdampak bagi dunia. Selamat datang di Sekolah Pascasarjana Universitas Airlangga, dan inilah program studi yang kami kelola.
Saya Profesor Dr. Teresia Indabudi sebagai Koordinator Program Studi Magister Immunologi Sekolah Pasca Sarjana Universitas Airlangga. Prodi Immunologi merupakan satu-satunya program studi Magister Immunologi di Indonesia.
All right, participants. So as we wait for the final results from the panel of judges to be announced, we invite you all offline and online participants to come and join this quiz that we will do together. Now, is there a reward for those who join? Well, technically, yes. If you are among the top 3 or top 5, if you are a winner, you will definitely get a door prize. So please feel free to go to quizzes.com, join as a participant and input the code. Oh, yes, there we go. Or you can scan the code directly, or you can go directly to quizzes.com and input the code 354278. We will play this quiz together. What is the quiz about? I have no idea. In fact, this is the first time I'm looking at the quiz, in fact. All right, so we've got 8 participants already. Those who are joining online can also join this quiz that has been set by the committee. In fact, again, if you are among the top 3 or top 5, we'll see later on, you will be able to get a door prize. And just to remind you, there are so many door prizes that we have. Some of them ranging from vouchers to also products from our amazing sponsors. We have 18 participants. If I stand here, can you guys see the screen? Nggak kelihatan ya? Okay, I'll stand here then. Okay, we have 19 participants. Is there anyone else here who would like to join? In fact, the committee, you are also welcome to join if you want. 20 participants. Again, those joining online can also join us in this quiz together. Let's play together. All right. Okay, 21 participants. Should we wait longer? 2 minutes, 2 more minutes, and then we will start. So anyone else? Okay, come join this quiz. You will be able to win a prize in the end. You know, I just realized on this side of the room, there's a lot of purple heads here. I like the uniform. I just realized it now, by the way. Anyway, even the staff members of Pasca Sarjana, you guys can join the quiz as well. I will, in fact, join the quiz, but I'm pretty sure I won't be able to answer any of the questions. 28 participants. Looks like we've got some activity also. 30 participants. Wow, it's getting 32. It's getting crowded. Huh? Wait. 10, 9, 8. 32 participants. Should we wait longer? Mr. Technical, Mr. Game Master? 1 more minute. Okay. Thirty-two participants. Anyone else want to join? Come on, join. Those online can also join this quiz. If you win and you are an online participant, can figure out how you can get the door prize later on. That's for later to decide. But the most important thing is that you join the quiz now. Okay, let's start the quiz with 33 participants. Game Master, let us start. Yes, dimulai. It's starting. 1, go! The first question. Loh. Oh. Oh alah. Oh gitu. I thought the questions would be shown on the screen. Okay, wow, wow. Okay. We've got people already. Top 3. Some with a 2 strike. Wah, wah, wah. No working together. Wow, Wims. 4 times strike. What is the quiz about, by the way, Game Master? Can you explain to us what the quiz is about?
The quizzes is about today's conference and about our immunology program study.
Ah, so it's a mixed.
Yeah.
Oh, and what time did you make these questions?
It's there are several questions with 20 seconds and there's so many question with only 10 seconds.
Would you say that they are hard?
Not really, but just a few occasion it needs the immunology knowledge. So I think it
okay.
will be harder for people who doesn't know about immunology.
Okay. Wow, we've got someone with 8 streak already. Wow, congratulations, Helen. It's now a race between R and Helen Magu. How many questions are there? 20 or 15? 15. 15, not 50, right? 9 times trick. Wow, okay. You are thinking, but you are losing at the same time. So come on, catch up. You guys must be smart. I mean, I will join the quiz, but I am still an undergrad student studying dentistry. In fact, Prof. Teresi is actually one of my lecturers also who teaches me oral pathology, which is an exciting course. 8 times strike, okay. 9 times strike, but it's broken, I think, from the red. 9 times strike, okay. 2 people, Calvin and Faris, good job. You guys are doing great. R, 3 times strike, nice. 7 times strike also. It's a tight race. Now R is in the lead with 12,000 points. Who is R here? Who is a R? you are you R? Man, okay, nice. that's okay. If they're not moving anymore, does that mean they're finished? Oh, okay. Can you scroll down and see how many more are? Let's see who's in the bottom. Look, let's see who's on the bottom. Okay. That's okay, guys. Christoph and Nia Ardelia, guys, come on, guys. You guys are still at 0. Whoever and wherever you are. Nice. Okay. Let's move back up. Let's move back up and let's see who's on the top 5-ish. Oh, R is still holding the lead with 12,000 points. And Safira is actually close by with 70 points left. Is there still other people who haven't finished? Game Master?
Pisa? Huh? Still, the bar is indicate the progress of someone.
Okay. Visa, come on, you can do it. Let's finish this. Visa, where are you? But you know what? Scroll back up, Game Master. Yes. Bro, scroll back up, bro. Thanks.
Okay.
Yeah. Okay, so it looks like the top 7-ish people haven't changed at all. So who should, from these people, who should get the door prize? The top 3 or top 5 or top 7 or top 10? Top 10?
The top 6 will get the reward.
The top 6.
Yeah.
Can it be 7? Because 7 is a much nicer number than 6.
Oh, we can talk for our treasurer.
Treasurer? Treasurer?
Miss Intan.
Miss Intan, where are you? 6 or 7 people?
6 or 7, Miss Intan.
6.
Oh,
Okay. When the treasurer says 6 and it's final, okay, it's final decision. Okay, so we've got this first 6 people who've maintained the leaderboard, and we see that there isn't any change, so we can close the quiz. So for R, Safira, Calvin, Helen Mago, Mary, and Felia, perhaps the committee can take note of these people. And if you are one of those people, screenshot your quizzes for evidence. And later on, please contact the committee in order to receive the door prize at the end of the conference. Reminding again, we have many interesting door prizes, right?
Maybe what they want to look at the question and the key answer.
Okay.
So the question is just like for number 1, who's the head of Immunology Study Program Universitas Airlangga? I think this is so easy since we are all know that Prof. Tere is the head
Yes,
of
that
Immunology
is
Study.
correct.
Okay.
But I'm curious, can we see how many people got that wrong?
2
people. Okay. That's still okay. That's still okay. Masih bisa diterima nggih, Prof? Okay, let's move on to the next question.
I think the number 2 is a little bit harder because
it's a bit of history as well.
The answer is 1993.
Really?
Yeah.
Wow. And this is the first time I'm also learning about it. And I'm a future immunology student here at Universitas Airlangga. So let me finish my undergrad first. One step at a time. Okay, number 3. Which of the following colors is
So Universitas Airlangga Immunology.
purple?
Yes.
Oh, no wonder you guys are wearing purple.
And then
one of the scope of immunology is physiotherapy. Is it correct?
False.
It's false. Why is it? Explain to me.
Because it's more like a therapy, not focusing on immunology.
Okay. Noted. SARS-CoV-2 was being epidemic only in Indonesia and Southeast Asia. Is this a question from the conference?
Not really. It's based on our real life.
Okay.
So the answer is false because it was being a pandemic worldwide. So it's not epidemic, but pandemic worldwide.
Okay. Interesting.
Next is about allergy. Of course, the answer is true. Name of a virus which causes immunodeficiency. The answer is HIV. Based on decree, or maybe this is the newest or the latest announcement about our immunology program study.
Bro.
So the answer is unggul. Excellent. Can we give applause for immunology program study?
That's a no-brainer. That everyone should have chosen unggul, right?
Yeah.
Did anyone choose besides unggul? How many people chose? 6.
Okay, maybe they didn't know about our latest news
yes.
about immunology program.
Mindset. It's all about mindset.
The next is Sekolah Pasca Sarjana is located in
Tunjungan Plaza.
No. The answer is Unir B, but one person got it wrong. I don't know.
Pretty sure they answered Tunjungan Plaza.
Okay, the next is which one of these diseases is caused by virus? The answer is influenza.
So the rest are bacteria?
Diarrhea it can be microbial. Malaria is parasitic. Ascariasis is about worm or touching parasite. Number 11. Which of the following brand is not COVID-19 vaccine? We got the wrong one is Uniqlo. Because Uniqlo is fashion brand.
Which one of you is the 5 people who chose Uniqlo? I mean, hey, you can't argue Uniqlo is a good clothing brand. For 200K, the shirt is nice.
Okay. Number 12, which of the following cellular components that not associated with immune system? Maybe this is the hardest part of this quiz because it's to immunology, and the answer is Schwann cell.
Yes.
13, one of our moderator today is Miss Salma Zulkaida, SSEMSC PhD.
Who got that wrong?
And this was wrong because our moderator today is Mrs. Vivin and Mrs. Ayu.
There's only two of them. How can you get that wrong? How many people got that wrong?
15.
It's okay. Mindset.
Okay, number 14, Professor Dr. Cita Rosita Sigit Prakoswa, doctor, is the director of which hospital? The answer is Sutomo Hospital.
Yes, that's a no-brainer.
And the last one is, what is produced by B cell? The answer is antibody.
Wow. You know, I feel like 90% of these questions are immunology-based questions and not from the conference. You even admit it. Wow. You know, even as a student, as an undergrad student who's still studying dental medicine, this is actually all very menarik sekali. And here we have our top three people. We have Aur, Safira, and Calvin. But for the people who are going to receive the door prize, it will be the top 6 people. Oh, yeah, I should. Yeah, there we go. The people who will receive the door prize are the top 6 people. So please don't forget to screenshot your result on the quizzes and then contact the committee to receive the door prize. Operator and committee, are we ready to announce the results? All right. So before that, as you know, the 4th International Conference of the World Immunology Day 2025 has been sponsored by ScienceWork, Kairos, BPE, and Welo. And previously, we have already shown the video and we have already invited the representatives of ScienceWork and Kairos. Now we would like to invite the representatives of Biotech Prima, Indublas, and Welo to present their company videos and also products. So to the committee in charge, the time and place is now yours to share screen their company videos.
Hai friends, perlu kamu ketahui bahwa radikal bebas dan polusi udara saat ini sulit untuk bisa dihindari. Nah, dampaknya bisa membuat kulit kering, kusam, dan penuaan dini. Nah, supaya tubuh kamu
PT Biotek Prima India Plus Laboratory Equipments and Reagents. PT Indolab Utama Jakarta is the head office which has two sub-distributors. The first is PT Biotek Prima India Plus for East Java area. The second is PT Mitra Utama Indolab for West Java area. PT Biotek Prima India Plus is located in Ruko Sentra Niaga Citra Garden Block RE number 10 Sidoarjo. This company is open every day from Monday until Friday at 8 a.m. until 5 p.m. This company offers various fields of product. The first is cell biology. The second is nucleic acid analysis. The third is human identification. The fourth is lab equipment and supplies. The fifth is protein analysis. And the last is molecular diagnostic. Here are the brands that we have.
Are you looking for a detection instrument that works right out of the box? With preloaded Promega assay protocols and an intuitive touchscreen interface, the GlowMax Systems gets you to your data faster without having to adjust or optimize instrument settings. Once the GlowMax Explorer or GlowMax Discover instrument is unpacked and plugged in, you are ready to start collecting data from your experiments. Easily study cell viability, genetic reporters, bioassays, and many more applications. Whether you are measuring luminescence, fluorescence, absorbance, or a combination of these, the GlowMax systems are able to offer reliable, quick, sensitive readings for your single or multiplexed assay. Promega reagents and GlowMax systems work together as one. Just press Start on your preloaded protocol. Explore our integrated solutions. Request a demo today and discover how quickly you get results.
Size is the only thing small about the Spectrum Compact CE system. With capabilities for Sanger sequencing and fragment analysis, the Spectrum Compact CE system has flexible run scheduling and an easy-to-use interface that empowers you to get your work done at your convenience. The plug-and-play, pre-filled consumables are easy to install and use 2D barcoding to track levels and expiration dates. The separate consumables give you more control and help minimize reagent waste. Once you've plugged in your consumables, use the integrated touchscreen to get started. The instrument uses the best available polymers for each application, and it can process up to 32 samples per run with flexible batch sizes. Gotta run? The remote access software allows you to edit, monitor, and download completed runs from any computer on the same network. Discover flexibility and efficiency at an affordable price point. Think Spectrum Compact.
Check. All right, ladies and gentlemen, the results are now in. It is time to announce the winners of the poster and oral presentation of the World Immunology Day 2025. We will be starting by announcing the top, for the poster first, the top 6 nominations, not necessarily in that order of winners, all right? So the top 6 posters, again, this is random. The top 6 posters are from Muhammad Amin, Felia Evelina, Dr. Septin Mauludiana, Lydia Fernanda, Salsabila Putri, and Merdiana Ayu. These are the top 6 poster nominations for the poster presentation. Now, From those 6 poster presentations, 3 people have shown significant results. 3 people have shown a very nice presentation by the judges. And so these 3 people who have won the first place, second place, and third place of the poster presentation are, for third place, we have Salsabila Putri Kinanti. And for second place, we have Lydia Fernanda. And for first place, we have Mardiana Ayu. All right. For the top 3 people, for Mardiana Ayu, Lydia Fernanda, and Salsabila Putri, please come forward. We would like to invite Professor Teresia as the Immunology Program Study Coordinator. We would like to invite Dr. Ayu to please come forward and present the award to our winners of the poster presentation. We congratulate these students from the Immunology Magister Program of Universitas Airlangga for winning the top 3.
But tasks are still going to be on both. Did nobody see my poppycock post? There was no clappy claps on it.
Place for the poster presentation competition. You guys have shown excellent resistance and you have dedicated excellent dedication in this competition. You have worked hard to be able to win first, second, and third place. So congratulations. Kudos on your achievement. We would like to, yes. We'd like to also invite Professor Teresia to come forward and take a group picture with the winners of the poster presentation. Perhaps for Miss Lydia, you can take off your mask for the group picture if you are comfortable with it. Unair hebat. Okay, thank you. May now return to your seats. Once again, congratulations to the winners of the poster presentation. Now, after the poster presentation, it is now time to announce the winners of the oral presentation. We've got 10 nominations. 10 nominations where we will choose 3 winners and also 2 runner-ups, if I'm correct. Okay, so the top 10 nominations of the oral presentation that I will state one by one, but not necessarily in that order of winning. First of all, we have Faris Rega, we have Venansi Victoria, we have Calix Suzozo. I am so sorry if I'm pronouncing your names wrong. We have Dr. Larasita, we have Dr. Helen, we have Calvin Konstantin, we have Armanda Dwi Prayugo, we have Ali Zainal, we have Brilliant Margalin, and we have Muhammad Ainun. So from these top 10 nominations, the top, first of all, we will announce here we are. Here are the winners of the oral presentation. So congratulations to, first of all, Dr. Brilliant Margalin for first place. Second place, we have Ali Zainal Abidin. For third place, we have Muhammad Ainun Najib. And for the first runner-up, we have Calvin Konstantin. And for the second runner-up, we have Armanda Dwi Prayugo. For the winners, please come forward. For this, we would like to invite the program study coordinator, Professor Teresia, to please come and present the award to our winners. For the other winners, please come forward to receive the prize. Once again, we convey our congratulations to our winners of the oral presentation of the Aralanga World Immunology Day 2025. You have worked hard to come to this position, and you do indeed deserve this winner. Once again, congratulations to our winners of the oral presentation. All right, for the top 10 nominations of the oral presentation, please come forward to take a group picture in front. For the top 10 nominations of the oral presentation, if you are in this room, please come forward to take a group picture in front. So we've got Brilliant Margalin, we have Ali Zainal, we have Muhammad Ainun Najib, we have Calvin Konstantin, we have Armanda Dwi Prayugo, we have Helen Cynthia, we have Lara Sita Prameswari, we have Venansi Victoria, we have Calix Siuzuzo, and we have Faris Rega. Please come forward to take a group picture with the program study coordinator of the immunology course of the postgraduate school of Universitas Airlangga. We also invite Congratulations to our top 10 oral presenters of the Erlangga World Immunology Day 2025. All right, ladies and gentlemen, the awards ceremony of the poster and oral presentation competition of the Erlangga World Immunology Day 2025 marks the end of today's conference and the day 2025. And on behalf of the entire organizing committee, we would like to thank you all for your enthusiastic participation in this conference and would like to apologize if throughout the conference there have been any mistakes or any mishaps that have been created that has inconvenienced you. I am Nabil Ahmad Wafiuddin, hereby as your MC bids you farewell. See you in the 5th International Immunology Day competition of 2026. See you next year, everyone. All right, ladies and gentlemen, let us take a group photo together outside in the photo booth. All right, to all the participants and the committee, let's all stand up and take a group photo session outside as our last memory together. Once again, we thank you for your participation in this conference. And we hope to see you next year. Once again, we invite all the participants and committee to come outside and take a group photo session together outside. Let's take one final memory together, and I hope to see you next year.